pas jam 8.30 kan mulai pembukaan. Ya, saya kira setengah jam siap lah kan.
Oke.
Ya nggak nampak gambarnya ya. Mungkin mereka yang desain barangkali nih, yang ya ya mereka desain nampaknya banyak orang gitu kan.
Mungkin juga lah.
Iya. Oke, sebagian nampak. Ibu
Jadi
Mas
kita nggak tahu siapa yang udah datang ya. Jadi karena nggak nampak ini, jadi kita nggak tahu siapa yang nampak udah.
namanya ada kan namanya?
Di mana lihat namanya? Nggak ada di sini.
Ada di masing-masing sebelah kanan ada namanya itu. Nama-nama yang hadir.
Aduh, aku nggak ngerti pula nih membuka-buka.
ada ada namanya kan. Namanya sudah ada. Ibu Mas Pria sudah masuk.
Ada.
Selamat pagi Ibu Mas Pria.
Selamat pagi Pak Hakim.
Ya, selamat pagi.
Saya masih
Ya.
tutup ini kameranya Pak Hakim.
Kamera saya terbuka kan?
Selamat pagi, Prof. Sehat kan?
Alhamdulillah sehat Pak. Sehat-sehat kita semua ya Pak.
Oh, itu yang penting.
Sehat-sehat orang tua, Pak.
Iyalah. Mungkin kita sehat orang muda kan?
Udah lansia semuanya ini.
Nggak mungkin sehat orang muda. Pasti orang tua. Harus menyadari itu.
Iya.
Supaya jangan lasak kali jalan-jalannya. Kaki harus dijaga.
Sehat masih bisa jalan-jalan ya.
Ya, itu dia. Harus pelan-pelan melangkah, apalagi melangkah ke tangga.
Apalagi kalau ada basah-basah sedikit, hati-hati.
Iya,
Jangan sampai tergelincir.
ya, basah dan kalau tidak rata. Saya lihat pengalaman
Betul.
orang tua itu banyak jalan tidak rata. Jadi meskipun dia
Betul.
10 cm ke bawah itu terasa apa? Jadi sakit. Dan sekali sudah kena kaki udah payah orang tua.
Iya. Apalagi sampai patah, udahlah nggak bergerak lagi.
Nggak bisa kembali lagi. Saya lihat ada beberapa itu. Nenek saya salah satu gitu.
Iya, saya juga itu saudara saya ada juga sampai sekarang udah setahun lebih di kursi roda, bisa lagi ya.
Dia turun dari tempat tidur, tapi dia tidak merasa bahwa dia sudah mau turun, nah, gitu.
Nah, itulah.
Padahal sudah turun gitu. Padahal berapa lah itu? Palingan nggak sampai 30 cm
Iya.
tidur dengan lantai itu. Jadi ini yang kedua, Prof. Hakim ya?
Iya, yang internasional yang kedua.
Yang pertama tahun berapa?
2002 tahun yang lalu, 2022.
Pas masa COVID itu lah itu ya?
Iya, pas masa COVID ya.
Ya.
Iya, kedua. Dua tahun sekali udah cukup lah barangkali kan.
Iya.
Tiap tahun sampai juga gitu. Tahun sekali kan. Dan ini pembicara yang lalu nggak boleh sama, mesti katanya dari USU nggak boleh sama.
Oh iya.
Kayaknya berbeda gitu orangnya gitu.
Iya.
Karena sekaligus memperkenalkan USU kan keluar gitu.
Iya betul.
Ini namanya nggak berurutan ya, jadi kalau masuk pembicara kita nggak tahu ya.
Iya.
Iya kan? Ibu Sumaya udah masuk kan? Selamat pagi Ibu Sumaya.
Selamat pagi Prof.
Iya, selamat pagi. Ya. Iya, kita nggak bisa tahu kan masa pembicara nanti kan belum nggak dicari-cari nggak nampak ini orangnya. Ini.
Ya.
Ya. Hmm. Sudah 176 ya yang hadir. Undang. Ya. Prof. Robert,
Kenapa?
kapan mau ke Jepang lagi?
Eh, Rencana tahun depan karena eh, akan menindaklanjuti MOU dengan Tohoku itu. Saya sudah 3 penelitian dengan Prof. Kimura Toshiaki, kata
Ya.
Prof. Dan FKG kan harus 2, nggak tahu kalau di tempat lain. Kalau untuk Tohoku harus sudah 2 melakukan kegiatan baru bisa membuat MOU.
Oh, dia ada di luar.
Ada peraturan mereka. Jadi waktu kami yang pergi itu hari itu ya belum belum bisa juga karena belum berlangsung. Jadi ini waktu dekan kedokteran gigi almarhumah Trelia itu kan sudah lakukan kegiatan dengan Prof. Sondang dengan yang sekarang. Kami dengan Prof. Hamzon sudah. Nah, setelah 2 fakultas ini melakukan kegiatan, nah, itu sudah memenuhi persyaratan untuk apa? Untuk buat MOU. Jadi saya sudah sampaikan ke dekan FIB supaya ditindaklanjuti ke apa? Ke biro rektor.
Iya, iya. Dan sekarang Tohoku nomor 1 ya sekarang di Jepang.
Tapi dia turun secara dunia turun.
Oh, di dunia turun ya?
Nah, kemarin itu dia mula-mula bekerja sama kita 79, sekarang udah 103.
Tapi di Jepang kok dia nomor 1 sekarang?
Iya, tapi memang dunia yang lain mungkin
Oh,
negara lain
ya.
yang naik. Nah,
Saya juga
kadang-kadang kan
terkejut.
gitu.
Bisa di lombanya Tokyo gitu.
Iya, Hokunya memang apa eh, luar biasa di Jepang ya, tapi kelihatannya universitas lain di dunia naik kan gitu.
Gitu ya, iya.
Itu dia.
Iya, syaratnya kan berat juga tuh ya. Duluan kerja apa? Mesti ada kerjasama dulu ya. Udah
Iya,
ada
iya,
kegiatan baru MOU ya.
iya. Dulu USM juga sempat begitu. Jadi apa MOU tidak untuk disimpan katanya. Tapi MOU itu sudah memayungi apa yang sudah ada dikerjakan gitu. Jadi bukan
Iya ya.
ah, jadi seringkali katanya MOU sudah tapi nggak ada kerjanya. Kemudian, nah ini mereka balik mungkin. Kebetulan kolega kami itu, partner kami selalu itu, Prof. Toshiaki Kimura sekarang jadi dekan. Kalau dulu kan dia hanya dosen biasa, profesor biasa. Sekarang dean dia.
Bidang sosial ya.
Iya, tapi dia kajiannya tentang apa? Apa ya? Lebih kepada keyakinan.
Dia PhD bukan?
Iya, PhD dia.
Karena bidang-bidang sosial kan jarang juga PhD kan di Jepang.
Iya, dia PhD.
PhD ya.
Karena umurnya pun masih seumur saya.
Oh,
Yang lama-lama mungkin yang senior mungkin seperti itu, tapi yang baru ini sudah PhD mereka.
udah PhD ya. Nah,
Hmm,
dulu
Selamat
susah
pagi,
kali itu
Prof.
bilang soal PhD.
iya.
Ini suara saya cukup jelas ya gini aja ya.
Bagus sekali, bagus.
Oh, cukup ya.
Ini pun
Oh,
bagus, Ibu apa tadi?
iya,
Eh,
iya. Nah,
Suara saya jelas ya, Bro?
Ibu
Selamat pagi.
pagi. Ah, cuman orangnya nggak tahu kita pagi, nggak bisa kelihatan.
Prof. Popi ini ya.
Ya Prof.
Iya,
Kopi.
Bro.
Ibu Arif.
Iya. Kok nggak muncul gambar apanya ya?
Hah? Gambarnya mana?
Gambarnya. Iya.
Ah,
Oh,
udah,
udah.
karena dimatikan.
Oh, udah muncul. Nah.
Bagus suaranya bagus.
Iya.
Mudah-mudahan nggak mati lampu.
Oh tadi kemarin itu pas saya
Kemarin.
uji, aduh mati lampu. Baru masih apa namanya pertanyaan pertama udah langsung mati lampu. Akhirnya pakai
Kami,
HP
ya.
semua.
Kami semalam sedang apa namanya tryout apa
Ya, GR itu ya.
ya
Ya.
atau mati lampu.
Prof. Popi, Pak Rektor hadir.
Katanya hadir.
Oh iya.
Mudah-mudahan on time.
Ya iya. Prof, kan Prof apa namanya atasan kami, kemudian Prof dari farmasi. Pak Hakim membuatnya saya bicara. Prof aja lah yang dari ILP, Prof ya dari penelitian.
Ya, Prof. Robert lah udah diundang.
Apa-apa,
Saya
kita
di sini
sudah
moderator.
kesepakatan di sini.
Saya di sini nggak representasi universitas dan fakultas.
Ya udah
Saya
aja sih.
anggota fakultas.
Ya Pak Safrudin ya.
Ya, selamat pagi, Bro Robert.
Ya,
Assalamualaikum, Bro Kopi. Selamat pagi,
Pagi,
Pak Hakim
Prof.
semuanya.
Sabarudin ya.
Saya di sini anak buahnya Prof. Hakim Bangun.
Oh,
Ya udah. Kalau begitu nggak apa-apa. Kami sudah ngomong sama Prof. Safrudin.
Kami
Tolong
lagi belajar
Prof. Popi
ini. Prof.
hadir
Hakim.
karena dari farmasi, biar Prof. Popi aja dari LP kami bilang.
Enggak lah,
Iya.
jangan lah. Emangnya ketua LP-nya siapa? Lagian kami ini sedang dilatih oleh Prof. Hakim untuk bisa.
Jadi luar biasa ini udah 192 dengan kita lah ini.
Iya.
192 peserta.
Cukup banyak, Bro.
Coba.
Aku udah bisa. Berarti dari
Jansen.
Pagi Prof. Johnson.
sudah kita.
Halo,
Ya,
Halo.
selamat pagi Prof.
Bu Popi, kedengaran suara saya?
Bagus
Kedengaran
sekali.
jelas.
Apa katanya?
Suaranya seperti anak muda.
Kedengaran katanya.
Dengar,
Prof. Jansen,
Kedengaran, Bu.
dengar. Bagus sekali
dengar.
suaranya.
Jelas, suaranya jelas. Oh, ke Pak Johnsonnya yang nggak jelas.
Udah
Kurang
dia.
dengar.
Tadi kami bercerita dengan Prof. Hakim, Prof. Popi.
Hmm,
Tohoku itu kan sekarang nomor 1 di Jepang.
ya.
Tapi memang dari dunia dia turun dari mula-mula kami bekerja sama 79, sekarang jadi 103. Nah,
Hmm,
persyaratan mereka waktu itu kami dengan almarhumah Trelia harus 2 universitas dulu, fakultas dulu yang melakukan kegiatan atau semacam IA, baru mereka mau diajukan MOU. Nah, sekarang ini kan sudah ada 2. Saya dari FIB, saya dengan Prof. Hamzon melakukan penelitian juga. Meskipun ya memang belum keluar skopusnya, tapi sudah 2 kali penelitian, 2-3 kali kalau tidak salah. Dengan juga ada mahasiswanya di FIB, Ali Mansar. Jadi kami kemarin bicara dengan dekan FIB, supaya kalau bisa tahun ini kita tidak lanjuti Prof untuk MOU-nya, karena sudah
Apakah suara saya kedengaran mereka? Nggak tahu.
fakultas itu.
Udah. Ya. Udah jelas.
Bapak dengar suara mereka?
Iya, kedengaran.
Udah. Mantap, mantap. Sudah jelas Prof. Jansen.
Jadi harus 3 dulu gitu ya.
2. Jadi udah
Kelas
2
2.
fakultas. Jadi
Ya,
kalau bisa kami izin dulu Bu, kami dulu menindaklanjuti. Setelah itu
ya,
nanti kami laporkan.
Iya,
iya.
udah.
Ya memang harus begitu ya. Jadi di beberapa universitas itu dia nggak mau MOU kalau belum ada
Iya,
kegiatan.
iya,
Kalau udah ada 2-3 kegiatan baru bisa MOU.
iya. Ini
Sebetulnya
sudah.
mungkin ini karena kita nggak tahu aja nih, barangkali seusuh ada juga yang udah kegiatan di Tohoku, cuma kita nggak tahu kan karena dulu kan nggak ini ya, inilah mau didata semua.
Iya,
Kalau bisa kita kita kabari juga ke dubesnya gitu ya, dubesnya
iya.
supaya tahu oh ada USU punya kegiatan di sana gitu.
Tes kedengaran suara saya, Pak Prof. Hakim.
Oh, kedengaran.
Oke.
Dengaran jelas.
Very
Oke.
clear.
Iya.
Ya, clear.
Prof. Jansen, apa kabar?
Baik, Pak Prof. Saya di lantai 3 ini.
Oh,
Oh,
boleh bisa.
berarti kan masih sehat lutut itu kan, Prof. Jansen. Kalau
Oh iya,
udah
masih
bisa.
bisa naik lantai 3, bisa naik tangga.
Nah, itulah cerita kami tadi begitu. Ada Ibu Wati bilang tadi kalau sudah tua kaki ini yang payah melangkah ke tangga katanya.
Oh, saya masih bisa tadi.
Bisa ya aja.
Ya.
Baguslah sehat.
Sehat.
Pikir tadi di kampus semua rupanya juga dari masing-masing kantor ya.
Iya, ada juga.
Iya.
Saya dari kantor juga. Takutnya nanti mati lampu di rumah.
Sudah 199 ini. Sebentar lagi 200.
Iya, betul.
Ya.
Ya. Jadi Tokyo University ini ada alumni kita yang sedang ambil postdoktoral gitu.
Oh,
Namanya Fadlina. Jadi melalui dia kita minta bantu dari pembicara dari Tokyo.
iya, iya.
Dia sedang ambil udah tamat dulu S1 dari farmasi kan, kemudian S3 di Nagoya,
Iya,
S2 S3, kemudian sekarang ambil postdoktoral di Tokyo University gitu.
iya, iya.
Nampaknya dengan bantuan alumni ini kita kadang-kadang mesti cari pembicara.
Iya, alumni dan pertemanan.
Pertemanan
Kita
gitu.
berteman dari awal. Kalau yang
Ya.
Profesor Toshiaki Kimura itu dulu
Nah.
peneliti di Sumatera Utara.
Oh, dia pernah peneliti
Alumni
di
kita,
Utara
Prof,
ya?
di Taiwan, Prof. Alumni kita di Taiwan. Dulu dia asisten, si Jiro.
Itu sekarang di BRIN dia kerjakan, BRIN ya?
Ya, di BRIN. Itu juga dia siap bantu kalau ada apa-apa gitu. Keren lah alumninya.
Iya. Nah. Yang di King College ini juga ada dari alumni juga. Dulu S1 di farmasi, sekarang diambil S3 di King College ya.
Stephen ya, Prof?
Ya, Stephen ya. Stephen. Yang di luar farmasi fisik kan yang asisten selalu kita sarankan ya, bisa ambil studi ke luar negeri gitu. Jadi sebagian mau pergi. Karena dari usul kan disampaikan kalau bisa kan pembicara tiap tahun. Di Tohoku ada juga farmasi ya, Pak Robert ya?
Tahu saya ada, kedokteran juga ada.
Oh ya.
Hmm.
Iya, Pak. Pembicara luar negeri ini biasanya jamnya pas baru dia masuk gitu. Dia nggak ikuti biasanya opening ceremony gitu. Pas jamnya langsung dibuka gitu.
Kalau begitu kita mulai saja, Prof, kalau udah bisa. Ini kan pesertanya sudah pas 200 ini. Begitu saya ngomong langsung 200 ini.
Ya,
Oh. Tapi Pak Rektor belum, kan?
ya Pak Rektor belum masuk lagi dia.
Hmm.
Udah 200 pas.
Iya, 201 udah.
Iya. Pak Bayu udah masuk kan? Halo Pak Bayu. Pak Bayu.
Ya, tes, Prof.
Sudah masuk kan?
Sudah, Pak.
Udah ya. Sudah ya, udah ada MC kan, udah bisa dimulai. Ah, kita tunggu lah Pak Rektor sebentar lagi kan. Atau kita tunggu sampai 8:30 kan gitu.
Selamat pagi, Prof. Hakim.
Ya, nanti 8:30 kita mulai. Ya.
Dengar aja. Atau yang diatur ya.
Good morning, Pak Bayu.
Good morning, Prof. Apa kabar, Prof Robert?
Kabar baik, sehat. Kalau sudah ada Pak Bayu, udah beres ini. Karena Pak Bayu yang mengatur jalannya acara.
Jalan kemana itu, Pak?
Nah,
Saya mah arahan aja, Prof.
ya,
Sama Prof Johnson udah lama nggak jumpa kita ya, Prof Robert.
oh iya
I-
betul.
iya,
Terakhir saya jumpa dengan beliau ini entah kenapa saya payungi beliau itu dengan apa namanya? Saya foto beliau itu
iya,
berpayung
iya.
dengan nyonya.
Tahun 2020 sebelum
Iya, jangan
pensiun.
jangan hilang foto itu, Prof Jansen. Itu kenangan itu.
Iya, 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 Prof.
Iya, luar
Terima
biasa.
kasih. Iya,
Setelah saya lihat foto itu dan saya kirim baru saya lihat, "Wih, ini kok luar biasa." Kita berjalan waktu dari rumah dinas rektor ya kan,
iya, iya,
itu ke Fakultas Kesehatan Masyarakat.
iya,
Jadi Prof Hakim, beliau ini berpayung dengan ibu.
Oh,
Lalu saya foto gitu.
dengan istri.
Ya lah dengan itu, dengan istri lah. Dengan istri maksudnya saya mengajak ibu ya, dengan istri beliau gitu.
Iyalah,
Ya, jadi
jarang kejadian itu kan.
iya dan
Jarang.
saya pun saya foto apa candid, jadi tak tahu bahwa kedua bukan apa namanya berdiri sengaja berfoto.
Yang
iya.
megang payung siapa, Prof?
Hah?
Yang megang payung siapa?
Saya lah.
Hansen. Nah, itu kan dia
Yang
dia
megang
tugasnya
payung.
itu. Tugas si bapak itu melindungi.
Iya.
Makanya saya lihat setelah saya lihat foto itu, saya kirimkan, saya bisa melihat belum tentu dapat foto seperti ini.
Iya, betul. Terima kasih, Pak.
Ya betul betul.
Ya, Prof Johnson sudah meninggalkan dunia hitam, Prof.
Udah ya, udah putih ya, rambut sudah putih ya.
Makanya tadi saya ditanya, masih bisa naik tangga katanya gitu.
Rupanya di lantai 3 beliau.
Iya.
Oh, beliau rajin berenang. Ya, Prof Johnson ya.
Iya, iya.
Olahraga rajin.
Oh, Prof Johnson berenang ya?
Bukan.
Bukan ya?
Lari.
Waduh, iya.
Lari.
Bukan lari dari kenyataan. Maksud saya jalan.
Olahraga ya.
Olahraga.
Tapi kadang-kadang lari dari kenyataan perlu juga. Jangan semua dipikirkan.
Oh iya, oh iyalah.
Iya, banyak enggak tahan lagi pikiran kita untuk itu.
Karena umur saya sudah 47.
Udah ya. Aduh.
47 ya, Prof ya.
Iya.
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 Iya, beda dikit lah sama saya.
Iya. Dibalikkan.
Iya,
Lah, iya.
dibalikkan.
Jadi gimana kita mulai aja sambil
Iyalah.
mulai barangkali ya sambil jalan aja.
Nggak perlu dikonfirmasi ke apa ya, Bu
Iya, iya.
Popi ya.
Tapi
Dikonfirmasi.
Ibu Popi barangkali bisa kontak ya.
Iya, ke Popi dulu ya.
Ya,
Baik, Prof, saya tanyakan ke sekretariatnya.
Baik,
iya, iya.
Introducing USU as one of the leading universities in Sumatera, Indonesia. USU has been part in the development of Indonesia. There are 16 faculties and 1 postgraduate school, started from the Faculty of Science and Technology to the Faculty of Humanities. The latest is the vocational faculty that was established as a hub center to excel human developments with highly competitive skills. USU is at the forefront of the implementation of Tridharma, which are providing education, advanced research, and community services. These are parts of our involvement in solving and addressing public matters, as well as contributing significantly in the national developments. USU aims to global competition, participating in the international community by targeting ourselves as one of the world-class universities. Industrial competition as well as technological advancement has led USU to prepare our students with necessary skills that are needed for modern-day communities. USU has been aiming to deliver exceptional education by improving our facilities and infrastructures, educational system, bureaucracy reformation to produce a world-leading academic institution. We also commit to contribute significantly to the development of Indonesia alongside with the nation's noble desire, and our transformation would remain the same as our past, current, and future promises.
Dear participants, the opening ceremony is going to start in a few minutes. Please be prepared. Make sure that you have a good internet connection so that you can enjoy this event. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. Welcome to the second international seminar on nanomedicine with the theme Technology and Development of Nanoparticles to Obtain Safer and More Effective Drugs. Let me introduce myself. My name is Bayu Eko Prasetyo from Faculty of Pharmacy, Universitas Sumatera Utara, and I will be the master of ceremony for this seminar. First of all, let's thanks to God, Allah Subhanahu wa Ta'ala, for His grace and guidance, so we can join this seminar. Salawat and salam to Prophet Muhammad Sallallahu Alaihi Wasallam. The Honorable Rector of Universitas Sumatera Utara, Profesor Dr. Murianto Amin. The Honorable Vice Rector 3, Universitas Sumatera Utara, Profesor Dr. Poppy Angelisa Zaitun Hasibuan. Honorable Head of Research Institute, Universitas Sumatera Utara, Profesor Dr. Robert Sibarani. The Honorable Head of Nanomedicine Center of Innovation, Universitas Sumatera Utara, Profesor Hakim Bangun. The Honorable Dean Faculty of Pharmacy, Universitas Sumatera Utara, Dr. Khairunisa. The Honorable, the keynote speakers, Professor Oleh Taratula from College of Pharmacy, Oregon State University, United States of America, and Professor Kanjiro Miyata from Department of Material Engineering, University of Tokyo, Japan. The Honorable, our invited speakers, Dr. Jitima Ami Lukanagul from Faculty of Pharmaceutical Sciences, Chulalongkorn University, Thailand. Dr. Alexandri Rosi Paul from School of Cancer and Pharmaceutical Sciences, Institute of Pharmaceutical Sciences, King's College, London, United Kingdom. Profesor Dr. Anayanti Arianto, Dr. Dwi Lestari, Dr. Sumayah, and Dr. T. Ismanili Hanum from Faculty of Pharmacy, Universitas Sumatera Utara. The whole of committee and all of the participants would take a part in this event. The second international seminar in nanomedicine is hosted by Nanomedicine Center of Innovation, Universitas Sumatera Utara, in a collaboration with the Faculty of Pharmacy, Universitas Sumatera Utara. Ladies and gentlemen, for a brief moment, we would like to invite you all to hear the Indonesian national anthem, Indonesia Raya. Ladies and gentlemen, we are now going to listen the welcome speech delivered by the head of Nanomedicine Center of Innovation, Universitas Sumatera Utara, and also as the chairman of the second international seminar on nanomedicine. Please welcome Professor Hakim Bangun. Time is yours.
Selamat pagi, salam sejahtera bagi kita semuanya. Good morning. The Honorable Rector of USU, Professor Nurianto Amin, the Honorable Vice Rector 3 USU, Professor Poppy Angelisa Zaitun Hasibuan, the Honorable Head of Research Institute or Lembaga Penelitian USU, Professor Robert Sibarani, the Honorable Dean of Faculty of Pharmacy, USU, Dr. Herunisa, the Honorable Vice Dean of Faculty of Pharmacy, USU, the academic staff from various universities in Indonesia and abroad, and students who attended this seminar. Ladies and gentlemen, nanomedicine can mean medicine formed in nanosize, where nanosize is below 1 micrometer. The preparation of drugs in nanosize is useful for increasing absorption so that the drugs become more effective and reduce the side effect. Especially, nanosized drugs are used as targeted drug delivery system for cancer cells. This type of research is currently being carried out to obtain safer and more effective anti-cancer drugs. In this seminar, four speakers will talk about nanomedicine for targeting drug for cancer treatment. Another speaker will talk about nanomedicine in the form of nanogel, nanosuspension, and nanodots for application in wound healing, anti-inflammatory, anti-aging, antibacterial, and so on. I would like to thank to Professor Oleh Taratula from College of Pharmacy, Oregon State University, USA. He will talk about multifunctional nanomedicine platforms for imaging and treatment of cancer and other diseases. I would like to thank to Professor Kanjiro Miyata from the Department of Material Engineering, University of Tokyo, Japan. He will talk about advanced polymeric medicine for nucleic acid delivery. I would like to thank to Dr. Alexandra Rossi Paul from the School of Cancer and Pharmaceutical Sciences, Institute of Pharmaceutical Sciences, King's College, London, UK. She will present about nucleic acids from drug discovery to drug delivery to cancer research. I would like to thank to Dr. Jitima Ani Lukanagol from the Faculty of Pharmaceutical Sciences, Chulalongkorn University, Thailand. She will talk about hyaluronic acid-based nanogel for molecular delivery and its application. The other four speakers come from the Nanomedicine Center, USU, as well as the academic staff of the Faculty of Pharmacy, USU. Professor Anayanti Arianto will talk about herbal nanogel for topical delivery, anti-inflammatory and skin anti-aging. Dr. Dwi Lestari will present her work on nanomedicine and tumorostroma, the importance of using a right tool in studying nanomedicine penetration. Dr. Tengku Ismanili Hanum will talk about development of nanosuspension ketoprofen as anti-inflammatory in vitro and in vivo evaluation. Then Dr. Sumaya will talk about carbon dots from isolated lignin of oil palm empty fluid bands for bacterial imaging of Escherichia coli and Staphylococcus aureus. I thank all the speakers from USU. Ladies and gentlemen, on this occasion, I would like to thank Director of USU, Professor Murianto Amin, who continues to maintain the Nanomedicine Center or PUI Nanomedicine at USU. I would like to thank Ketua Lembaga Penelitian or Head of Research Institute USU, Professor Robert Sibarani, for his guidance and facilitation in holding this seminar. I also thank the committee of the Second International Seminar on Nanomedicine and my student from the Laboratory of Physical Pharmacy who worked seriously to hold this seminar. Ladies and gentlemen, please enjoy the seminar. I hope it will be useful in research and drug development in the world. Thank you. Terima kasih.
Prof. Hakim. Then I would like to invite the head of a research institute, Universitas Sumatera Utara, Professor Dr. Robert Sibarani, to deliver the opening speech. Please welcome Professor Robert. Time is yours.
Thank you, Mr. Bayu, as the master of ceremony of this international seminar. Our honorable rector of Universitas Sumatera Utara, Professor Dr. Murianto Amin, the honorable vice rector here, Professor Poppy Hasibuan, as a vice rector for research and corporations attend the seminar. Our Honorable Head of Nanomedicine Center of Innovation, Universitas Sumatera Utara, Profesor Hakim Bangun, PhD. The Honorable Keynote Speakers, Professor Kanjiro Miyata, PhD, from Department of Material Engineering, University of Tokyo, Japan. The Honorable Keynote Speaker, Professor Oleh Taratula, PhD, from College of Pharmacy, Oregon State University, United States of America. The distinguished invited speakers, Dr. Jitima Ami Luka Nagul, from Faculty of Pharmaceutical Sciences, Chulalongkorn University, Thailand. The distinguished invited speaker, Dr. Alexandri Rossi Paul, from School of Cancer and Pharmaceutical Sciences, Institute of Pharmaceutical Sciences, King's College London. And the distinguished invited speakers from Universitas Sumatera Utara, from Faculty of Pharmacy, Universitas Sumatera Utara, Profesor Dr. Anayanti Arianto, Dwi Lestari, PhD, Dr. Ismaneli Hanum, Dr. Sumayah, and ladies and gentlemen, and also the distinguished Dean of Pharmacy, Universitas Sumatera Utara, and also the Vice Dean. First of all, on behalf of Research Institute Universitas Sumatera Utara, we expressed our deep thanks to the head of Center of Nanomedicine, Research Center of Nanomedicines, Professor Dr. Hakim Bangun, with all the committee which host and hold this second international seminar on nanomedicine with the very important theme, technology and development of nanoparticles to obtain safer and more effective drugs. We are from Research Institute of Universitas Sumatera Utara, give a high appreciations for holding and hosting this kind of international seminar, because we are sure that it will give or contribute the significant impact for all participants, for the Research Center of Nanomedicines, for Faculty of Pharmacy, as well as for Universitas Sumatera Utara. So, last but not least, we are going to say happy international seminar and enjoy this very important international seminar on nanomedicines. We hope that now the participants are already 230, of course, including us. We hope that until the end of the seminar, the participants remain 230, even more than it. Again, happy seminar, international seminar, and enjoy it. Thank you.
Thank you, Prof. Robert, for your opening speech. Now, we invite Rector of Universitas Sumatera Utara to give the opening remark and officially open our international seminar today. To Professor Dr. Murianto Amin, the screen is yours.
Thank you. Good morning. This is health and property for all of us. Om Swastiastu, Namo Buddhaya. The Honorable Vice Rector for Research and Community Development, Professor Poppy Kasibuan, Head of Research Institute Universitas Sumatera Utara, Professor Dr. Robert Sibarani, Dean and Vice Dean, Faculty of Pharmacy, Universitas Sumatera Utara, Head of Nanomedicine, Center of Innovation, Universitas Sumatera Utara, Professor Dr. Hakim Bangun, and our keynote speakers, Professor Kanjiro Miyata, PhD, from the University of Tokyo, Japan, Professor Oleh Taratula, PhD, College of Pharmacy, Oregon State University, USA. Our invited speakers, Dr. Itimani Ratanagul from Faculty of Pharmaceutical Sciences, Chulalongkorn University, Thailand. Dr. Alessandri Rosi Paul, School of Cancer and Pharmaceutical Sciences, Institute of Pharmaceutical Science, King's College London. Profesor Dr. Anayanti Haryanto, Fakulti Farmasi, Universitas Sumatera Utara. Wilestari, PhD from Faculty of Pharmacy, Universitas Sumatera Utara. Dr. Sumaya, Faculty of Pharmacy, Universitas Sumatera Utara. And Dr. Teis Maneli Hanum, Faculty of Pharmacy, Universitas Sumatera Utara, and all the participants in this seminar conference. Ladies and gentlemen, esteemed professor and participant of this conference. First, we express our gratitude to represent Allah Subhanahu wa Ta'ala, the most gracious and most merciful, for allowing us to gather this morning to attend the second international seminar on nanomedicines, organized by the Nanomedicine Center of Innovation at the Universitas Sumatera Utara. Respected professor, participants, and distinguished guests, I want to extend to thank you and welcome to all of you in this virtual forum, including our esteemed speakers and all participants from various countries. We appreciate the organization of this international seminar as part of Universitas Sumatera Utara effort to promote the sharing of knowledge and the latest information, particularly in the field of nanomedicines innovations. Beyond serving as a platform for knowledge exchange among practitioners and academic presence, this forum will enhance communications and foster connections with academic forum different countries. I think this is potentially leading to collaborative activities in the scientific realm. The second international seminar in nanomedicine to take to them is technology and development of nanoparticles to obtain safer and more effective drugs. This seminar represents a tangible step towards advancing scientific knowledge, especially in nanoparticle technology for developing safer and more effective drugs. As an institution with a vision to become a center of excellence in sciences and technology development, we are proud to host this event. We believe that collaborations and exchanges ideas among scientists, researchers, and practitioners from various countries which contribute significantly to the progress of nanotechnology development in the pharmaceutical industry. Nanoparticle technology brings breakthrough in drug manufacturing and positively impacts treatment efficiency, reduces from the side effects, and opens possibilities for advances therapy development. Therefore, through this seminar, we can collectively explore the potential of this technology and design innovative solutions to global health challenges. I thank you to the organization for the committee. They hard work in organizing this event. May this seminar be a productive forum, opening new collaborative opportunities and inspiring ongoing research in the future. This international activity is also part of Universitas Sumatera Utara efforts to enhance various programs supporting campus internationalization, increase the number of international scholarly publications by faculty and students, and rekindle research interested leading to innovation in health and medicine. The evolution of time undoubtedly influences shifts in knowledge, treatment methods, and patient care, introducing nanomedicine with nanoparticle technologies as a continuous and leading innovation in the health field. The ability to create functional nanoscale material innovation is the foundation for further development in nanotechnology. I hope this international conference brings forth new ideas from the speakers, share information, experiences, and knowledge to the attending participants. The conference is also expected to delve deeper into the science and technology of nano, providing new opportunities for developing various pharmaceutical products, vaccines, and therapeutics methods. We warmly welcome to researchers attending the conference, believing that it will be generate fresh ideas and targeted recommendations and distribute knowledge for the greater good of society. Ladies and gentlemen, respected attendees, in conclusion, these are the remarks I can share this moment. Enjoy the entire seminar program. I officially open this international conference, and may Allah Subhanahu wa Ta'ala, God Almighty, always bless all our efforts and aspirations. Thank you. Assalamualaikum warahmatullahi wabarakatuh. Om Santi, Santi, Santi. Om Namo Buddhaya. Rahayu.
Thank you very much, Professor Murianto Amin, for the opening speech remark. Ladies and gentlemen, before we are going to the next agenda, please turn on your camera because we would like to take a pictures of all of the participants. Please the committee to help me to take the pictures.
Okay, I'll take the screenshot. Slide number 1. 1, 2, 3. Slide number 2. 1, 2, 3. Slide number 3. 1, 2, 3. Slide number 4. 1, 2, 3. Slide number 5. 1, 2, 3. Slide number 6. 1, 2, 3. Slide number 7. 1, 2, 3. Slide number 8. 1, 2, 3. Slide number 9. 1, 2, 3. And the last slide. 1, 2, 3. Okay, thank you.
Okay, thank you for the committee. Dear participants, before starting the main agenda, I would like to announce some rules that all of the participants need to obey during the seminar, so that the seminar goes smoothly. 1. For the participants, please rename using a format name _ affiliation. 2. Please turn on the camera and turn off your microphone during the presentation. And 3. You might submit your question to the speaker by type the questions in a chat room with format name _ affiliation _ question. Or you may ask directly by raising your hand at the Q&A session. And number 4, don't forget to write down the important things, because there's a post test at the end of the seminar, and the participant is required to submit the post test in order to get the certificates. Thank you. Now, we are continuing our seminar to the main agenda. For the first keynote speech will be delivered by Professor Oleh Caratula from the College of Pharmacy, Oregon State University, United States of America. The moderator for this session is Professor Dr. Poppy Angelisa Zaitun Hasibuan. Professor Poppy Angelisa is a Vice Rector 3 in Universitas Sumatera Utara, and also as a lecturer at the Faculty of Pharmacy, Universitas Sumatera Utara. Her area of research interest is in the pharmacology pharmacy, particularly in the cancer research. Please welcome Professor Poppy Angelisa. Time is yours.
Thank you, Mr. Bayu. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. We are entering the first session of this seminar. Prof. Oleh have entered the meeting. Okay. We have Professor Oleh Caratula, PhD, our distinguished speaker from Department of Pharmaceutical Sciences, College of Pharmacy, Oregon State University, USA. Hello, Professor. How are you?
Hello, I'm doing well. How are you today?
Nice to see you. Thank you for being with us. We are happy because we can meet you even in online meeting, when we will get an interesting topic. It's 7 p.m. in Oregon, right?
Yeah, it's 6 p.m. 6:05.
Oh, 6 p.m. It's still first. It's 9 a.m. in Indonesia, and we are already 1st February now.
So
Ladies and gentlemen,
you are 1 day ahead of me.
Okay. Yeah. Okay, ladies and gentlemen, firstly, I will read Professor Caratula CV in brief. Professor Caratula is a co-director of the Center for Innovative Drug Delivery and Imaging from 2021 until now. He is also an affiliate research member of the Knight Cancer Institute from 2016 until now, and adjunct faculty in the Department of Biomedical Engineering, Oregon Health and Science University from 2015 until now. In 2016, he received the Richard T. Jones New Investigator Award. Wow. The major focus of his research is the development of a multifunctional nano platform for imaging and specific eradication of cancer, endometriosis, and other diseases. He has many publications, so his H index is high, 36, and he has some patents. He is also actively involved in institutional and professional service activities and has mentored many students in his laboratory. He was also recognized as P1 Professor of the Year 4 times. Wow. And received the 2022 OHSU Senate Distinguished Faculty Teaching Award. Now, Professor Taratula will deliver the topic multifunctional nanomedicine platform for imaging and treatment of cancer and other diseases. Yeah, we will hear him for about 80 minutes and continue to Q&A session for 10 minutes. Ladies and gentlemen, you can type your question before he finishes or raise your hand at the end of this presentation. All right, Professor Taratula, the time is yours.
Can you allow me to share my screen, please? Okay, perfect.
Committee, please. Okay.
All right. Good morning, everyone. First of all, thank you so much for your kind introduction. Second of all, I would like to thank Dr. Hakim Bengud and the organizing committee for their kind invitation. This is my great pleasure to be here and share my research results with you. As was already mentioned, I'm a faculty at Oregon State University College of Pharmacy, and my lab is located in a city named Portland. So Portland, Oregon, because actually we have 2 Portlands in the United States. So Portland, Oregon is in the northwest part of the United States. And here in this picture, you can see our building. This is the building where my lab is located. So this building was opened back in 2014, and the idea of this building was to bring scientists from different Oregonian institutions to the same place. And so it allows us to work together on different projects. As you know, these days, all the projects, the collaborative project, it's very difficult to run a big project on your own. You always need different expertise. And so we are very lucky that we can work in this building. As you can see from the title of my presentation, today I'm going to talk about different nanomedicine platforms which we develop in our lab. And we use those nanomedicine platforms for treatment and diagnosis of different diseases. And specifically, so we work a lot with cancer. We also apply our nanomedicine platforms for the treatment of endometriosis. We have also a big project focused on application of nanomedicine for cancer cachexia. And recently we started to work with ectopic pregnancy and inflammation. So my goal today, since I have about 80 minutes, I will try to give you several examples of our project and discuss them. And at the end, I will be more than happy to answer your questions. The first project which I want to address is titled Theranostic Nanomedicine Platform for Image-Guided Surgery and Intraoperative Phototherapy. So many of you know that surgery is a main or cornerstone treatment for many cancers. And especially for those cancers which didn't metastasize yet. And so in this case, if the cancer was diagnosed at the early stage, a surgeon can simply remove the tumor, and if there is no cancer cells left behind, so that patient has very high chance for recovery and survival. And according to American Association of Cancer Research, so surgery is used for different types of cancer, such as you can see here, there is the list for breast cancer, ovarian cancer, cervical cancer, head and neck cancer, and so on. So as I mentioned, so surgery works very well for the for cancer at the earliest stages, especially when the tumors is localized. But surgery is also a very important treatment even for metastatic cancer. And specifically, surgery is very important for ovarian cancer. So ovarian cancer is very deadly cancer. It's very often it calls the silent killer. And the reason for that, because patients, they don't have symptoms till the very advanced stage. So according, to the statistics, more than 19,000 cases we have of ovarian cancer we have in the United States each year. And approximately more than 13,000 people died the last year from ovarian cancer. So the overall survival rate for ovarian cancer is very low, is about 45%. And major reason for that, because as I mentioned earlier, many patients, they are diagnosed when ovarian cancer tumor is not localized to the ovary, but rather spread to the abdominal cavity. And for those people who have advanced ovarian cancer, the survival rate is very low. So what is the standard treatment for metastatic ovarian cancer? So this is the first step. This is optimal debulking surgery followed by chemotherapy. And usually for chemotherapy, they use like cisplatin, carboplatin, this type of chemotherapeutic agent. And optimal debulking surgery means that the surgeon, they try to resect as many as possible cancer lesions in the abdominal cavity. And according to the clinical studies, the surgical outcome is very important, and actually it significantly determines the survival rate after the treatment. So here you can see two tables. For example, on the left side, this table represents clinical data, and it was a relatively small clinical trial, several hundred patients were involved in that. And what they show here, if the surgeons were able to resect all visible cancer tumors, the survival rate for those patients was more than 100 months. And when small tumors like less than 0.5 centimeters were left behind, so for those patients, survival rate went down significantly. Like here, you can see 66 months, and so on. You can see very similar statistics from the second clinical trial on the right. So it was a big clinical trial with thousands of patients, and you can see the same. If no gross visible tumors were left behind, the survival rate was 99%. And if the tumors less than 1 centimeter were left behind, so the survival rate dropped down to 36 months. So what it tells us, the surgery played a significant role in the treatment of metastatic ovarian cancer. And as you can see here, so depending on the surgical outcome, the survival rate can decrease by several years. So we're talking about like 5-6 years of human life. So now the question is, why it's so challenging to remove all the cancer tumors? And one of the reasons for that, because we don't have very efficient operative imaging techniques. So, like to diagnose cancer, we use MRIs, a CAT scan, ultrasound, and so on. But all those techniques, they are pre-operative techniques. So meaning patient, they're scanned before the surgery, and based on those imaging, surgeons, they can plan the surgery. But what happened during the surgery? So surgeons, they usually rely on their experience, on their eyes, and on their fingers. If they can distinguish properly cancer tissue and resect them, so you can understand that the survival rate can increase a lot. So when I joined the College of Pharmacy back in 2011, so I really wanted to contribute to the cancer surgery. And my goal was to use my knowledge in nanomedicine and nanotechnology. So what I wanted to do, I wanted to develop nanoparticles which could be injected, let's say, 24 hours before the surgery. And during that period of time, so they can, after systemic administration, they can localize in cancer tumors. And what they can do, they can generate some signal. And in our case, so we were focused on the fluorescence. So you can imagine, so those nanoparticles, for example, you can see the cancer tumors, like the picture of cancer tumor, and on the right side, so the nanoparticle, we wanted to develop nanoparticles which potentially can delineate those cancer tumors by generating the fluorescent signal and just make those cancer tumors to glow. In addition to that, so we want to make these nanoparticles multifunctional or theranostic nanoparticles. So those nanoparticles, they also could be used for the preoperative treatment. And like, for instance, if there are some tumors which are difficult to remove because they could be very close to some vital blood vessels or nerves and so on. So in this case, it's possible to treat those tumors, unresectable tumors, during the surgery. And the way how we seen that, so you can have nanoparticles which can also absorb a light of the certain wavelengths and convert that light energy into heat and destroy cancer tumors. And now, what I'm going to do, I'm going to describe you how we work on this idea and how we develop this platform. So, because most of you work in pharmaceutics, if you want to develop any imaging or therapeutic agents, you want to make sure, like, first of all, it's safe, because if it's toxic, it doesn't matter how efficient it is. So it's not going to move in, far away in clinical trials. Second of all, it has to be efficient. And for instance, it can, it can generate significant fluorescent signal, that what we wanted, or generate significant heat. And third of all, it has to be simple, because it's very difficult to translate, especially nanoparticle-based imaging or therapeutic agents, if it's challenging for them to prepare, since they have many components. So as a result, just to make it simple, we want to choose a molecule or agent which can do both, which can generate the fluorescence and also can provide the photothermal therapy when you shine the light and increase the intratumoral temperature. One of the first consideration was the excitation and emission wavelengths of that dye molecule. And the reason for that, because not every molecule or not every dye can be used for in vivo imaging. And the primary reason for this is that our body fluids and body tissue, they absorb light, especially in the visible part of the spectrum. And also, and you can imagine if you want to excite or just record the fluorescence signal, so the signal is going to be weak because tissues, they are going to absorb that. On the top of that, our body fluids and tissue, they also have autofluorescence in the visible part of the spectrum. So that also could be a problem because that autofluorescence can give false positive signals to the surgeon, and apparently surgeon can resect the healthy, non-cancerous tissue, or simply is not going to detect that cancer tumor. So what we want specifically for fluorescence imaging and phototherapies, we want molecules which absorb light and emit fluorescence in the near infrared part of the spectrum. And the reason for that, because our body tissue, they have limited absorptions and emission in this part of the spectrum. So this part of the spectrum calls biological optical window, since there is a low absorption of light by our body tissue. So after that, our goal was to choose the right molecule, and we analyze like number of different molecules, and here you can see several examples of them. And one of the first candidate was ICG, indocyanine green molecule. So that molecule is FDA approved and being widely used. But one of the major problem is that molecules is not photostable. So if you shine the light on that molecule, you're going to degrade that molecule and lose the optical properties. So eventually we identified the molecule which is highlighted here, and we call it silicon naphthalocyanine, or silicon NC. And that molecule has all the required property which we needed. So first of all, it can absorb and generate the fluorescence in the near infrared part of the spectrum, as you can see here, around 800 nanometers, which falls in that biological optical window. And on the top of that, in addition to the fluorescence imaging properties, that molecule also could be used for phototherapy. And you can see here, so when you have a solution of that molecule and you shine the light around 780 nanometers, so that solution, that molecule absorb that light and generate heat. And the heat, it has very high efficiency in that respect. And also, I just want to mention here that the light which we use to excite that molecule cannot generate heat on its own. So we identified that molecules, but the major issue with that molecule was it was very hydrophobic. So technically, it was not water soluble in water. Well, to overcome this challenge, we take advantage of our skills in nanotechnology. And specifically, we develop the polymer-based nanoparticles as a delivery vehicle for those molecules. So our approach, we develop, as I said, we always, in order to make sure that it has the translational potential, so we wanted to develop a simple and reliable, reproducible, and robust approach for the preparation of these nanoparticles. And what we did, we use a block polymer called PEG PCl. PEG stands for polyethylene glycol, PCl polycaprolactone. So that molecule has, as you can see here, it has the PEG, which is hydrophilic part, and PCl, which is hydrophobic part. But since PCl is longer or has a higher molecular weight, so that molecule is mostly soluble in such solvent like THF, and the same our dye was also soluble in THF. So the way how we prepare these nanoparticles, we dissolve the polymer and the dye in THF, and after that, when we added water and started evaporating organic solvents, So those block polymers, they are arranged into nanoparticles in the way that PCl is in the core and the PEG is outside, making those nanoparticles water soluble. And what happened, and if you have hydrophobic molecules during the formation of those nanoparticles, so those molecules, they are encapsulated inside of those polymeric nanoparticles. And as you can see here, as I mentioned, our approach gives very uniform nanoparticles, as you can see here from TEM image. And also those nanoparticles, they are around 40 nanometers. And what we learned like during all these years, so this size is very favorable for the passive accumulation of nanoparticles into the cancer tumor after systemic administration. And on the top of that, those nanoparticles, they have very extended shelf life. So here you can see dynamic light scattering profiles for those nanoparticles when they are freshly prepared, the black curve, and after 9 months. So it just shows that those nanoparticles stable, they don't increase in size, and so on. And another important thing what we demonstrated, you remember a few slides ago, I mentioned that there is also a concern about the photostability of the dye molecules. And as I said, like one of the famous molecules, which is ICG, so it has the issue with the photostability. And you can see here the absorption spectra for the solution of that molecule. You can also see the picture of the solution and the fluorescence imaging of that solution. So what happened if you expose that molecule to near infrared light, let's say for 30 minutes, so that molecule started to photodegrade. And as a result, you can see on the left after exposure to the light, so that solution, the fluorescence went down and solution started to lose its color, and also absorption spectrum, they show they show a significant decrease in the intensity. It shows that this molecule is not photostable. And at the same time, if after that light exposure, that molecule also cannot generate heat. So you can see here, so when you shine the light, and after a certain period of time, the temperature in the solution goes down. And the reason for that, because that molecules went through the photodegradation. In contrast to that, ICG, other molecules, which we call silicon and C, so it's very photostable. And you can see here the molecule treated with light, similar as ICG, and there is no change in the absorption spectrum, there is no change in the fluorescence, and also this molecule, can produce the heat, and that heat could be maintained as long as the light is on. So yeah, it's a very important feature, and as I mentioned, because otherwise, if the molecule photodegrade during the surgery, so it means that surgeon can get a false negative signal, so just showing that there is no fluorescence, meaning there is no cancer tumor, but it's related to the photodegradation molecule, but not to the absence of the tumor in that specific location. So in the next step, our usually first study was we evaluated the biodistribution of that molecule, of those our nanoparticles in mice with subcutaneous ovarian cancer tumor. And what we can see here, so by using this imaging system, we can detect the fluorescence in animals. So after like 30 minutes, you can see the fluorescence is all over the body. And the same story after 2 hours, and it indicates that those nanoparticles, they circulate in the bloodstream. And after 24 hours, so we started to see the localized signal in the cancer tumor. And obviously, that at least in the drug delivery field, we believe in passive targeting because cancer tissue, they are angiogenesis dependent. So they, have blood vessels, and very often those blood vessels, they are leaky. Nanoparticles, they can escape from those pores in the blood vessel, and they retain in the tumor due to impaired lymphatic drainage in the cancer tissue. So right here, also we euthanize those animals at different time points. And what we see here, a similar story, right? So after 30 minutes, the nanoparticles, they're in all the organs and still very little accumulation in the tumor. But 24 hours later, so the nanoparticles and dyes, they just cleared from the organs, and we have a higher signal in the tumor. But we still see some fluorescent signal in the kidneys and in the lungs. So our next step, what we want to do, I mean, that's kind of a disadvantage of this approach would be, because obviously during the surgery, the signal could come not only from cancer tissue, but from the non-malignant tissue. And our goal, what we want to do, we want to make nanoparticles which don't have any fluorescence when you inject them or when you keep them in the solution, or when you inject them into the bloodstream. And we would like those nanoparticles only activate their fluorescence when they accumulate in cancer tissue. And we tried many different strategies to achieve that. And so we try to chemically modify those molecules and attach them to the quencher molecules, but it didn't work very well. So apparently the solution for us was very simple. So what we realized, if you have inside of those polymeric nanoparticles a small number of those dyes molecules, the final polymeric nanoparticles, they always have the fluorescence. But what we also noticed, when we increase the number of the molecules inside of that hydrophobic core of polymeric nanoparticles, the fluorescence signal disappears. And the reason for that, because those molecules, dye molecules, they are very close to each other. And what happened, the quenching of the fluorescence happened. So technically, when those molecules, they absorb light, they simply, don't emit fluorescence, but rather transfer that signal to each other. And as a result, that fluorescence is quenched. And here you can see the fluorescence spectrum just showing what's going on here. So when you have very low amount of that dye silicon in C, we have a very strong fluorescence signal. And I just want to mention here, so the red color represents the stronger signal. And when we started to increase the amount of molecules inside of the polymeric nanoparticles, so we can see also the signal, fluorescence signal is decreasing. And eventually, when we hit, this concentration of the dye, so we don't have the fluorescence. So in the next step, our hope was, so those nanoparticles, they don't have a fluorescence, but when they internalize into the cancer cells, so because you can imagine, those are self-assembled nanoparticles, and our hope was that in the cytoplasm, due to different interaction with different molecules there, so those nanoparticles, they disintegrate, and obviously fluorescence molecules, they will be released, and as a result, so the distance between those molecules will increase, and we will get the fluorescence again. So in order to see here what's happening, so you remember that we have the dye molecule silicon in C, which is near infrared dye. And we also label our polymer with FITSI, the green color dye. And what we see here, when we incubated those nanoparticles with cancer cells, after like 30 minutes, we only see the green color. So the green color, it represents the polymer. So it just shows us that the nanoparticles, they already inside because we see the fluorescence from the polymer, but we don't see any near-infrared fluorescence from the dye because that dye is inside of the nanoparticles and it's quenched. Also, when we check the fluorescence in the cell culture medium, we also only see the green color. So it means nanoparticles, they were not disintegrated in the culture medium. But what happened like a few hours later, we started to detect also the near-infrared signal. So it just tells us that nanoparticles, after they get into the cells, so they become disintegrated, they release the dye, and they started to generate fluorescence. And again, it only happened inside of the cells. We don't see any near-infrared signal in the culture medium. Now, so we went back to the same experiment. We used mice with subcutaneous cancer tumor, and you can see here, so this is the mouse, the fluorescence image of the mouse before the injection. Here, like 30 minutes after the injection of our nanoparticles, 2 hours, 4 hours. During this period of time, we don't see any fluorescence. And you remember previously when we have another nanoparticle, when the fluorescence was always on, so those mice, they were red. So now it shows us that nanoparticles were injected, but we don't see any signal. So it indicates that those nanoparticles, they are, they are in the bloodstream and the dyes stay inside. But 6 hours after injection, we started to see some signal, and this signal was increasing this time. And at 24 hours, you can see very strong signal, the red color here. So it just represents here that nanoparticles during those 24 hours, they, after systemic administration, so they accumulated in the tumor, and they become, and they get inside of the cells and become disintegrated and release the dye, and dye started to generate the fluorescence. And here you can see the fluorescence imaging of the resected organ. So the main signal was only in the tumor, and we only have some background signal in liver, kidneys, and the spleen. So now we come to the point how the image-guided surgery works. So here we have the system called Fluobeam 800. So this is this instrument was approved by FDA for clinical trials. And the way how this instrument works, so it has the excitation light, it has the detector, and it can technically detect the fluorescent signals, and you can see it on the screen. Here we had the mouse, right, with subcutaneous tumor, and we injected that mouse 24 hours before the imaging with our nanoparticles. And what you can see here, right? So we recording this video with this instrument. So you can see this part of the tumor is glowing, and this, this glowing comes from our nanoparticles, because our nanoparticles were accumulated there, released the dye, and now they generate the fluorescence. So this is how image-guided surgery works. So obviously you can say that, it's why do we need, nanoparticles to detect subcutaneous tumors? And I agree with you. So after that, we went to the more advanced model. We prepare mice with metastatic ovarian cancer. And here you can see, so in order to prepare the mice with metastatic ovarian cancer, we use ovarian cancer cells stably transfected with luciferase. So those cells, they generate bioluminescence, and it's kind of easy to follow the cancer development, because our body doesn't generate bioluminescence. So this yellow color, actually, this is the bioluminescence which comes from the cancer cells. And when we inject our nanoparticles and open the abdominal cavity, you can see that we have very strong fluorescent signal. And that signal actually overlap with the signal, bioluminescent signal with, from cancer cells. It just shows us so that our nanoparticles, they accumulate there in cancer tumors and they become activated there and they generate this fluorescence. Now, I just wanted to show you another video of the this intraoperative surgery. So in this case, what we done, we open the mouse and we remove all the large tumors which were visible, and we wanted to see if that image-guided approach can help to identify the small tumors. So because actually this is the whole purpose of image-guided surgery, to find some small tumors. Now I just get stuck, sorry about that. Okay, so now you can see this video, and in this video we, as I said, we remove all the big metastatic cancer tumors. And what we see here, that we can see that small glowing spot. So that small glowing spot is the cancer tissue, and that tissue is very small. It's only like a few millimeters. It's just showing that these nanoparticles, they can get there and they generate the fluorescence, and we can actually find that tumor during the surgery, which was very difficult to see with your eyes. And we confirmed that it was the cancer tissue because that tumor has the bioluminescence. And you remember, bioluminescence is bioluminescence comes from those cancer tumors. Now, we also tested this technology in rabbits with the liver cancer, and you can see here, again, this is the video recorded by fluorobeam. So this is the liver tumor here, the glowing one, and we can see that those nanoparticles, after systemic administration, they accumulated in the liver tumors. Now, so I told you at the very beginning that our goal was to develop nanoparticle with a dual function. And the first function was obviously fluorescence imaging, intraoperative fluorescence imaging. And the second goal was nanoparticles which could be used for intraoperative treatment of cancer tumors. So, and the way how we tested if our nanoparticles can actually eradicate cancer tumors. So see here in the first step, you have a mouse, and that mouse has the subcutaneous tumor. So we injected that mouse with other nanoparticles, and 24 hours later, we can have the special device. We generate the fluorescence light of the certain wavelengths, like in our case, we use 780 nanometers. This is the absorption peak of that dye molecule. And here there is the temperature probe. So, and when we shine when we expose that tumor to that light for like 20 minutes or so, or even less than that, so I believe it was 15 minutes. So what we can see here, after one treatment, the tumor basically started to shrink and disappeared and never regrow. But at the same time, so this treatment is very specific because what we can see here, that nanoparticles alone, this is the dark blue curve, without light, or the light, when we shine the light on the tumor and didn't inject nanoparticles, it didn't have an effect on, the cancer growth. And what you can see here, so this treatment worked very well in different types of tumor. Like, for example, we have cancer tumors which was doxorubicin resistant and cisplatin resistant. So, and we had the same effect. Here also, and the reason why it happened like that, because when we measured the temperature during this light exposure, So you can see here the tumor which had nanoparticles, those nanoparticles, they convert the light into the temperature, and we can easily raise the temperature of the tumor up to 46, 47 degrees and keep this temperature as long as the light is on. But I also told you that the light by itself, without nanoparticles, doesn't generate any heat. We confirm that treatment also in the rabbits, and you can see here. So we also measure the intratumoral temperature inside of the tumor. When we shine the light, temperature went up, and in the tissue adjacent to that tumor in the liver tissue, the temperature increase was minimal. So it just tells us that nanoparticles were mostly located in the cancer tumors. Now, always we want to make sure that these nanoparticles are safe, and one of the way, how we test the safety, we check the body weight of animals, treated animals, and we didn't see any decrease in the body weight. We also did the blood analysis, and we measure all these biomarkers, which when you go to the clinic, so when doctors, check your blood, such as BUN, creatinine for kidneys, for liver function, ALT, ALP, cardiac and muscle function, CK. And what we can see here in the control and treated group, we didn't see any difference. So it means that the treatment was safe. Another problem is these fluorescence dyes when you inject into the body. So those dyes together, for example, with nanoparticles, they can also accumulate under the skin, and they can cause the phototoxicity. So here, this is the example when the mouse, from one paper, when mouse was treated with one photoactive agent, and when that mouse was exposed to the daylight, so you can see what happened to the skin. So we did the same studies. We also injected our mice with nanoparticles, exposed them to the light, and we didn't see change in the skin. So it means we don't have that phototoxicity. Finally, what was done, we started a small veterinarian clinical trials. And here at our university, we have the veterinarian hospitals, and in collaboration with my colleagues from that hospital, so we recruited the patient dogs with cancer. So here you can see examples of images from 2 different dogs. So our goal was to see if nanoparticles are safe when they injected into large animals, and if they can really go to the tumor and delineate in the real tumors, because before we have mostly grafted tumors, and they might be different than spontaneous tumor. And you can see here that in both cases we can see this white color, and you remember that white color, it comes from the fluorescence. It just showed that nanoparticles were accumulated there and they generate the fluorescence. And also, since those dogs, were owned by people, so after the surgery, my colleagues, the veterinarians, they follow those dogs, and we didn't see any toxicity. So we patented this technology, and this technology was licensed from us by one of the startup company here in Oregon. But at the same time, we also started to work with. We tried to explore if this technology could work for some other diseases. And one of the diseases which we started to explore was endometriosis. For those who don't know what is endometriosis, I just give you a brief idea about that. So endometrium is the tissue which covers the uterus inside. But endometriosis, so what can happen in certain people? So those cells, they can go into the abdominal cavity, for example, and they form the lesion there. And this is the, showing the laparoscopy image of the endometriosis lesion. Exactly why endometriosis happens in people, it's not very well studied yet. But there is something what we call the Sampson hypothesis. And according to that hypothesis, endometriosis forms when endometrial cells here from the uterus, they pass through the fallopian tubes during the menstruation, and they sit in the, in different parts of the body. So endometriosis is very bad disease. I mean, obviously it's not cancer, but it causes very severe pain and infertility in many patients. And according to the statistics, more than 170 million women worldwide, they have endometriosis. And unfortunately, currently, there is no medical treatment just to treat endometriosis. So the therapies which we have here, at least in the United States, those are hormonal therapies, and they rather deal with the pain. And very often, these therapies they cause infertility. So another options for patient who wants to have children, this is the surgery. But what the problem is, the recurrence rate after the first surgery is very high. It's more than 50%. It means that many endometriosis patients, they need like multiple surgeries. And one of the reasons behind that because probably not all the lesions, they were removed from there. So, and our hypothesis when we started this project, so we started like several years ago, and at that time, nanoparticles were not used for endometriosis. So were only like limited number of publications. But our hypothesis was that endometriosis, the same as the cancer, is angiogenesis dependent disease. So the lesions, when they grow, they form a new vasculature. And those vasculatures, they are also leaky. And our idea was that when we inject our nanoparticles, we will get the similar accumulation because of the passive targeting. And you know about passive targeting. Also, we can also take advantage of active targeting. So we can modify our nanoparticles with certain peptides, which can target the receptors on the endometriosis cells. And here, those are our first studies. And this is a mouse model of endometriosis. The challenge is to work with endometriosis because endometriosis doesn't happen spontaneously in mice or like small animals. So it only happens in the people and in monkeys. So, but lucky, here in Oregon, we have the National Primate Center, and I found a collaborator who worked with monkeys and worked with that disease. So first of all, like for the initial studies, he can graft those endometriosis tissue from monkeys into mice, like subcutaneously, like you can see here, treat them with hormones to create artificial menstrual cycle. So those lesions, endometriosis lesions, they respond and they grow. And you can see here, when we inject our nanoparticles, we use this fluorobeam, that imaging system, and we can see the bright signal. It comes from the lesion. So again, it just shows us that nanoparticles, they accumulate there. And also when we resected the organ, we saw the highest signal in those endometriosis lesions. And the most important here, you can see the sections of those lesions. The yellow color, those are blood vessels. They're stained with CD31 antibody, fluorescence antibody, and red color, it comes from our fluorescence nanoparticles. So what it shows us that apparently this passive targeting also work in endometriosis lesions. So the nanoparticles, they can get there and they escape from the blood vessel. We also did the treatment studies, and we show that when we increase when we expose those lesions to the near infrared light, we also can increase the intralesional temperature up to 46 degrees, similar to cancer. And what we also show here, that when we have mice treated with nanoparticles and after that we expose them to light, so we can destroy those lesions. And like, for example, here, this is the leftover tissue where the lesion was, we only see the connective tissue. In the case when we have control, and this is the histology proof that we have endometriosis lesion with stroma, glands, and so on. All right, so my collaborator, he also can graft those lesions in monkeys. And because one of the problems, as I said, monkeys, they can develop spontaneous endometriosis. But, you know, we don't have very good diagnostic tool for that. And very often it's difficult to find a monkey with endometriosis. Or when they usually, when monkeys diagnose with endometriosis, it's usually at very late stage and they have to be euthanized because they're in severe pain. So my collaborator also can graft those lesions like subcutaneously, which is very convenient for the, for the initial studies. And what we show here, so when we inject a rhesus macaque with our nanoparticles, so it accumulates in those lesions and give that signal. And on the top of that, when we shine the light, so we use the temperature cameras, special near-infrared camera, which can measure the temperature. And you can see here in 30 seconds, we can increase the temperature in the lesion to, over 50 degrees and just destroy that. So this is one of the approach when we use fluorescence imaging and photothermal therapy. One of the problems is that photothermal therapy, which I just showed you, that this therapy only can work intraoperatively or when these lesions are very close to the body surface. Because, as I said, even near-infrared light has limited tissue penetration. So, and now we started, we also in parallel, we work on another treatment, which calls magnetic hyperthermia. So how the magnetic hyperthermia works? So it's based on the concept that magnetic nanoparticles, when they exposed to the alternating magnetic field, they absorb this energy and convert it into the heat. And this approach is already in clinical trials. So the way what they do is they just inject magnetic nanoparticles like iron oxide nanoparticles into cancer tumors, and after that they expose the patient to the alternating magnetic field. And here you can see the clinical device which used for this magnetic hyperthermia. But the major problem with this conventional magnetic hyperthermia is that conventional iron oxide nanoparticles, which they use for this type of treatment, they have relatively low heating efficiency when you expose them to the magnetic field. And because of that low heating efficiency, they have to be injected intratumorally. Because and many of you, since you work in nanomedicine field, you understand that even though that passive targeting work for cancer or endometriosis, but still very, usually less than, I would say, 5% of the injected dose of nanoparticles, they accumulate in the cancer lesions, for example. So the rest of nanoparticles, they are cleared from the body. So it's very difficult to deliver significant amount of conventional iron oxide nanoparticles in order to increase the temperature to 45 or 50 degrees to treat the tumor. And my group, we work a lot on the new type of magnetic nanoparticles with high heating efficiency. Our first discovery was in 2019. We published these nanoparticles in ACS Nano. So we changed, because conventional nanoparticles, they have the spherical shape, and so we changed the shape of nanoparticles. And we also dope these iron oxide nanoparticles with cobalt and manganese in very small amount. And what we notice here, so this is the temperature profile in solution of those nanoparticles. You can see here, this is conventional iron oxide nanoparticles, and they can, for example, when you have two solutions of the same concentration, and when you expose them to the magnetic field, you can see only a small increase relatively small increase in temperature. But our nanoparticles, have way higher heating efficiency. So specific absorption rate, or SAR, this is the way to measure the heating efficiency. And our heating efficiency was like almost 4 times higher. So those iron oxide nanoparticles, usually they are covered with oleic acid and they are hydrophobic. And what we done, we also encapsulate them in our PEG-PCl nanoparticles. And I described you previously, how we encapsulate that dye in those PEG-PCl nanoparticles. So, and the way how we encapsulated those iron oxide nanoparticles in the polymeric vehicles. So we put like multiple of them inside. And here you can see TN images. So each polymeric nanoparticle has about 8 to 12 those iron oxide nanoparticles. Again, they're very stable. So what we did, we formed the nanoclusters of those iron oxide nanoparticles. And what we demonstrated that nanoclusters, because they are together, they have a higher efficiency when we only encapsulate one iron oxide nanoparticles in the polymer. So now, as usual, we tested the accumulation, and it was not a surprise because we know that PEG-PCl nanoparticles, they accumulate in cancer tumors, and they deliver iron oxide nanoparticles inside. Here you can see the section of the tumor, and this blue color, it calls portion blue staining. So it's just staining for the iron. So it showed the nanoparticles were there. And we also measured the temperature, and when we exposed those mice to the magnetic field. And what we saw here, that these other nanoparticles, for the first time, we were able to achieve the temperature up to 44 degrees. Because before our publication, all this, maximum, the tumor temperature which was achieved was less, 40 degrees or even less than that. And also we did the treatment, and so what we did, we exposed mice, I believe every 3 days to the magnetic field for like 20, 30 minutes. And after that, we measured the tumor volume. What we can see here in case of our nanoparticles, we see real very good inhibition of cancer growth, of the growth of cancer tumor when you compare it to the conventional iron oxide nanoparticles. And obviously, we checked the toxicity of this treatment, and again, we didn't see any decrease in the body weight or the change in the blood marker. So it showed that magnetic hyperthermia is safe. But since we have to use multiple, we have to expose those animals multiple times to the magnetic field. And the temperature was only 44 degrees. Our next step, what we want to do, we want to even increase further the heating efficiency of our nanoparticles. And for those who work with with iron oxide nanoparticles, you know that we use the thermal decomposition approach to make these nanoparticles. So we just put all the reagent and we heat the solution to like 300 degrees. But also there is one thing which no one tested before us. So usually you run this reaction under the nitrogen. And what we figure out that nitrogen flow during the reaction is very important. You can see here, right? So the nanoparticles were prepared like by using exactly the same procedure, but we only changed the nitrogen, the rate of nitrogen flow. And what we notice here that at the high nitrogen flow, we usually produce like smaller nanoparticles. And when we decrease the nitrogen flow, we have the nanoparticles. And what is important about that, those nanoparticles, they have the core shell structure. So we have the magnetite core and magnemite shell. And you can see here, if you have less because you have less nitrogen, so there is some residual oxygen, and that oxygen oxidize magnetite to magnemite and form this shell. So we confirm that in these nanoparticles that we have two phases for magnetite and magnemite, and what we see here that we can increase those nanoparticles with core shell structures. They have very high heating efficiency. So especially nanoparticles prepared with the low nitrogen flow, they went like to 10,000 specific absorption rate. So our previous nanoparticles, which we published in ACS Nano, were like, I don't remember, like little bit more than 2,000. So in order just to increase the heating efficiency of those nanoparticles even more, what we did, we just increased their size. So the nanoparticles which we prepared in the first step, they were about 14 nanometer, and we used them as the seed just to grow another layer. And you can see here, so we eventually end up with nanoparticles about 20 nanometer inside. And EDX analysis showed that we have iron and cobalt amount is about like 3% or so. And those nanoparticles, when we increase the size, you can see here the heating efficiency also increase. So you can see here that when we apply the magnetic field, we can increase the temperature from what 20 degrees to like 75 degrees in like less than 10 seconds. At the same time, this is the conventional nanoparticles, spherical iron oxide nanoparticles, which has way lower heating efficiency than our nanoparticles. So we did some studies, and so first, those nanoparticles, they are hydrophobic, as I said, and we convert them into hydrophilic nanoparticles. And what you can see here, these nanoparticles, we cover them with PEG-PCL, and also we make them targeted. So we conjugated LHRH peptide, and the receptor for that LHRH peptide is overexpressed in many cancer cells. And here you can see our iron oxide nanoparticles, and they cover with the polymer layer. This is PEG-PCL. They're about like 40 nanometers in size. Also, one of the important thing about these magnetic nanoparticles, they're MRI contrast agents. They're T2 weighted contrast agents. And you can see here also, this is the MRI images of mouse with the cancer tumor. This is without nanoparticles injection. This is the injection with non-targeted nanoparticles without LHRH peptide, and here with the LHRH targeted nanoparticles. So what we can see here, the drop in the signal is significantly higher in case of targeted nanoparticles. So it shows that our nanoparticles, they are MRI contrast agent. Their accumulation could be tested in the cancer tumors with MRI, and after that, the magnetic field could be applied. And here, this is the treatment study. So what we can see here, first of all, when we inject our nanoparticles at the lower dose that what we published in that ACS nano paper, we were able to increase the temperature up to 50 degrees. And you remember previous time maximum temperature was 44 degrees. And here, this is the how the tumor looked like after only one session of magnetic hyperthermia. So we also show that we can deliver these nanoparticles systemically to the metastatic ovarian cancer tumor, and we also can increase the temperature in them. We also study these nanoparticles for endometriosis treatment, because we already, from our previous work, we knew that endometriosis could be, nanoparticles, they also accumulate in endometriosis lesions. First of all, we wanted to make them targeted, and we modified our nanoparticles with KDR peptide, so the receptor for KDR receptor, also it calls VGFR receptor, is overexpressed endometriosis, but not in the endometrium. And now we use the same approach. So we use carboxylic acid the PEC-PCl was terminated with carboxylic group. We had our peptide with NH2 group. We conjugate them and we encapsulate our iron oxide nanoparticles. And we also make targeted and non-targeted nanoparticles. Next, we also check the internalization of these nanoparticles into endometriosis cells, and we definitely can see increase in case of the targeted nanoparticles when compared to non-targeted one. In endometrium, kidney, and some other cells, so we didn't see the difference. It means that the expression of those receptors is relatively low. And you can see here the qPCR data showing this is the expression of KDR mRNA in endometriosis, and this is in endometrium and kidneys. Here you can see, you can see a mouse, and this mouse has the subcutaneous endometriosis lesions. And we also use MRI. We can see the drop in the signal, MRI signal, so it shows our nanoparticle there. This is the Prussian blue staining. It also, you can see the blue color everywhere in that lesion. It shows that our nanoparticles, they accumulated in that lesion. And finally, you can see also the staining for other organs. So nanoparticles actually, if you wait long enough period of time, they can be cleared from other organs, but retain in the endometriosis tissue. And what we saw, like for example, 5 days after injection, so the nanoparticles, they only stay in the lesion. And when we apply the magnetic field, we can eliminate this lesion. We can increase, first of all, the temperature in the lesion up above 50 degrees, but at the same time, the temperature, next to the tumor was minimal, and the same story was in the lesion without nanoparticles. So magnetic field by itself doesn't increase the temperature a lot. And here you can see the treatment effect. So here, this is the mouse with 2 endometriosis lesions. And when we apply, when we expose this mouse to magnetic field for 20 minutes, so what we see, like 42 days later, so those lesions, they disappears. And this is another example that this treatment could be selective because magnetic field could be only applied, to a certain part of the body. And you can see we had like 2 lesions. That mouse was injected with our nanoparticles, and we expose only the lesion number 1. So you can see 23 days later, the lesion treated with nanoparticles and magnetic field disappeared, and we only have the lesions which was not exposed to the magnetic field. Obviously, we did the safety study, and we checked, the body weight. We did the blood work, and we didn't see any, obvious toxicity to those animals. Here, this is our ongoing studies, and this is, I told you that we can here work with rhesus macaques. And we were lucky that recently we identify monkey with spontaneous endometriosis, because we always wanted to see if the nanoparticles really can go to endometriosis lesions, because whatever the model which we used before, so we grafted those lesions, so we just put, surgically put them under the skin, and obviously natural lesions, they could be different. And what we see here, so when we got the rhesus macaque with spontaneous endometriosis, this is MRI image, and this red circle just depict the lesion. So after 24 hours after our nanoparticles were injected, so we took another MRI scan, and you can see here there is a drop in MRI signal. So it means the nanoparticles there. And also when we collected that lesion post-euthanasia and did the histology, we can see that there is a lot of blue color. So when we stain with Prussian blue, so it shows that iron oxide there. And the most important part, we can increase the temperature when we expose this lesion to magnetic field. So we can go like more than 46 degrees. So that's kind of our ongoing study, and it was, very exciting part for us because we can see that nanoparticles, they also work in spontaneous lesions, in like natural endometriosis lesions. And the most important part, we also show that these nanoparticles, they also save in big animals like rhesus macaques. And this is the story about magnetic hyperthermia. Do I still have a few minutes?
Yeah, we have still
Okay.
time.
Okay, so and at the end, I just want to show you the last project. And the last project, I just want to shift gear a little bit and show you the gene therapy and how we use specifically messenger RNA therapy for cancer cachexia. So first of all, what is cachexia? Cachexia, actually, this is, you saw that, you probably noticed previously that many cancer patients, they look, they look skinny. And many of them, they don't lose body weight and muscle mass from not only from chemotherapy, but actually this is what calls cachexia. So this is due to cancer signaling. Those patients, they spontaneously lose the muscle mass. And so technically 50% of cancer patients, they have this cancer cachexia. And cancer cachexia defined as the involuntary loss of body mass by more than 5% in 12 months. So about 50% of cancer patients, they have cachexia, and especially cachexia is very pronounced in patients with advanced cancer, like pancreatic cancer, for example. So 80% of those patients, they have cachexia. And when I started to work with cachexia, I learned many scary facts about that. So I learned that 30% of cancer patients, they die from cachexia, but not from the tumor by itself. So meaning they lose muscle mass and they die from that. And also when cachectic patients, they are very weak, they cannot move very well. Quality of their life is very bad. Also, for those patients, since they are very weak, for them it's very difficult to handle any treatment like chemotherapy, surgery, and so on. And they, and especially, they toxicity really toxic effects are very enhanced in those patients. And currently, there is no treatment for cancer cachexia. So what we wanted to do, we wanted to develop the treatment which can increase a muscle mass or at least preserve the muscle mass in those patients. And the idea of that treatment was that this treatment, so they should the patient should gain the muscle mass without any special diet or exercise, because actually diet cannot, reverse the cachexia really in cancer patients. And also those patients, cancer patients, they lose appetite, and for them, they cannot increase the muscle mass. So the way how we decided to approach, what we know from the biology, that in our body, muscle cells, they produce and secrete such proteins like activin A and myostatin. So those secreted proteins, they bind to the activin receptors on the muscle cells, and they actually inhibit the proliferation of those muscle cells. And those proteins, they are very important. So, and here you can, and also I just wanted to mention that especially the level of activin A, one of those proteins which negatively decrease muscle mass, is very high. Serum level is very high in cancer patients. Here, this is just an example how that, for example, those proteins are important. So this is the cattle, and you can see how bulky it is. And one of the reasons for that, because it has the mutated myostatin, the protein which decreases the muscle mass. So because of that mutation, it can grow big and have lots of muscle. So now, obviously, the therapy which we wanted to have, we wanted to decrease the level of activin A, for example, in cancer patients. So how do we do that? We also know that in our body, we have another protein which calls follistatin. And that follistatin is secreted protein, and what it can do, it can, in the systemic circulation, it can bind to activin A or myostatin and prevent the interaction with those receptors on the muscle cells. So after that, we got an idea that what we want to develop the therapy based on the messenger RNA. So, and I'm pretty sure now many of you familiar with messenger RNA approach, because especially after this COVID vaccines, right, like Moderna or Pfizer COVID vaccine. So our idea was like this. We wanted to have messenger RNA coded for the follistatin protein. And after that, so we put this messenger RNA into the nanoparticles because, you know, you need a delivery vehicle for mRNA or siRNA, all these molecules, because they are not very stable in the systemic circulation. So those nanoparticles, after systemic administration, they deliver the messenger RNA, for example, to cancer cells or to the liver cells. And those cells, they use messenger RNA, and they started to use their own machinery. Those cells, they started to produce follistatin and secrete the follistatin in the bloodstream. So, and since we increase that follistatin, we can decrease that follistatin can bind the activin A and myostatin and help at least to preserve the muscle mass or increase the muscle mass. So here we use the lipid nanoparticles similar to this Pfizer and Moderna nanoparticles, and we loaded them with our follistatin messenger RNA, and we use this ovarian cancer model. You can see this is the mouse without cancer, and this is a mouse with ovarian cancer. First of all, you can see the abdominal cavity is huge. So that mouse has lots of ascites. That's a fluid with cancer cells. And also you can see at the muscle, right? So that mouse, has a cachexia because it loses the muscle mass. And the most important, the mice with cancer tumors, they have more than like 3 times higher serum level of activin A, of that protein, which decreases the muscle mass. So now what we did, first of all, we just wanted to test it if, in general, this idea can work and nanoparticles can deliver any messenger RNA to for example, to cancer cells. And what we did, we used the luciferase mRNA. So you remember the luciferase mRNA can get into the cells. So luciferase, form and can generate the bioluminescence. And when we injected our mice with nanoparticles containing luciferase mRNA, what we can see here that especially in these ascites where we have lots of cancer cells, we can see strong bioluminescent signals. It means our nanoparticles, they get into the cancer cells, and those cancer cells, they uptake them, and they can produce the protein which was coded by that mRNA. Now, the last thing what I just wanted to show you, the treatment part. So, for example, when we inject now our mice with follistatin mRNA, so here you can see we measure using ELISA, we measure the follistatin and activin level. So first of all, those blue bars show the level of follistatin in healthy mice. So this is the mice treated with PBS only, and this one with our nanoparticles. And you can see we can increase the level of follistatin in those mice. The same story here in cancer mice. So cancer mice, they have slightly also higher follistatin level because the body try to compensate for that activin A. And we also, in cancer mice, we also can increase the follistatin level. The most importantly, we can in cancer case, you can see this red bar when cancer mice were injected with PBS and cancer mice injected with messenger RNA. So what we can see here that we can really decrease that activin A level. And later on, when we injected our mice for like 9 days for 7 injection of messenger RNA, and we measured the muscle mass, what we saw here, right? So this, first of all, we can see this is the healthy mouse, and this is gastric muscles in healthy mouse, the weight, and this is in cancer mouse. So in cancer mouse, this muscle mass decreased like more than like 35%. And when we started to treat our cancer mice with these messenger RNA nanoparticles, we can basically increase the muscle mass almost to the level which was in the healthy mice. So just this is the example.
Professor.
Okay, so yeah, this is my last slide. We also show that this therapy has the anti-cancer effect. It decreased the level of ascites. And now this is what I wanted to tell you about this project. Finally, I would like to thank my lab members, my collaborator. I would like to thank NIH for the financial support, and thank you so much for your kind attention. And I will be more than happy to answer your questions.
Thank you, Professor Taratula. What an interesting topics. I almost forget to stop you because the time is end. But we have 3 questions. I will read the questions. First from Professor Hakim Bangun. Why nanoparticles containing fluorescence dye can accumulate in the tumor cells even without using a ligand or targeting agent? And then
okay.
You ask one by one, or
yeah. So let me answer the first question.
Okay.
So first of all, let me go. I have one slide here when I talk about where is it? I'm sorry, I have to find that slide and won't. That's okay. So here we go. So why nanoparticles accumulate in cancer tumors? First of all, they don't need targeting ligand to accumulate in the cancer tumors, because the major mechanism for the accumulation of nanoparticles in cancer tumor, this is the passive targeting. As I said, so cancer cells, they have a highly developed blood vasculature, and those blood vasculature is leaky blood vasculature. And what happened, nanoparticles, when they reach the cancer blood vasculature, they can escape from those pores or fenestration in those blood vessels and accumulate in the cancer tissue. And cancer tissue also have the impaired lymphatic drainage. So the nanoparticles, they stay longer in cancer tissue than, for example, in some other organs. Active targeting, actually, this is only complementary to the passive targeting. So those nanoparticles, first of all, they have to be there in the cancer tumor. They have to be passive. They have to passively reach the cancer cells in order to take the advantage of the active targeting. So and in this case, for example, some peptides, they can interact with receptor of cancer cells. So if nanoparticles, they don't go to tumor, but for example, they end up in the liver, and if the passive targeting doesn't work, and in some cases it doesn't work, so the active targeting is not going to help. So the nanoparticles, technically the answer is that passive targeting plays the most important role. Active targeting actually help the passive targeting, and actually active targeting is responsible for the intracellular delivery of the or the intracellular uptake of the nanoparticles. I hope I was able to answer your question.
Okay. Okay. I will read the second participant. Is there any specific requirement for a compound that can be used for the nanoparticle theranostic agent? And the second question is, what is the critical point when develop a nanoparticle theranostic agents?
All right, so
Yeah,
I mean, first of all, probably I didn't even specify in my lecture, in my presentation. Theranostic, it stands for therapy and diagnostic. So this is the nanoparticles which can, do both diagnose and, and treat. And what the requirement could be. Like in our case, we had I had the example when you had one molecule, one dye molecule, which can do both, right? Imaging and phototherapy. So yeah, the requirement, as I said, even for any agent, including theranostic agents, they have to be simple in preparation, because some theranostic nanoparticles you can make when you can combine like 2-3 agents together, but it adds some complexity to that. And what is the critical point when develop nanoparticles theranostic agent? The critical point, I guess, you have to have simple nanoparticles, and these nanoparticles, they should have appropriate properties like size, surface charge, surface coating, because in order for them to take advantage of this passive targeting, they have to accumulate there. And obviously, all these components which are there, they should be efficient. They should be efficient contrast agent and efficient therapeutic agents. So that's what I would say.
Okay, thank you, Professor Taratula. Ladies and gentlemen, we are already at the end of this session. Thank you once again, Professor Taratula, as you have shared your knowledge in this session. Yeah.
Yeah, and thank you again. As I said, it was my great pleasure. Thank you for inviting me, and and
thank you very much, Professor Taratula.
My pleasure. Yeah. Thank you.
Thank you very much. Yeah, we can meet sometime.
Yeah. Okay, I pass the screen to the moderator.
Yeah, thank you very much. Yeah,
thank you.
Yeah.
Okay, thank you for Prof. Taratula for the interesting talk, and thank you for Prof. Poppy for nice arrangement of this session.
Thank you so much.
Ladies and gentlemen, then let's proceed to the next session. The speaker of this session is Dr. Dwi Lestari from Faculty of Pharmacy, Universitas Sumatera Utara. This session will be by Professor Johnson Sidalahi from the Faculty of Pharmacy, Universitas Sumatera Utara. Professor Johnson is one of the professors in the Faculty of Pharmacy, Universitas Sumatera Utara. Professor Johnson's research interests are at the nutraceutical field, particularly in probiotics. Well, please welcome Professor Johnson. Time is yours.
Okay, thank you very much for this session, for the MC. Good morning, ladies and gentlemen. Especially good morning, Dr. Dwi Lestari. So next session will be presentation delivered by Dr. Dwi Lestari. But before her presentation, let me read her CV. Dr. Lestari, the graduate of bachelor degree is from University Bandung Institute of Technology, and then master degree from University of Sumatera, Indonesia, and then doctoral degree she obtained from University of Twente, Netherlands. And position and employment at the moment as a reviewer of research journal, and also a member of Ethics Committee on Health Research, University of Sumatera, head of Pharmaceutical Laboratory, Faculty of Pharmacy, University of North Sumatra, and lecturer in Pharmacy, Faculty of Pharmacy, University of North Sumatra, pharmacist and pharmacy supervisor, Sentry Healthcare, Sultan Plaza, Bandung, and also internship pharmacist, Sentry Health Plaza, Senayan, Jakarta. So this session will be presented by Dr. Dewi Lestari. So we invite Dr. Lestari to present your topic. Please, time is yours. Thank you.
Okay, thank you, Prof. Jensen, for your introduction, and good morning, everybody. And also good morning, Professor Tarantula. It's nice to meet you here, and it's yeah, I have very enjoyed your presentation.
Good
And
morning.
I think
thank you.
Yeah, it's very nice presentation. I think, and let me to extend this talk about nanomedicine. Can I share my presentation? Yeah, okay. So this morning I will continue this seminar with the talk of nanomedicines in the area of the cancer chemotherapy, especially, and I will share you my research in the last few years, especially the interaction of nanomedicine in this cancer environment. It's called the tumor stroma, and we'll see how it affect the efficacy of nanomedicines. Okay, so it's that what we will talk today, and then let me a bit back to the basic knowledge of nanomedicine, that we know it's the application of nanotechnology to medicines using engineered nanostructures. It can be any medicines, not only for cancer, but also other disease, especially chronic and also life-threatening disease, as well as for imaging and diagnosis, as explained by Professor Tarantura before. So, and also for then we can improve also the quality of patient life. And especially, of course, it's for treatment that it allows for delivering medication to a specific site. That's, we already know that from our talk this morning. Yeah, so why it can do that things that because of the size of this nanomedicine scale is of course in nanometer size. It's to reach the, especially for the cancer cells, it's about 1 to 100 nanometers. It will work, and maybe up to 200, as we know from previous session, that this gap in the cancer cell about 300 to 700 sets. But yeah, for the safe, we usually develop this nanomedicine in the range of 10 to 100 nanometers, and it includes different range of nanomedicine platform. So you can see that. So it's compared to another things around, like a virus. It's more even with bacterium, and even more with the tennis ball. So it's very, very small that we cannot see. So why we go for nanomedicines? As also explained before by Professor Tarantura, that this nanomedicine give several benefits that it can encounter current cancer therapeutic pitfalls, which are it's not site specific usually, and then gives enormous side effects. So people now, especially for cancer treatment, we go for these nanomedicines. So these nanomedicines will target only the diseased organ without destroying healthy tissue, and by its due to their nanometer size, it can extravasate through leaky blood vasculature, as also shown by Professor Tarantura before. Leaky blood vasculature present into tumors, and it will remain there in this tumor microenvironment due to pure lymphatic intratumoral fluid flow. So it's the phenomenon that we call as EPR effect, enhanced permeability and retention. And so we can also design to deliver chemotherapeutic as actively targeting nanomedicine by surface functionalization, as also do by Professor Tarantura research by surface functionalization, and also targeting region. Yeah, so it's a range of nanomedicine platform that we can tune physical and chemical properties of nanomedicine. We can do surface functionalization for longer circulation using polyethylene glycol chains or specific tissue cell targeting using ligands that we can attach targeting ligand that will attach to the receptors that specifically produce very much in specific tumor. And also with the composition, we can have like lipid-based nanomedicines of liposome or anorganic-based nanoparticles as gold nanoparticles, iron oxide nanoparticles, or polymeric-based nanoparticles such as polymeric micelles and etc. And also we can tune the size, shape, or stiffness, porosity, elasticity, and roughness of our nanomedicine, so it has certain physical properties. So since it's discovered this nanomedicine concept by the discovery of liposome in 1964, and then it's continued, and about after 30 years, it's in 1995, the first liposome approved by FDA to be used in clinical. It's a doxil, the doxorubicin loaded liposome. And now it's growing and still growing until now for the nanomedicine, this nanomedicine technology. Yeah, so it's from the data in 2020 that until now, maybe it's now it's more than this. It's more than 100 nanomedicine approved, either by FDA or another agencies in European, etc. And in clinical trials, it's maybe now it's reached about more than 500 to 600 nanomedicine in clinical trial. So from this, we can see it's a bit, yeah, only a fraction of the therapeutics that seems promising in the experimental stage will eventually reach the clinic. So instead of the successfully story of this experimental medicine in the lab that reach the clinical, it's only a fraction. Why? Maybe one of the result of this low success rate is because of the fact that the cancer cells actually are not alone. They have the this tumor microenvironment surrounding of tumor cells. So what is this tumor microenvironment? It's actually in a tumor, cancer cells only occupy about like less than 10% of this tumor microenvironment, and more than 90% it consists of stroma. What is stroma? Stroma is the structural components that holds the tumor together. So it consists of different cell types such as cancer-associated fibroblasts, called CAFs, endothelial cells, inflammatory immune cells, and etc. And the important thing that it's contain a large amount of extracellular matrix material that forms a highly fibrotic and dense structures. And this stroma will support tumor progression and increases the resistance to treatment. Like it will hinder the so it will affect. So we see this is the illustrative image of breast cancer, one of the solid tumor that contains a lot of the stroma component. So we see in the left side that we have the structure of normal breast tissue, that the cell lining in the vasculature it's well arranged, and we have the quiescent fibroblasts and also the nice arrangement of tissue-specific extracellular matrix. But once it's forced to for the gene mutation maybe, and then it's will change to have a loss of architecture. So the like a quiescent fibroblast will transform to more to transform to cancer-associated fibroblasts, and it's different with different characteristic, and also the extracellular matrix is now more aberrant, and also it will produce many cytokines and also ROS, and then it will promote the tumor progression more. And another, and it proved by human breast tumor tissue samples that it's shown by staining. By staining, we see these trauma markers. We identify the alpha SMA and collagen 1 as trauma markers here, shown by brown color. You see it's most of the, this picture is all brown, means that the breast tumors are enriched with stroma tissue. It's from the sample from human breast tumor tissue. And the stroma components occupy about 50% of the breast cancer. And we see another solid tumor in pancreatic ductal adenocarcinoma that also have a lot of this fibrotic and highly dense stroma. Here, the cancer cells in the middle, and it's surrounded by the extracellular matrix and stromal components, where then it can give the high interstitial pressure, so that the blood vessel will collapse here. Then the consequence is that the EPR effect may not work here, because there's a physical hindrance here by the highly interstitial pressure from the stromal components here. So it's the vasculature, although it's leaky, but it's then hindered by the highly dense stroma. And also there are immunosuppression phenomenon here. So it's the more clearly illustrative images, that if it is this tumor cell, and then if it's happened that nanoparticles can enter the this leaking vasculature, but then it will face the barrier from this stromal component, and then it will it will hinder the nanoparticle to reach the tumor cell. And it's also proved from another research from the group of Professor Jain that study this nanomedicine penetration after extravasation from the tumor vasculature. It's with even for the small size liposome, this with doxil that injected to the mice, the liposome size of 90 nanometer. So after extravasation from vasculature, they did not move far from the vasculature. They're still near the vasculature. And also for with the experiment with HSV, this is a virus particle of 150 nanometer size. It also shown the same, and also even for the smaller size of 60 nanometers, so the same after extravasation. And after 2 hours, they are still near the vasculature. It means that this stromal component hinder the penetration of nanomedicine. So then therefore, this tumor stroma component should be integrated then in our study, any study platform during nanomedicine development, even from the early stage. So if we see the paths of experiment of nanomedicine, we usually from the lab scale, we synthesize and develop this nanomedicine platform, and then we test usually in 2D culture, do in vitro studies, and then usually we then go for the preclinical study towards the small animals like in mice, and then go further with the bigger animals, and then finally for the clinical, and then approve. Yeah, that's what we hope. But here, if in vitro study, what happened is 2D monoculture that we usually use with the cell culture, conventional cell culture, compared to the in vivo situation that already shown previously, that we have here in this in vivo or even in biological tumor in human, that we have this stromal component. So we need something in between that can bridge these 2D models to the in vivo models. So, but it can still affordable in the lab scales. So we need some kind of these 3D models for in vitro experimental that can resemble the clinical complexity and heterogeneity in vivo situation or in biological tumor in human. So in this 3D model in vitro, hope that can bridge the research results from 2D model before going for the in vivo that will cost us a lot, and also time consuming, and etc. So we need something in between, still in vitro at high throughput, easy to read out and something. So we need this 3D model. You can see that with 3D model, it will allow for co-culture different cells. Then we can mimicking the clinical tumor heterogeneity, so that we can grow together these tumor cells and the fibroblasts, so it can produce relevant ECM and etc. Then it will suit the study drug or nanomedicine penetration. And to encounter what is lack from the 2D model, So now we go for the 3D. What we have so far in 3D in vitro models. In general, we can develop this 3D in vitro by 2 approaches. First, by cell cell-based approaches, we can develop spheroids or organoids, or by engineering structure. We use bioprinting technology, and also we grow it with the scaffolds to develop this 3D in vitro model. So what we will expect that we can expect from this 3D spheroid model? This it can mimic the tumor microenvironment. So in this 3D, there is a different situation between the core, the cells in the core of the spheroids, this ball-like structure, and at the periphery. So such as this hexagon gradient, which in the middle is usually will exist the hypoxia situation, the same as the like in biological clinical cancer, that there's a hypoxia in the middle of the big cancer or big tumor, and also there's a gradation of lactate accumulation, and also the apoptosis, and also ATP distribution, and also different proliferation activity between the core and the peripheral cells. So then in our research, we developed this 3D model in vitro to study. It is aimed to develop a tool to study nanomedicine by forming the spheroids. So we develop first, we do stages of develop, and then we characterize to check whether it's our model can be used for nanomedicine study. So then we use it for investigating the penetration of nanoparticle, and also to study the impact of tumor stroma on penetration and efficacy. So it's a study has been published in GCR, Journal of Control Release. Yes. So see what form we use. We only use the normal patchy disease found in any labs, especially as cell culture labs, and then we stamp hot emboss to this plastic to the bottom, so we can have this micro wells. This micro wells size of 200 micrometer and 400 micrometer size, where we place the cancer cells and stromal component. We use here from mice 3 fibroblasts to grow together in this dish, hot embossed stem dish. So it will grow together, and they will generate spheroids by naturally force. They will aggregate by time. So after we grow it after 48 hours, it will grow as spheroids, compact spheroids. So in our experiment, we compare cancer cells only, and also we do this co-culture 3:3:4:1 in different ratio, and so for the comparison only the fibroblasts. They all form as compact spheroids. So it's the growth that these loose cells in 0 times 0. And then after 24 hours, they already aggregates each other, and it's more compact after 48 hours. And we use these 48 hours old spheroids for further study. So to check whether these spheroids produce the stromal component, etc., and we then characterize of the spheroids for the stromal marker. which are alpha SMA and collagen 1. It's shown by this red color, and then quantify at after 48 hours, it contains alpha SMA and collagen about 50%, which is in line with what we found in human breast tumor tissue analysis. So it's from the patient samples, and it's from our spheroids. So it's content the stromal marker about in the same amount. So we decided to use this then for further study, and we also do the analysis by QRT-PCR. Then we see another marker for its EMT marker, and also stromal marker, and also the epithelial marker. And what is interesting for tumor cells that we grow in monoculture and then in spheroids, it's only cancer cells. Cancer cells without fibroblast or stromal component. It's shown there is increasing of alpha SMA in 3D or in 3D structure. So alpha SMA means the increase means that there is transformation of the cancer cell from epithelial to mesenchymal cells. Here, and then so 3D itself growing as 3D culture already changed the feature of the cancer cells 41. And then this model, this spheroid model, we use to study the penetration of nanomedicine. We use the silicon nanoparticles that are commercially available, so we can do the experiment with different size of silicon nanoparticles and also with different charts. So we can study the effect of charts and also size to the penetration of nanomedicine. And it's fluorescently labeled, so we can trace the penetration, how deep it is, and how much that penetrates into the spheroids by fluorescence imaging. So here we compare the 30 nanometer and 100 nanometer size, and then for 24 hours. So we incubate the silicon nanoparticles into the spheroid culture here. And then after 24 hours, we see this first line, it's only cancer cells that both sides that can penetrate deeper. And compared to the co-culture, it's contain fibroblasts and 41 in same amount. Yeah, it's still penetrate, but not so far as this 41 only. But even in fibroblasts only, they almost cannot enter the spheroids. So it's the quantification of the this penetration. And so in this experiment, we do in animal cell lines. It's from mice breast tumor cells 41 and mice fibroblast cells. We did also to form spheroids from human cell lines, which are from breast tumor models. We use MDA-MB231, and we use also pancreatic tumor model from human. We use PEN1 as cancer cells and PSC cells, pancreatic stellate cells. And for breast tumor model, we use BJ fibroblast cells. And it shows the same, more or less the same result as in animal cell lines, that the penetration is hindered in a spheroid containing of stroma. And between 30 and 100, of course, the 30, it penetrated more than 100 nanometer, the larger size. And then we also do penetration study with the PLGA nanoparticles. It's polymeric-based nanoparticles, labeled with size 5, and it shows more or less also the same results. It's from cancer cells only. It can penetrate the PLGA size of about 150 nanometers. So it's bigger than the silica one. And also, so the same results even in 5:1, as it's much more bigger, 150, it's almost cannot enter the spheroids, the stromal containing spheroid. So from this first study, we can conclude that our 3D in vitro model confirms that stroma hindered nanoparticle penetration, in which smaller nanoparticles penetrate deeper than larger NPS. And then we continue to study with the paclitaxel-loaded polymeric nanoparticles. This paclitaxel is the first-line drug for the breast cancer treatment that we load in polymeric nanoparticles, which are micelles, in polymeric micelles and in polymeric nanoparticles. Between these two nanoparticles, they have different characteristics. Also, by the polymeric that form them, and also the size. So polymeric micelles has the characteristic of the size of about 70 nanometers, and PLGA nanoparticles form the bigger size, about 150 nanometers. And also, they have different charts. So polymeric micelle almost neutral, and while the PLGA nanoparticles more negative. So it's from the zeta potential. So first we check the uptake of these two nanoparticles in 41 cells monolayer, and both side 3. And we label the micelles and nanoparticles with side 3 to allow us to track them by fluorescence imaging. So it's shown that both side 3 PMC, polymeric micelles, and side 3 PLGA nanoparticles are taken up by 41 cells. So we confirm first that 41 cells can take up the both nanoparticles. Then we form the spheroid, and now in the step we characterize them better with the photomicroscopy, so we can image them through the core, that it's the picture from the top, and then if we go further, imaging to inside this spheroid structure, it's in the middle, they form streets, and then also until the back. So with this 2-photon microscopy, it's possible for us to see the growth of the cell inside, and also the penetration of nanomedicine later. Yeah. And then same, and then also we do the staining. This the imaging from the 2-photon. It's actually the video, so that So it's how it looks when we go for this photon microscopy. You can see the video. Sorry, you can see the video?
Yes, we can see.
Oh, okay. Yeah.
Yeah.
That's yeah, it's the nice to have this photon so we can characterize and we can check until the core for the nanomedicine penetration, also how the cell arrangement and yeah, how it's arranged inside this spheroid. So then we do another penetration study that we compare site 3. Now it's with polymeric micelles and also PLGA nanoparticles. Then as the polymeric micelle has smaller size of 70 nanometer, it showed a better penetration than site 3 PLGA nanoparticles. Yeah, so it's 70 nanometer versus 150 respectively. So now this we can take picture of fluorescence and then we do quantify with image and that's so we can quantify them. And it showed that site 3 polymeric micelles showed a better penetration. And then we continue with the efficacy study in 2D culture first that we found both of 41 only 33 this is 2D culture and the co-culture they shown until the variability they can decrease by about 80%, and here also, and in for yeah, and between paclitaxel free and then paclitaxel loaded polymeric micelle and paclitaxel loaded in PLGA nanoparticles, paclitaxel loaded in polymeric micelle so the better efficacy in all cultures, in cancer cell only, in fibroblast only, and in co-culture. And then the paclitaxel loaded in PLGA nanoparticles, which has a bigger size, 150, they so the less effective compared to paclitaxel and paclitaxel loaded polymeric micelle. It's in 2D cultures. And how is it in 3D cultures? They saw here only about only 20 and 20%. It's in it's for paclitaxel and it's for
Sorry, Doctor, they will study the time several minutes left.
Yeah, okay. Yeah, it's almost done. Here we saw that there is a decreased efficacy shown by 3D cultures. And for polymeric micelle, it's on cell debris surrounding spheroids, which is likely due to the erosion of the cell in the outer cell. And we can also, what's interesting that we check also for gene expression for drug resistant. It's for BCL2 and ABGC2. So we found that in 3D culture spheroid, we found the increase of the resistant markers here, BCL2 and ABGC2. So this model, and so the resistance developed by this tumor microenvironment. So this tumor cell stroma interaction lead to induction of PMT shown by alpha SMA and hypoxia shown by HIF1 alpha, as well as drug resistance shown by BCL2 and ABGC2 markers. and then poor efficacy of paclitaxel and nanoparticles in both 3D mono and co-culture spheroids. So in this we can conclude that this study demonstrates the significance of 3D culture model for the evaluation of nanomedicine before testing them in preclinical tumor models. So it's needs for different strategies to overcome tumor stroma models to enhance nanomedicine penetration, and hope that these tumor models can be further developed and adapted to according to the particular tumor characteristic before it's going further for the clinical translation of preclinical research findings. So I would like to thanks to the respectivity and also University of Sumatera Utara for the funding and also the collaboration, and this research it's done by collaboration with University of Twente, especially with engineer therapeutics section in Department of Advanced Organ Bioengineering Therapeutics, and also with the Applied Microfluidics or Bioengineering Research Amber Group and Biomedical Photonic Imaging and also with University. And this research has been published in Drug Delivery and Translational Research, and I would like to thank you for your attention, and we can receive for the question, comments, and discussion. Thank you.
Okay, thank you, Dr. Dewi Lestari. So I invite to the participant who want to ask a question to Dr. Dewi Lestari, please. But before the question asked to you, so I want to ask you a simple question. What is the relation in this study, in your study, you have done related to the apoptosis process?
The apoptosis. Oh yeah. So this study actually for the efficacy study, I check also for the apoptosis actually that this paclitaxel, this with the paclitaxel loaded nanoparticles. So the apoptosis is mean that we add the chemotherapeutics, right? Yeah. So in this study, we indeed do the apoptosis study for the efficacy of paclitaxel loaded this polymeric micelle and PLGA nanoparticles, and then we also tested both in 2D and 3D culture, and then we compare, and it showed that it's shown less in this 3D. So for this, actually, with this model, we can better drug screening that need to go further for the preclinical study in vivo, in animal. So we can, we can show the apoptosis from the study with this 3D spheroid model. Almost the same with the region we use for the 2D culture.
Okay, thank you. Okay, another question, simple one. So what is the nutrient or medium for the cancer to grow? So we can prepare. What is the nutrient or medium to the cancer to grow?
Oh, okay. As I use 41 cells, it's cancer cells and fibroblasts. So for 41, we use FPMI medium that it contains all the nutrient it needs. And for the fibroblasts, I use DMM, Dolbeco medium. And for co-culture 41 and C3, I use the DMM medium. So this special medium for cell culture, it's already contain the medium nutrient for cell to grow. So
Mute.
Sorry, Prof. Tarantula will ask you a question. Please.
Yeah, I have a question. Yeah, thank you so much for noticing
Yeah,
me. Dr. Lasteri, thank you for a nice presentation. I think you work on a very important problem. I have a question. So apparently the size of nanoparticles has an influence on penetration into the stroma. What do you think about the charge like zeta potential? And also, do you think the material of nanoparticles has some influence on that? Like, for instance, polymeric nanoparticles, do they have different penetration than silicon, porous silicon nanoparticles, for example?
Okay, thank you, Professor Tarantula, for the your question. Yeah, it's interesting, and it's actually the same question that we always ask ourselves to ourselves. Yeah, in this study, as shown by these silicon nanoparticles, that we use different charts of silicon nanoparticle. We use about first 20 millivolt, and then more negative 40. Based on our research, in this research, the one that's have more negative, it penetrates more, and then the last one is lesser. In our hypothesis that for there is a let's do we haven't really looked into it, but from our hypothesis that we can say that the more positive charge, it somehow more interaction with the cells surroundings, so then it like they attach, or maybe it's like hinder the penetration, something like that. And for the materials, some materials with the polymeric micelle, we use this polymer is developed in the lab in Utara. It can form the smaller size of nanoparticles, so it will tune the size of the nanoparticle itself. So with the polymeric micelle, usually we will have the more lesser size, the more smaller size, like we can reach 30, 50, about 70. I think in our research, the biggest one from polymeric micelle, we got the 70 nanometers. But like for the polymeric, like PLGA, polylactic glycolic acid, we can only reach about 150 nanometers. It's based on the solvent we use. So different materials and also different solvent we use when we develop these nanoparticles, it will form different size or specific size of nanoparticles, something like that. Then the implication, of course, with the smaller size, then we will have the better penetration. That's something. So this material will affect the size of the nanomedicine we form.
Yeah, thank you, Dr. Lasteri. I really appreciate your work, because you do very systematic work, and this fundamental knowledge, they are really needed in our field in order to understand the real properties of nanoparticles, because I really like your explanation about negative charge and why it improves the penetration of the stroma. I think, just keep up doing your good work. I appreciate that. Thank you.
Thank you, Professor Tarantula. We still look further with these charts, because from our study also that another research study also find even the opposites of the, but it depends on also with the material of the nanoparticles, thing like that.
Okay, thank you for your question, Tarantula. And the time is out, so thank you for your interesting presentation, Dr. Lasteri. Thank you.
Yes.
I give it back to MC.
Okay, thank you, Dr. Dwi Lestari, for the great presentation. And thank you also for Prof. Johnson for moderating this session. Ladies and gentlemen, now let's proceed to the next session. The speaker for this session is Professor Dr. Anayanti Arianto from Faculty of Pharmacy, Universitas Sumatera Utara. This session will be moderated by Mrs. Lia Laila. Mrs. Lia Laila is one of the lecturers from the Faculty of Pharmacy, Universitas Sumatera Utara. Her area of research interest is in pharmaceutical technology, particularly in the drug delivery system. Mrs. Lia, time is yours.
Thank you, Mr. Bayu, for the introduction. Good morning, ladies and gentlemen. I will continue this session. Our next speaker is Professor Anayanti Arianto. Good morning, Prof. Anayanti.
Good morning.
Great pleasure to have you here in this seminar. Professor Anayanti is one of the professors at the Faculty of Pharmacy, Universitas Sumatera Utara. He is also one of the members of Nanomedicine Center of Innovation, Universitas Sumatera Utara. Professor Anayanti obtained her master and doctoral degree from Universitas Sumatera Utara. He had conducted many research and worked numbers of publications in pharmaceutical field. A research area of interest in the development of nano-dosage form. In the next 35 minutes, Professor Anayanti will deliver her presentation with the title Herbal Nanogels for Tropical Delivery, Anti-Inflammatory and Anti-Aging. And then after that will be followed by question and answer sessions. During the presentation, all the participants may also type the question in the chat box. Okay, without further ado, now please welcome Professor Anayanti Arianto. Time is yours.
Thank you for moderator, Mrs. Lia Laila. Now I will share screen, my slide. Can you see the slide?
Not yet, Prof.
Not yet.
Yeah. Oh,
Prof.
Sorry. Ini nggak share screen. Nggak share-nya. Saya keluar.
Okay, Prof, we can now see your.
Huh?
Just now we have already seen your. Okay.
You already seen
Now
the
you
slide?
can. Yeah.
Oh, yes.
But it
Thank
have
you.
not been in a slideshow mode.
A slideshow. Oh,
Yes.
yes.
Thank you, Prof.
Yeah.
Okay, thank you, Prof.
Thank you. Yeah. So the topic that I want to talk now is herbal nanogel for topical delivery, anti-inflammatory, and skin anti-aging. Our research use herbal or plants. Plants also have emerged as a good source of cosmetic ingredients that meet the characteristic of efficiency and safety with fewer side effects, thus increasing replacing synthetic ingredients. Accordingly, there is a growing demand for natural-based cosmetic worldwide. Based on hereditary experience, the use of herbal plant is considered effective enough to treat various kinds of disease. Topical use of herbal anti-inflammatory drug will reduce side effects from long-term use of oral anti-inflammatory drugs such as NSAIDs. This drug can cause side effects of irritation and bleeding in the digestive tract, disrupting kidney and liver function. So topical gas have emerged as a suitable vehicle for delivering and releasing herbal bioactive compounds because their texture is not greasy and easier to rinse from the skin than cream or ointment preparation, but particle size will affect the penetration of the active ingredient through the skin. So nanogel is more effective if made in a nanogel preparation. Nanogel are three-dimensional hydrogel material in the nanoscale side range formed by cross-linked soluble polymer network with a high capacity to hold water. Nanogel will provide a higher efficacy than gel with drug distribution over a wider surface area due to its small particle size. A nanosite has better stability, increased penetration, solubility, bioavailability, and increased therapeutic activity. And so longer retention of the drug loaded because the contact of the product with the skin and the release period of the active ingredient are prolonged. Possess biocompatibility because similar to biological tissue and degradability properties, and this delivery system does not usually produce an immunological response. A nanosuspension formulation show an increase in the amount of the drug accumulation in the skin epidermis up to 5 times compared to ordinary gel formulation. This nanogel provide higher efficacy compared to gel with a wider distribution of the drug on the surface area due to the small particle size, for example, in the nanoscale, according to Chevron et al. 2011. So this slide, we can see the many applications of nanogels. For example, as anti-inflammatory, anti-aging, wound healing, anti-cancer, autoimmune disease like Alzheimer's disease, and neurodegenerative disease, diabetes, vaginal drug delivery, and etc. And now I want to talk about the first is nanogel as an anti-inflammatory. Inflammation is a common and very important pathological process, often manifests as a red, swollen, hot, and painful. It is a defensive response of living tissue with a vascular system to various damage factors, and it is closely related to various diseases such as asthma, rhinitis, arthritis, and arteriosclerosis. If inflammation is slow to develop and is not controlled, it can cause serious illness. Currently, the drug is used for treatment of skin inflammation, corticosteroid and non-steroid anti-inflammatory drugs, for example, NSAIDs. Yeah. However, routine treatment of this inflammatory disease using this substance over a long period of time causes side effects, including pruritus, irritation, dry skin, allergy, contact dermatitis, irritation, and bleeding in the digestive tract, reducing kidney and liver function. So it is necessary to seek traditional medicine as an alternative to inflammatory therapy. One of the plan, for example, we use a Rapidopora pinnata Scott leaf. Rapidopora pinnata Scott is one of the Indonesian plants, as known as a medicinal plant. Empirical Rapidopora pinnata extract is used to treat cause rheumatoid arthritis, antibacterial, and anti-cancer. Rapidopora pinnata plant contain active substance from alkaloid compounds, flavonoids, saponins, tannin, and 3-terpenoids or steroid. According to the Masperia et al. 2030, their research found that the chlorum fraction show positive for containing alkaloid, saponin, and 3-terpenoid, and ethyl acetate fraction show positive for and N-hexane fraction was positive for containing 3-terpenoid or steroid, while the water fraction show the presence of tannins and saponins. The previous study showed that ethanol extract of Rapidopora pinnata leaf had the potential to be an anti-cancer and anti-inflammatory. It's well known that the common property of many tree products have the anti-inflammatory effect. This is the picture Rapidopora pinnata leaves. In this picture, this is the structure, molecular structure for anti-inflammatory activity. Have anti-inflammatory activity, but we have not found any article on which there is research on chemical compound from terpenoid class containing in Rapidopora pinnata leaf. Yeah, perhaps this could be the next topic of research to research what compounds of terpenoid from this leaf of Rapidopora pinnata. But according to the Tarigan et al. 2021, mechanism action or anti-inflammatory activity of terpenoid compound in Rapidopora pinnata leaf was indicated by inhibiting phospholipase A2 enzyme, an enzyme involved in the synthesis of arachidonic acid in the formation of inflammatory mediator. So arachidonic is not formed, which means that neither prostaglandin or non-leukotrienes are formed. This is the figure inflammation pathway. So the terpenoid compounds mechanisms was indicated inhibiting phospholipase A2. This is the stage of research. Sampling is carried out purposefully without comparing with the same plant from other region. So we collection the sample taken from Jalan Gunung Arca, number 10, Medan, Sumatera Utara, and we also determination of plant was carried out by the Indonesian Institute of Science, LIPI, Biological Research Center, Bogor. And then processing Sympesia into an extract of Rapidopora pinata leaf by percolation method for 3 days using 19 6% ethanol. Fractionation of extract with N-hexane to produce N-hexane fraction using split funnel, and then with determination of topical doses of the N-hexane fraction to be further made into nanogel and gel preparation with inflammation associated edema methods, according to Widiarini et al. 21. And then we preparation of nanogel and gel using carbopor as a gelling agent and stability solution, twin 80, PVPK 30, and mixture of twin 18 and PVKK 13 by using high steering magnetic stirrer. And finally, we evaluation of physical stability and anti-impurity activity of nanogel and gel preparation. And this is the our study. So in first pinata leaf we cleans and dry in oven to produce symbresia, and then we done using percolation method with 19.6% ethanol as a solvent at room temperature to find extract, and the extract was concentrated in rotary evaporator to find the thick extract and kept in the desiccator. Extract was fractionation using a mixture of water and ethanol and N-hexane and concentrate and to find N-hexane fraction. And then the extracts we prepare the cream preparation to determine the doses for topical. So the enhanced fraction rapidopol pinata squid leaf was made into the cream preparation, and then anti-inflammatory activity was determined and compared with market hydrocortisone cream. Based on this test, it will be known the doses of the N-hexane fraction compared to the anti-inflammatory effectiveness of market cream. The most effective dose will next be formulated into a nanogel preparation. Anti-inflammatory activity was determined according to the swelling of the back skin or edema of the mice after carrageenan induction by measuring the thickness of back skin every hour for 6 hours using a calibrate caliper, refer to the inflammation-associated edema methods. The mice were saved their back hair and left for 24 hours to avoid the presence of inflammation caused by epilepsy and hair loss. And then the percent inflammation and inhibitory inflammation was calculated. This is the procedure. So we use the experiment animal mice, 2 until 3 months with a weight of 20 up to 30 grams. The test animal were divided into 5 groups of 25 mice, received subcutaneous injection gamma carrageenan 3%. So the group 1 is a negative control without drugs, and group 2 is group hydrocortisone, given hydrocortisone cream as a positive control, and group 3, 4, and 5 were treatment group that we given cream with concentration of 2.5%, 5%, and 10%. cream preparation topical applied to the dorsal skin of mice that has been induced by 3% carrageenan. And then the thickness of the pulse was measured before and until 6 hours after the injection of the carrageenan with digital caliber. The idiom was expressed as the presentation difference between the thickness of the pulse after and before of the injection. Also, the values of the area under of the curve after 6 hours were calculated for each group. This is the result of determination of topical of the enhancement fraction of Radipora pinnata Scott ring. Based on the statistical analysis, we can see that it was concluded that the control group was not significantly different from the group treatment. 2.5% cream and 5% and 10%. So the anti-inflammatory effect produced with the NXM fraction of 2.5% still show no significant difference with the negative control group. The same anti-inflammatory effects or as hydrocortisone cream is the fraction of Rapidopora pinnatilif 5% and 10%. So this is the data from these two tables. These two tables show that the most effective dose of Rapidopora pinnata scoot leaf and NXM fraction was 5%. It was the dose selected to be further made into nanogel and gel preparation. The preparation of Rapidopora leaf and hexan nanofraction nanogel based on the modified method of Venkataraman and Nagarsenkar. We start by making a nanosuspension first, then mix with carbopol base. Nanosuspension are made to obtain nanoparticles from drug compounds that are homogenized with a stabilizer solution. A stabilizer solution includes surfactant or polymer or mixture of the both. The stabilizing solution serves to aid the wetting process of active sustained particle, reduce the occurrence of agglomeration and aggregation between nanoparticles, and prevent the occurrence of oswald ripening in nanosuspension preparation. So this is the formula or composition of the nano suspension and to make to the nanogel. Yeah? We use carbopol 1%, 1% of carbopol as a gelling agent, methylparaben as a preservative, propylene glycol as a humectant, and 3-ethanol to neutralize the carbopol to find the pH close to the skin, pH of the skin. This is the procedure preparation nanogel containing fraction of enhanced Rapidopora pinnata leaf. First we prepare the nano suspension. Yeah. So Rapidopora pinnata leaf and NXM fraction we homogenize using magnetosterol 2400 RPM for 11 hours to find a nano suspension, and The gel base we prepare using the carbopol, dissolved in aqueous water, distilled water, and methyl paraben, and we add the 3-ethanolamine, and then the gel base to mix the nano-suspension, the obtained or nano-suspension, and then we homogenize with magnetic stereo until to find a nanogel, a transparent nanogel. And then the nanogel was allowed to stand 24 hours at room temperature. The nanogel we evaluation for organoleptic physical stability after storage for 12 weeks at room temperature, low temperature, and high temperature. And then the measurement of particle, we use freeze laser particle site analysis, and the pH and viscosity were measure. Viscosity we measure using viscometer and DG 11S. And then anti-inflammatory we evaluate in mice compared to conventional gel with the same dosage. This is the result of physical stability of nanogel. The formulation show a clear nanogel, transparency, yellowish green color, smooth and homogeneous. The particle size analysis revealed that all nanogel formulations show the particle size in nanometer, and the size is still below 300 nanometer after 12 weeks of storage, except for nanogel F3. And there was no significance in color, odor, and appearance. Based on the previous reports, particle below 300 nanometer are more ideal for skin uptake, according to Allen 2011. This is the result of the pH value and viscosity measurement. The pH value were within in the range of the pH skin normal from 4.5 to 6, which are considered acceptable and less irritable for use in human skin after storage for 12 months. 12 weeks, sorry. And then this is the result of the anti-inflammatory activity. We can see that the nanogel 5% enhancement fraction of rapidopropionate has the percent inhibition of inflammatory more than the gel 5% enhancement fraction of rapidopionate. So the nanogel has more effective in inhibition of inflammatory compared to the gel preparation. This is because the permeability of the drug through the skin is influenced by the particle size. The release improves as the size of the particle decreases and the provide better penetration of active ingredient. So its anti-inflammatory effect will be better compared to the larger particle size of the gel. The previous study that used ethanol extract, the percent of inhibition of inflammatory only 48.91%. Terrigan et al. 2021. So they use ethanol extract, but our study used enhancement fraction. So we can say that the N-hexane fraction is more effective than the extract ethanol as an inflammatory drug. And the third topic I want to talk about anti-aging nanogels. The skin is the most visible indicator in the aging process. Source of exogenous free radical come from UVB. The free radical is defined as a species that can exist independently without one or more unpaired electrons. In living systems, free radicals are predominantly represented as the reactive oxygen species and taking form as oxygen center oxidizing agent. UVB causes photoaging. UVB increase the production of reactive oxygen species, increase breakdown collagen, and decrease collagen synthesis cause photoaging, according to Tanya et al. 2020. UV-induced loss have been shown to decrease collagen production and enhance elastin production, and degrade the structural integrity of the skin by way of altering the collagen and elastin component of the extracellular matrix, result in the formation of wrinkles on the skin. The fibrous network of collagen and elastin are responsible for the wrinkles and elasticity. The most common oxygen-based reactive oxygen species are superoxyanion, peroxide, hydroxide radical, hydroxide ion, and singlet oxygen, and excited state of molecular oxygen. ROS are unstable. In biological system, ROS add electron oxidize to other nearby molecule to release the extra energy and return to stable state. When not quenched by antioxidant, the oxidant reaction can continue with diminishing constituents to cause the aging of the skin. Topical antioxidants have the potential to neutralize free radicals. Antioxidants work by donating one of their electrons to oxidant compound or ROS species so that the activity of those oxidant compound can be inhibited. Antioxidants stabilize free radical by reducing the electron deficiency that free radical have and inhibit the chain reaction of free radical formation. This is the picture, yeah. We can see that chronic exposure to UV radiation often result in wrinkles, skin dryness, loss of elasticity, rose texture appearance, and pigmentation, and give histological feature of change from the epidermal thickness and degradation of collagen elastic fibers. This is the histological picture. So A is the area of skin that are not exposed to sunlight, but the B is exposed in sunlight to sunlight, and we can see that the area of the skin exposed to sunlight show the irregularity of elastin fibers. Is the same in this picture, and so this is the effect of the exposure of the sunlight or UVB radiation. And the use of cosmetic contain natural actives are increasing preferred due to safety and effective effect compared with synthetic compound. So this study, our study, we use a Capaficus alferegidot, also we know a red algae ethanol extract as anti-aging. Capaficus alferegidot is a species of red algae. It's a Rhodophyta family Solieriaceae. Capaficus alferegidot contain high level of polysaccharide carrageenan, a male component, and other phytochemical constituents, including phenolic compounds, flavonoid, alkaloid, terpenoid, phytosterol, and lectin have been found to possess various pharmacological activities such as antioxidant, anti-inflammatory, and anti-cancer properties. Previous literature reported the potential antioxidant compounds such as some pigments, glucoxanthin, astaxanthin, scarotone, and polyphenol, like phenolic acid, flavonoid, or tannin, that are widely distributed in seaweeds and are known to exhibit higher antioxidative activity. According to Payet, Capivicus alvarezii contains ascorbic acid and polyphenol, which contribute to the antioxidant activity. Previous research has shown that Capivicus alvarezii have antioxidant properties with EC 50, about 90.30, and total phenolic about 11.9, and as expressed as a milligram gallic acid equivalent per 100 gram, and total flavonoid is 50.26 milligram expressed as quercetin equivalent per 100 gram extract. This is our research flow. So the first we done a plan identification, preparation phytochemical screening of ethanol extract calva rigidoti, determination of total phenolic equivalent content of extract, and preparation of physical stability evaluation of non-containing Capivicus alvarezii, ethanol extract, and evaluation of skin irritation, and evaluation of skin anti-aging activity of non-gel compared with gel preparation. The plan identification and preparation of ethanol, we do a collection and processing of sample. So Capivicus alvarezii was collected from a cultivation site, Banggai Island Regency, located at South Sulawesi, Indonesia, during September 2021. And then we plan indication conducted at laboratory of plant systematic faculty of biology, Gajah Mada University. The sample was truly washed with water to remove epipet and dirty particles, followed by set drying for 2 days, and it was then brought to the laboratory, oven dry at 70 degrees Celsius for 4 hours to obtain a constant weight and pulverized in the grinder. And then we extraction of the sample. For the sample, put inside the chocolate apparatus. So we extract by chocolate apparatus and extract with 96% ethanol by the hot chocolate extraction methods. The temperature about 70 degrees Celsius for 5 hours until the droplets are colorless anymore, and after complete extraction, the solvent was evaporated in rotary vacuum evaporator with a temperature about 45 to 50 degrees Celsius. This is the schematic preparation of the extract. And then the procedure preparation of red algae ethanol according to Raisami 2021. In the procedure determination of total flavonoid content, the total flavonoid content was measured with the aluminum chloride colorimetry assay, and orange yellow color was developed the absorbance was measured at maximum absorbance wavelength by using UV visible spectrophotometer. Quercetin was used as a standard, and the amount of total valproate of extract were expressed as milligram of quercetin equivalent per 1 gram of extract. And then the determination of total phenolic contents was estimated by Pauling-Chiochaltios methods. Pauling-Chiochaltios method reagent oxidate the phenol in plain extract and change into the dark blue color is then measured by UV visible spectrophotometry. A Pauling-Chiochaltios solution was added to ethanol extract solution, and then after 5 minutes, we add the sodium carbonate, and then intense blue color was developed after inclusion for 60 minutes, and absorbance was measured by using visible spectrophotometry. Gallic acid was used as standard, and the amount of total phenolic contents of extract were expressed as milligram of gallic acid equivalent weight per 1 gram of extract. This is according the Ahmad et al. 2050. And then this is the composition of the nanogel. The phytospora, the red algae, we variation the concentration 1, 2, and 3%. We using a carbopor as a gelling agent, and twin 80 as a surfactant, and propylene glycol as a humectant. Ethanol is a solubilizer, and 3-ethanol is a alkalizing agent to find the.
Sorry, Prof. Anna.
Yeah.
The time is 2 more minutes.
Oh yes, yeah. So this is the preparation of nanoemulsion. Yeah. So we use the magnitude 0. Yeah. And for 11 hours. Yeah. So to find the nanogel. Yeah. And then this is the preparation of gel. So we prepare the gel as a compared to the nanogel. Yeah. The formula of the gel is the same of the nanogel, but we not use the twin. And then a physical subject evaluation nanogel, organoleptic evaluation, and primary skin irritant, we use 5 rabbits. This is the result of primary skin irritation test using the 4 rabbits. So, and then we evaluation nanogel protect UVB in this photoaging using rats, yeah, with the rats. The rats we divided into 11 groups. This is the group, yeah, normal control, not given test preparation and without irradiation, and negative control not given test preparation and irradiated with UV radiation, and group 3 until 4 is given nanogel, and group 6 and until 8 given gel. We use the UVB lamp. This is the UVB lamp. Yeah. And the irradiation was 3 centimeter from the red skin and irritated for 100-second precision. And the nanogel and gel preparation applied daily once a day immediately after irradiation process on the skin for 10 weeks. And anti-age activity was determined microscopically by visually and histology examination. So this is the result of total phenolic and flavonoid contents, yeah. And this is the mechanism of radical splitting of quercetin, and this is the, for example, yeah, how the antioxidant as a radical scavenging. And this is the result of nanogel formulation and physical stability results. The result of the physical stability show that the nanogel and gel, stable stored for 12 weeks at room temperature and high temperature and a low temperature. This is the average particle size of the nanogel. So the particle size is increased, but significant increase. The longer the storage, the greater of concentration of the extract, the larger particle. However, estimate the ideal particle size for skin uptake below 300 nanometer. The average nanoparticle size can range from 100 nanometer up to about 500 nanometer for cosmetic purposes, so to meet the requirements of the particle size for cosmetic. Yeah? And this is the physical stability about the pH value and viscosity. Yeah? So the pH value will be within the range of pH seeking normal, which are considered acceptable and less eatable for use in human skin after storage for 12 weeks. And this is the result of primary irritation test. Yeah. So all the nanogel and gel didn't show the irritate the skin after 7 to 72 hours. And this is the result, the morphology appearance of red skin in all groups of after 10 weeks. After 10 weeks of UVB exposure in the effective test of anti-aging. And then you can see that the group negative control change in the skin. Yeah, you can see that the skin is rose appearance. Yeah, compared to the normal control and the group treatment. So we can see that the treatment of the nanogel and the gel preparation can protective the UVB radiation after 10 weeks. After 10 weeks. And this is the histological picture. Yeah, yeah. UVB damage the skin layer, create change within the skin layer known as the dermal elastosis. Is an accumulation of abnormal elastin. This present as fragmentation of collagen elastin fiber. And we can see there's the group negative control. Yeah. Elastin include decrease as well hyaluronic acid decrease in the tiny epidermis. So this is the change in the epidermis, the thickness of the epidermis after irradiation of the UVB. This table show that the epidermis thickness of the skin after irradiation of UVB. So the nanogel, the application of nanogel and gel, the thickness of epidermis was increased compared to negative control. So both has been shown by a reducer in negative control, the thickness of epidermis thickness. Yeah. So the conclusion is the total phenol content in ethanol extra algae was 4 femtogram gallic acid equivalent gram of the sample, and total content of flavonoid in the ethanol extra algae was 11 milligram as a quercetin equivalent per gram of the extract. The resultant nanogel containing algae or Capicus alvarezii is physical stable over 12 weeks of the storage at temperature variation, room and high-low temperature, and does not irritate the skin ravage. Nanogel containing red algae preparation have better anti-aging activity in the skin of rat induced by UV rays compared to gel preparation through by visual microscopic and histology microscopic examination. Thank you for the attention.
Thank you very much for Nayanti for the interesting preparation. Now we can open for the question and answer session. If there are any from the participant would like to ask directly, you can also raise your hand, or you may type your question in the chat box. Maybe before any participant want to ask, I would like to ask one question, Prof. Anna.
Yeah, please.
Okay, based on your experience, what are the challenges that you have to face to formulate the plant extract in the nanogel research? What are the challenges or any limitations from the plant extract that we want to formulate to be a nanogel?
New challenges, you mean?
Yeah, the challenges.
Maybe we can nano cream or the other preparation. We can prepare for anti-aging and the extra.
I mean, is there any obstacle or limitations in the nanogel research that use a plant-based extract?
The plant extracts have the different location, so the content maybe is different. Yeah, so we must know the location, or we how to say to the source of the plant. So we must prepare a much of plant. Yeah, the sample is the source of the yeah.
So the plant is will affect the result, okay, from the nanogel research form.
Yeah.
Okay. Is there any participants want to ask? Okay, maybe the time is already up. Okay, thank you very much, Prof. Anayanti, for your presentation and for your sharing.
Thank you for all. Yes.
Okay, then I will return the session to Mr. Bayu. Thank you very much, Ms.
Okay, thank you, Prof. Anayanti, for the interesting talk. And thank you for Mrs. Laila for moderating this session. Okay, ladies and gentlemen, that's the last speaker for this morning session. The seminar will be continued after the lunch break, and we will start the seminar again at 1 p.m. So please kindly be on time so you don't lose for the next session. Thank you very much.
Thank you.
Faculty of Pharmacy, Universitas Sumatera Utara, provide high-quality education in clinical practice and pharmaceutical sciences, developing innovative, competitive, and sustainable research, escalating network among institutions in the field of education, research, and community services. The pharmacy program in Universitas Sumatera Utara, established in 1969 under the auspices of Faculty of Mathematics and Natural Science. In 2006, the Faculty of Pharmacy, Universitas Sumatera Utara, officially dissociated from the Faculty of Mathematics and Natural Sciences. The first dean of the Faculty of Pharmacy, Universitas Sumatera Utara, was Professor Dr. Sumadio Hadisaputra Apoteker. Students can graduate from various programs in Faculty of Pharmacy, Universitas Sumatera Utara. All departments are providing and improving lecturers' competency in the field of research and education, supporting innovative, competitive, and sustainable learning in order to reach excellent quality education in clinical practice and pharmaceutical sciences. More than 50% of faculty members are holding the title of professor or the doctoral degree, and many have been honored nationally and internationally. Learning and teaching in all levels are supported by excellent curriculum and facilities. Faculty of Pharmacy Universitas Sumatera Utara curriculum is based on the belief that successful students are built through a continuous educational process that is not only in academic role but non-academic activities to explore the potential of students' soft skills in dealing with their future career. The Faculty of Pharmacy Universitas Sumatera Utara embraces diversity in teaching approach, including active learning, traditional teaching strategies, up-to-date laboratories, robust use of technology, and an emphasis on learning as a lifelong activity to develop philosophy of the 9 Stars Pharmacy. The curriculum is meant to be constantly evolving to maintain a progressive and reiterative connection to interprofessional patient care in pharmacy practice and pharmaceutical sciences. Proper learning and teaching process provides an intellectual framework on which to build experiential knowledge. Faculty of Pharmacy, Universitas Sumatera Utara students have various of achievements both nationally and internationally. Faculty of Pharmacy, Universitas Sumatera Utara have a wide range of high-quality research equipment and facilities available to support staff and students in their research programs and projects. Our research laboratory are modern and supported by sophisticated lab equipment such as gas chromatography mass spectrometry or GCMS, high-performance liquid chromatography or HPLC, liquid chromatograph mass spectrometer or LCMS, ITTOF, thin layer chromatography or TLC densitometry, atomic absorption spectroscopy or AAS, ultraviolet visible spectroscopy, nuclear magnetic resonance or NMR 300 MHz, and other various laboratory equipments. Besides having good learning facilities, Faculty of Pharmacy Universitas Sumatera Utara is surrounded by a green campus environment where students can interact socially well and develop their soft skill activities. The distribution of alumni who have graduated from the Faculty of Pharmacy, Universitas Sumatera Utara, has occupied high positions in the fields of government, hospitals, and the pharmaceutical industry.
Saya Agus Tama, Kepala Dinas Kesehatan Provinsi Sumatera Utara, sekaligus sebagai Ketua Ikatan Farmasi Fakultas Farmasi USU. Dengan ini merasa bangga terhadap Fakultas Farmasi yang telah terakreditasi A, dan harapan kami ke depan tetap dipertahankan. Semoga para alumni bisa berkiprah dimanapun berada, termasuk di kancah internasional. Terima kasih.
Saya Gita Baringin Nasution, apoteker dari Balai Besar Pengawas Obat dan Makanan di Medan. Fakultas Farmasi USU telah berkembang dengan pesat, di mana telah memiliki laboratorium dengan peralatan yang canggih, sehingga nantinya alumninya dapat diandalkan dan bersaing di bidang pengawasan, produksi, distribusi, obat, dan makanan.
Saya Rima Elfitra Rambe, Sarjana Farmasi Apoteker. Sekarang saya bekerja sebagai salah satu apoteker di Rumah Sakit Universitas Sumatera Utara. Saya merupakan alumni dari Fakultas Farmasi Sumatera Utara. Saya bangga menjadi salah satu alumni karena saya ditempa untuk mampu berkarya di dunia kerja, khususnya di rumah sakit. Semoga Fakultas Farmasi Universitas Sumatera Utara semakin jaya.
Saya Dr. Andes Budiono APT. Saya bekerja di PT Motiva sebagai manajer produksi dan sebagai penanggung jawab teknis. Saya sangat bangga sebagai salah satu lulusan dari Fakultas Farmasi USU, dan harapan kedepannya saya berharap Fakultas Farmasi akan lebih maju dan menghasilkan tenaga-tenaga atau SDM yang dapat bersaing di dalam industri dunia farmasi.
The Faculty of Pharmacy, Universitas Sumatera Utara. We act with integrity, honesty, and accountability. We involve community services and promote civility, diversity, and mutual respect. We engage networking locally and globally among institutions in the field of education, research, and community services. We seek and develop resources, infrastructure, and knowledge to promote pharmaceutical discovery. We can strive for excellence in all we do.
Escalating knowledge through excellent education. We are excellent.
Introducing the English test designed
Dr. Jitima, Dr. Ami,
Hi.
hello.
Hi. Nice to meet you.
Yeah, nice to meet you too. Yeah. So the time in Medan is the same with Bangkok,
Oh, yeah.
right? Yeah,
Yes.
exactly the same.
Yes.
Yeah. Yes. Yeah, from here from Medan to Bangkok, it's near. Maybe is it the same with to Jakarta? Only 2 hours.
Oh, really?
Yeah, and the flight is cheaper to Bangkok than to Jakarta.
Oh, okay. Really? So that's great.
Yeah, I went to Bangkok maybe 2 years ago, 3 years ago. Yeah, to Bangkok.
Okay.
So it's not far.
You and your team should come again soon.
Yeah, yeah. I hope. Yeah.
Right. Should I start or
Just a moment. Maybe
okay,
the
sure.
will say I will arrange time.
Thank you.
And the moderator is that moderator.
Hello, Professor. Hello, Professor Ami.
Hi.
From
Hello, Ronnie here. Nice to meet you.
Yeah,
Nice to meet you too.
Ronnie is in Korea now, so the moderator from Korea, speaker from Bangkok.
Right, Professor, can we start, Professor?
Yeah, it's okay. Yeah.
Okay. Bismillahirrahmanirrahim. All right, just to let you know, we will continue our agenda for today. So please go back to your screen and make sure you turn off your microphone. Okay, thank you. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. Welcome back to the second international seminar on nanomedicine. And let me guide us this afternoon. I'm Fauzan, your master of ceremony. All right, I hope the time is enough for you to recharge your energy. Now, I encourage you to turn on your camera. I want to inform you that our invited speaker for this session is Dr. Jitima Ami Lukanagul. All right, in this session, also already with us, the moderator, Dr. Roni Abdi Sahputra, and he is a lecturer in the Faculty of Pharmacy, Universitas Sumatera Utara, who has published several publications in index international journals, most of them about cardiology and pharmacology research. He will bring this session with Dr. Jitima. So let us welcome Dr. Roni. Time is yours.
Okay, thank you very much, Mr. Fauzan. Okay, in this afternoon, we are so glad having you, Associated Professor of Pharmaceutics, Jitima Ami Lukanagul from Chulalongkorn University. We welcome you in Faculty of Pharmacy, Universitas Sumatera Utara. So this session will be very informative, will be very useful, and also Dr. Jitima Ami is from Chulalongkorn University, graduated Bachelor of Pharmacy in Chulalongkorn University, and continuing his PhD in Chemistry and Biochemistry in University of South Carolina, USA. And currently, his position as an associate professor in Chulalongkorn University, and also has several award and honor as a Best Invention Trophy issued by World Invention Intellectual Property Association. I believe that Professor Jitima also has several projects, a research project, collaborated with international and national researcher and we hopefully in the future we can collaborate between Faculty of Pharmacy and Chulalongkorn University under supervised Professor Jitima. We have a 45 minutes, Dr. Jitima, for presentation, and let's begin. Time is yours.
Okay, thank you so much, Ronnie, for the nice introduction. So I'm Amy. You can call me Amy if that's easier for you to pronounce. And yeah, I'm now based in Bangkok, Thailand, at Chulalongkorn University, the Faculty of Pharmaceutical Sciences. So shall I share my screen now? Yeah. Okay.
Yeah, sure.
You see my screen, right? Okay. Okay. This is full screen. Yes.
Yes.
Okay. I see there are a lot of audience today, a lot of participants. So if any of you have questions, you can stop me at any time by raising your hand through Zoom system, or leave the question in the chat in the meantime that while I'm talking, so that you don't forget where you have questions along with my presentation. So I would be happy to answer all the questions at the end of the talk, or during the talk is fine. So my title today is Designing Hyaluronic Acid-Based Material for Implants and Molecular Delivery. So I basically have a background in biomaterials. In my PhD, I did the thesis about nanomaterials that induce tissue regeneration for bone and cartilage. And also my background in a bachelor degree was from here, Faculty of Pharmaceutical Science, Chulalongkorn University. So I merged the expertise from PhD with the background in pharmacy to continue the project on drug delivery. So now I'm focusing on hyaluronic acid-based materials to make new biomaterials to be used in medical application towards the ATMP's product or advanced therapeutic medical products like gene deliveries, RNA delivery, mRNA vaccines, or CAR T cells, things like that. That's the part for molecular delivery. And also at the same time, I have another group of students that are working still on the regenerative medicines, but focusing on the development of the new scaffolds to build the organ supplements. So I would like to start with the tissue engineering part first. So in our lab, we utilize hyaluronic acid, or I would like to call it HA from now throughout my presentation, because it's easier to pronounce. My tongue will not be twisting so much, because hyaluronic acid is a very difficult term. Even I worked with it for a long time already. Well, I will be working. I will be talking about HA-based nanogel, nanogel and hydrogel carriers as platform for tissue engineering, hydrogel scaffolds, and also for drug delivery. So let me give you some general idea why I have been interested in using HA. So HA is a kind of natural linear polysaccharide, which is the major content of articular cartilage and a lot of many parts of the body, and as well as the deeper skin tissue. And it's a component of extracellular matrices in many places, in many organs in human. And also the monomeric structure of HA is composed of glucuronic acid and N-acetylglucosamine that link together with glycosidic bonds that can be cleaved with hyaluronidase enzyme, or we call HAS enzyme, which is we have it in our body. So this makes this backbone degradable in human body. HA also has been studied extensively in biomedical application because of its abundant in nature, and it's great compatibility, biodegradable, non-immunogenic, and low toxicity. HA is a great monomeric backbone that contains many functional groups that are available for modification. That this can emphasize the potential use or versatility of HA for many biomedical applications. If you can see here, there are many researchers working on modification of the carboxylic acid of monomer of HA or hydroxyl group, or those can be conjugated with many type of molecules and cross-linker to be click or cross-link with other molecules, including macromolecules like proteins or antigens, antibodies, things like that as well, not just small molecules. So this, there are a lot of studies that chemists modify HA for particular application. So let's talk about tissue engineering first. Tissue engineering is, as you may know, is part of regenerative medicines, where it applies the principles of engineering and life science towards the development of biological substitutes that can restore, maintain, or improve function of an organ. So the ultimate concept is to extract some stem cells out of a human body and expand them and grow partial function and tissue outside the body in three-dimensional premature scaffolding materials, and implant these new parts into a patient's body to replace or to fix the part of the body that has been defect. The scaffolding part needs the extensive design and selection of biocompatible materials that could function as expected, for the clinical outcome. So research publication on tissue engineering has begun since late 1900s, and the interest has been growing very fast. And until earlier last year, New York Times have this new case of three-dimensional transplant of human ears that made from patients' own cells and the 3D printing scaffolds. This is the very first successful clinical case that the complete tissue engineering concept has been adopted in clinical use in human. And before, we have a lot of medical device, but did not include the cells in the system to replace the organ defect. So this been a success in the first case in stem cells cells encapsulated scaffolds to be implanted in human. So in our lab, we are interested in designing the injectable hydrogel as the scaffold, since it could fill up a regular shape defect, such as bone cranial defect like this one. We have actually, before I developed this injectable scaffolds, I used to work with implantable scaffold, which there are some different. The major difference is that the implantable scaffolds is the preform scaffolds or preform materials that are in three-dimensional format, that after you grow the cells, then you outside the body, then you implant it back in. But the problem with this pathway, with this approach is that, so this is the picture of the rat skull bone, the cranial defect, and we create the bone defect by drilling the skull out and then replace it with this three-dimensional preformed scaffold that implant in the hole. And after 10 weeks, we found that the hydrogel that we implant in has some calcification, which means the bone can regenerate within the scaffold that we implanted. However, if you can see here, this is the cross-section scan. You can see here that there are gaps between the implanted tissue and implanted scaffolds that pre-made, pre-formed outside the body, and the native bone around the defect is not merged. It's not completely merged together. So that's why we thought about having, injectable platform that it can act as a glue that fill also the filler and also the scaffolds that solid enough to house or to make the cell grow inside the scaffolds. That's why we designed this injectable platform for the next step. And so we found that hydrophilic polymers can be made for in situ gelation, and we picked HA because HA, like I already told you, why that we are interested in using HA as the backbone polymer. However, HA is not a gel forming material because it's, like I said, it's the linear polymer. So it could only increase the viscosity of water, but it could not make a hydrogel by itself. So we need to modify the structure of HA in order to have the cross-linked between, like, inside the microstructure of the polymer to make it solid enough for the cell to grow and to make the tissue replacement. So we design the material by using HA backbone and then cross-linked it with functionalize it with methacrylated functional group. And once we have methacrylated HA, we could induce the gel formation or cross-link of the gel by adding DTT cross-linker, which has two thiol group on the different ends that can, act as the bridge to cross-link between the vinyl group of methacrylated HA. And then we found that after we have that methacrylated HA cross-link with DTT, we could successfully make gel. And it's injectable gel because once we integrate the DTT crosslinker into the solution of methacrylated HA, the gel was formed within like 1 minute after mixing. However, the resulting gel was not very strong. It's still very soft gel. So we try to enhance the mechanical property of the gel and the microstructure by adding the nanomaterials. So the nanomaterials we use as the to strengthen the hydrogel mechanical property is the virus-like particle, which is the rod-shaped nano-sized particle called TMV. And in order to incorporate TMV as it's like a very small chopstick in the nanometer range, the size of each rod shape TMV is the length is around 300 nanometers, and the diameter of this tube is 18 nanometers. So it's very, very small, but it could strengthen the gel as a result. However, we still had problem because if we use TMV itself, it cannot really cross-link to the scaffold, which is the methacrylated HA. So we had to modify the functional group of also the virus-like particle of TMV to have it to have the surface that can be cross-linked with the methacrylated HA. So we made TMV cis version of TMV. So we can conjugate cysteine on the material, the virus material, or virus-like material that we call TMV. So we come up with TMV cis, and this cysteine can be clicked, together with the backbone of methacrylated HA. So that's why we have, this virus-like materials, nanoparticles, to strengthen the scaffolds better. And then, so we tested on the dilation time and mechanical property of the resulting gel. The figures show the mechanical property of the resulting gel while it undergoes dilation. And after dilation, the mechanical property was steady. So if you look at the graph here, there are two types of hydrogel. The first type with representing by the solid lines, the blue color and the green color. The blue color is the storage modulus, which is the well, it tells the mechanical property of the solid property of materials. But the green line is telling the loss modulus, which represent the liquid part of the hydrogel. Which usually, if you talk about the mechanical property or the strength of the gel, you're looking at the storage modulus because gel is already solidified water. And so if you see the solid lines is the hydrogel with nanomaterials or nanoparticle virus like nanoparticle that we call TMV-SH, which is TMV cysteine, then it's a lot higher than the storage modulus of the gel without TMV. And you see the change in gelation time as well. So when you have injectable gels, once you mix polymer in water and you add the cross-linker to make gel, right? So there is the lag time, like that you allow the cross-link reaction to occur. So that time we call that time from mixing until they form gel, we call it gelation time. So if the gelation time is short, then it makes the material maybe easier to be used in clinic. So we don't want the gelation time to be like 1 day or 2 days, right? So you want to mix the two components together and then ready to be injected into human body. So that's why we have to optimize the gelation time, and it cannot be too fast, because you have to be able to inject the gel mixture into the body before it cross-link or before it gelation. Otherwise, it gels in the syringe. You are not able to. The doctors cannot inject those gel into human body again. So that's why we have to optimize, and we have to know what's the gelation time. And the machine or the method that we can identify gelation time is this crossover between storage modulus and loss modulus. So you're looking at the crossover between the green line and the blue lines like this. But this is odd. When you don't have virus materials, it's very, very strange that, at sometimes the storage modulus becomes the same as loss modulus, meaning that you don't really get a stable gel, but for the solid lines, you have this crossover between the storage modulus and the loss modulus, meaning that with the virus incorporation, the gel forms better and the gelation times is a lot faster. Okay. And then once we get the materials, we culture the cells inside the hydrogel. The cells we use was mesenchymal stem cells that extracted from a rat, and we grow the cells in the encapsulation form, meaning that we mix the cells, stem cells, together with the hydrogel mixture before it cross-linked, and then we add cross-linker, and then we create three-dimensional gels with the cells, with the stem cell that was embedded inside the gel structure. And then we culture the cells in culture media, and then we observe the collagen production as a protein inside this three-dimensional structure of hydrogel with cell embedded inside. And we found that with the TMV or nanomaterial incorporated hydrogel, we had the higher amount or higher efficacy in collagen production inside this matrix. As you can see from confocal microscope here, that's stain for collagen type 2. And you can see that the gel, HA gel alone or methylated HA alone doesn't produce a lot of collagen. While if you incorporate the TMV as the virus-like nanomaterials, it even further enhance the production of collagen type 2 out of stem cells that we encapsulate in the gel. Okay, and this is confirmed by total collagen staining also, and we have the many other characterization. For example, we also quantify the amount of collagen relatively quantify comparing between the HA material alone and HA together with the nanomaterials. It was found that with nanomaterial, it could enhance the amount of collagen type 2 produced in the stem cell matrix even better at the different time points. Okay, and also we at the endpoint of each time point, like day 0, day 21, and day 28, we found that at day 28, we had the higher mechanical property because we thought that, with the process of cells growing and creating its own extracellular matrices that can stiffening the structure or the scaffold even further. So we got the modulus even higher in day 28. So next, we moved on to transferring these materials and study in the animal models. We tried the materials like this HA-based scaffold with this virus-like materials in two type of rat models. The first one is the general rats, or we call non-OVX model. And the second model was OVX osteoporotic model. That means it's representing the menopause women, because OVX is abbreviated from overactomized, meaning that the rat was undergone the removal of ovaries. So when it doesn't have ovary, it does not produce estrogen, so it has higher risk of osteoporosis. And we do the same protocol. We take the part of the skull out, and then we replace with our materials and see how the bone is created or how the bone is being healed. Okay. And then we compare both group of rats, and we saw that incorporation of TMV, which is the virus-like nanomaterial that I mentioned, it even further enhanced the efficacy of bone regeneration in both model, especially with the OVX or the menopause animal model. We could enhance very obvious that we could enhance the bone formation even better than other controls material. So that was the tissue engineering side of work, and they are still ongoing with many tested in human and also in vitro tests in terms of the molecular biology, looking into gene expressions or other type of markers for both chondrogenesis, which is the cartilage regeneration, and also the osteogenesis, which means the bone regenerations. So Next part of my lab work is involving the development of the new drug delivery system, or encapsulation system. So we focused on nanomaterials that can carry the drug molecules, or either it's a small molecule or macromolecules like proteins, or nucleic acids like genes or mRNA. So among the nanocarrier systems, the hydrophilic nanogel make a very promising platform as candidate for drug delivery. So before, people do a lot of study on liposomes, or emulsions, or two-phase system, but nanogels make a little difference in the drug delivery, because nanogels is a kind of polymer self-assembly with three-dimensional networks in the nanoscale size range. So it's different from my first part of the project that we talk about macrogel, where you can see that's like jelly-like or gummy-like structure that you can really touch and feel the hardness of the materials. But for nanogel, it's just like water. Like the viscosity is just like water, but the gel is happening to be in the nano size matrix form. So you don't really see it, you can't touch it, but if you look into the transmission electron microscope, you see this spherical particle that was caused by the cross-link network of hydrophilic polymer. So there are different types of nanogels based on the gel mechanism or the nanogel formation. Today, we will be focusing on the self-assembled platform, which can be manufactured in larger scale for the industrial technological transfer. Simply speaking, nanogel is a gel in colloidal system. Being mostly hydrophilic in nature, nanogels are highly biocompatible with a high loading capacity that can be optimized for the gas molecules or the cargo, and their unique physical properties offer them distinct advantages over other types of nanomaterials for biomedical application. For controlling the drug release, we can tailor-make the system with the stimuli responsive polymer. And moreover, nanogel can be administered IV or parenteral route. And of course, the mucosal administration is very much possible as well, because it contains a lot of water in the system. You don't have to deal with oil phase or surfactants or things that are very risky for, to regulate the osmotic pressure inside the blood or serums. I have some chat here. Should I answer or? Oh, okay, sorry. All right. So based on that introduction, we designed the polymer graph with HA backbone. So I stick to HA still, even though I moved to the colloidal drug delivery system. So the polymer graph with HA backbones, and we modified to be able to self-assemble into three-dimensional networks. Like I said, HA is linear, so it could not really do the encapsulation with the HA alone. So we have to modify the structure of backbone of the HA polymer. So these three-dimensional networks is in nanoscale that are swellable in the spherical particle, and it can be formed by simple self-assembling of the right proportion of hydrophilic and hydrophobic regions that we conjugate onto the backbone when it's dispersed in aqueous fluid. So it could self-assemble itself into spherical shape. So our patented nanogel, we already patented in the PCT, and it was already published. And it's water soluble and very easy to use. It's simple, one tank, one component, mixing it together with the active ingredients, and then it automatically encapsulates the active ingredient. with the simple incubation method. So many types of active compounds could be incorporated and released by diffusion. And you could encapsulate the hydrophobic molecules or hydrophilic molecules, small molecule, proteins, DNA, RNA. It's highly compatible with most of active ingredients that I tried. I tried more than 30 active ingredients to be incorporated into this nanogel. And it's very well compatible. Okay. So I would like to give one example of the molecules that we tried at the very first molecule that we tried to encapsulate with this nanogel, which is curcumin. You all probably very familiar with curcumin already. So curcumin is a compound from the rhizome of the herb called Curcuma longa or turmeric. And it has received a wide spreading attention. Sorry, my tongue is twisting. Over the past decades, the excitement originated after the encouraging in vitro discoveries showed its influence on many biological mechanisms associated with several diseases. In particular, in vitro and animal models confirmed that curcumin has an anti-inflammatory, antioxidant, anti-cancer, antiviral, and neurotrophic effects, and many other, therapeutic potentials. Investigations also indicated curcumin's potential to treat several cancers or GI diseases, metabolic diseases, and also include the dermatologic conditions like psoriasis, eczema, atopic dermatitis, things like that. And also the autoimmune conditions was studied with the curcumin treatment. But unfortunately, these encouraging initial findings have not been consistently supported through human clinical trials. So a lot of clinical trials was done on curcumin, but it was not very successful. So the major barrier of curcumin's clinical efficacy is its poor bioavailability. So we have efforts that a lot of efforts in research, therefore, been dedicated to developing curcumin formulations with greater or enhanced bioavailability and systemic tissue distribution. So that's why we are so much interested in improving curcumin efficacy in the physiological conditions. Oops. Okay, so now we combine this curcumin in our nanogel. So first we had to make or synthesize the nanogel and characterize them. So we found that with HA alone, it's got TGA or thermal analysis graph like this, meaning that we have one component in the system. However, after modification of HA with the another grafting molecule we call p-napalm, it creates some two peaks, which means we have the conjugation of two types of polymer. And then we characterize it for the particle size from dynamic light scattering together with the transmission electron microscope. And we found that we could produce this beautifully distributed nanogel in the size of 0.1 micron, which is 100 nanometers, or 200 nanometers or above, depending on how much you conjugate it or how much you graft another polymer onto HA. And the size range is satisfying. So after encapsulation with curcumin, you see this dark spot or dark particle that contain the drug molecules, and we can characterize the loading of curcumin to be higher than so it improved the loading capacity of curcumin in the formulation as well. And then we know that HA enhances the intracellular delivery via the mechanism of CD44 receptor, because HA has been known to bind with CD44 transmembranal protein receptor on the particular type of the cells. And one of which is the NIH3T3 cell lines, which is the fibroblast cell lines that it's been shown to internalize this curcumin encapsulated HA nanogel that we produced. And we found that curcumin in the cells was higher in the with the delivery of curcumin, or with the treatment of the cells with the curcumin inside grafted HA nanogel. And so these are confocal microscope proving the localization of curcumin inside the cells. This is fibroblast cells. The first panel stains nucleus that's stained blue by DAPI, and the third panel stain actin, which show the localization of the cells, and the second column is curcumin. So you see the curcumin in the cell regions. And then we tested the skin irritation test with our material. So there was a question whether or not this nanogel is safe to be used. So we tested it on the EP-skin, which is the three-dimensional model of the human skin full structure. So basically, the topmost layer is stratum corneum, which is the dead layer of the epithelial cells, or it's actually keratinocytes, dead cells. And then underneath are keratinocytes, that human keratinocyte that they, culture it on the what do you call, on the membrane, or the, the surface, membrane surface. And then they culture this multi-layer of the cells to have this full tissue or full skin structure. And then we treated this three-dimensional skin culture with different samples. The no treatment is just the culture media that we buy from the company that make this EP-skin full tissue construct. And then the second control was to add PBS, which is the media for our nanogel. And then this picture showing the samples, or the cross-section of this skin construct that was treated with the HA nanogel only. But the topmost picture representing the resulting cross-link histological analysis of the full tissue construct that was treated with curcumin encapsulated HA nanogel. So you can see that this is the worst, because there are irregular arrangement of the cells, meaning that PBS did something to this skin tissue. Well, it was expected, because PBS is not a very healthy condition for cell culture. So if we compare our treatments with the culture, that was treated in the media that we bought from EP-skin company, then there were no different, and even strengthened the the layer of the stratum corneum better when you have curcumin nanogel with HA. So as you can see here, stratum corneum, this start to fall off, but this a little bit falling off, but for other sample at other area, it will still be contact. Intact, sorry, on the topmost layer. So, but there's no significant difference between sample the first, the second, and the fourth one. But definitely the treatment with PBS was the worst. So we concluded that the nanogel was not toxic or create irritation of skin structure. And so next we were testing on anti-cancer and anti-psoriatic activities of curcumin after we encapsulated in our nanogel. So we first did the cytotoxicity assays with the treatment of curcumin-loaded nanogel on 4 cancer cell lines, including MDA-MB231, HT29, and HepG2. HepG2 is hepatitis model. The liver cancer model. And CACO2 is the colon cancer model with these 4 cell lines. So the graphs here represents the viability data resulting from the titration of different dilutions of each curcumin-loaded nanogel treated on each cell types. And the overall relationship between the cytotoxicity and the drug concentrations across all experimental cell lines was dose-dependent, with the IC50 of curcumin controlled treatment dissolved in DMSO for all 4 cell lines were averaged around 5.29, 10.2, or 13.41, and also the for CACO2 is 12.24. And the resulting IC50 of curcumin in nanogel towards the cell lines were slightly higher than that of curcumin in DMSO, except for HepG2 cells, yet still in a comparably effective range when tested on the same cell lines. However, with the curcumin HA nanogel formulation, the aqueous-based formulation could be beneficial considering the physiological compatibility of the product to be administered. Like you can never use DMSO in human body. So that's the rationale why we want to use our delivery system. But if you look into the bottom graph here is the test on the anti-psoriatic model of cell culture model. So I believe it's the HACAT or keratinocyte was treated by TNF alpha to induce psoriatic character of skin of the cells layer. And once we treated it with curcumin and curcumin in the nanogel, it reduces the cell viability, meaning that it reduces the psoriatic activity of the skin that created or induced by TNF alpha. So it could reduce to nearly to 100% cell viability, which is the normal range. So in psoriasis, once the cells was treated with TNF alpha, you will see over proliferation of the skin cells, which you don't want. That means the psoriasis is progressing. So you want to suppress the cell viability or the cell proliferation rate. And curcumin can do that. And even better when you have curcumin with nanogel encapsulation. So another type of molecules that we tried is the larger molecules, which we aim for immunotherapy and vaccines. And this is my review paper. You can scan the QR code to learn more about the concept of using HA in the mRNA and vaccine delivery. So we use a promising immunostimulant candidate in clinical studies for improving therapeutic vaccine efficacy. This is a synthetic analog of double-stranded RNA structure we call polyinosine polycytidilic acid, or poly-IC, that mimics viral RNA through TLR3 expressed on the endosomal membrane of the immune cells, stimulate many signals to secrete the inflammatory cytokines and increase the immunity to anti-cancer. So many antigen-based cancer vaccines are soluble biomolecules that have poor retention in tissues along with the suboptimal antigen processing and presentation by dendritic cells. The low retention in the circulation and the tissues imposes a significant limitation because multiple booster doses are often needed for the effective of the vaccine. And therefore, safer immunotherapy approaches are also needed because these could be very toxic substances towards the human use. And at the newer discovery or newer researchers emphasize on the importance of the design and development of cancer vaccine adjuvants as security while we have a good clinical efficiency with low toxicity as well. So this calls for the delivery system that can elevate the stability and delivery of poly-IC. And to overcome the limitation of poly-IC by utilizing HA nanogel platform for incorporation of poly-IC by self-assembly of the modified HA polymer to get the nanogel particle formulations that can also extend the half-life of the adjuvant or poly-IC in the tissue, and also to protect the molecular stability of poly-IC and reduce the toxicity of poly-IC as well. So we again first characterize the system with and without poly-IC. All nanogels presented in the negative charge with no significant differences, with the TEM images demonstrated that the nearly spherical particles in blank nanogel. The nanoparticles formed with poly-IC showed even more irregularly spherical shape with higher degree of roughness in the surface nanostructure. The particles were well distributed due to the slight negativity charge on the nanoparticle surfaces. However, for the hydrodynamic size measurement from dynamic light scattering or DLS using Zeta sizer machine, the sizes are somewhat different from TEM, which was in dry stage. So, the condition of test is different. So you could see a slightly different in terms of the size. And in each of 0.5 polymer formulation with poly-IC encapsulation had significant size difference with the increasing concentration of poly-IC loads. Okay, so you see the left picture of figures here will be not exactly the same as the result of the size measurement using Zeta sizer or light scattering methods. And so then, as our hypothesis was that to incorporate poly-IC in the nanogel could stabilize the structure of poly-IC better, then we perform the gel separation of the degraded product of poly-IC, and we challenge the samples with total serum that's abundant of enzyme that can kill poly-IC. So with the mixing of enzyme together with the poly-IC, you see the degraded product bands here with different time point of incubation. So at 24 hours, you see a lot of degraded product of poly-IC in the serum. However, when you encapsulated poly-IC with the nanogel, we call the trade name became high-end. It's the hyaluronic acid-based nanogel, the same material that I introduced to you before. You could see the disappearance of the degraded product bands of poly-IC after 24 hours. So it could stabilize the structure of this poly-IC. And then we had a question of whether or not the poly-IC that was encapsulated in our nanogel could get inside the cells or not. Then we conjugated poly-IC with the red fluorescent molecules. So now our poly-IC can be seen from confocal microscope in the red panels here. So we found that with the encapsulation of the nanogel with poly-IC that was tagged by red fluorescence here, we can see that the poly-IC can get internalized into the cells with the encapsulation of the HA-based nanogel. So this is HA-based nanogel in different concentration. So you see that the higher the concentration, the higher amount of, the red dots here representing our poly-IC molecules that gets into the cells more, than the poly-IC alone without HA nanogel. Okay. So next we move to the to prove the efficacy of this HA, or we call high-end HA nanogel for in the application for adjuvant for SARS-CoV-2 subunit vaccine. So this is another project in collaboration with another company that produced the SARS-CoV-2 subunit vaccine, but they had a problem that this protein cannot boost the immunogenicity of in animals as they expected. So they want to use the adjuvant in the formulation of COVID vaccines. Traditionally, people use alum as the adjuvant. So then we compare our nanogel adjuvant with the alum adjuvant in the market. And we found that it's the non-significant difference in terms of the IgG production in mice that was immunized with SARS-CoV-2 subunit vaccine that's mixed with our nanogel, HA nanogel, when comparing to the alum adjuvant. And the toxicity was fine. Like for at the same time, we observed the kidneys, spleen, liver for the toxicity that might occur with the new materials like our HA nanogel adjuvant, but they all look okay. And the weight of the animal was normal. And then we move to the HA-derived delivery system for enhancing cancer therapy. We just want to see if our nanogel can deliver some small molecular drugs into particular organs. And we found that it was selective for a particular organ as we expected once we injected into mice. So that's it. This material was commercialized after patented for cosmetics application first because of the barrier of registration. The Thai FDA allowed this material to be registered in cosmetic product at the moment, and then we are working up to making these materials in the pharmaceutical excipients and also the topical drug excipients and injectable excipients in the regulation pathway. Okay, that's it. Thank you for my lab, my people, my postdocs, my team, and also Napsalute is the spin-off company from our Faculty of Pharmaceutical Sciences that commercialized the product and also produced, manufacture this high-end or HA nanogels materials currently selling in the cosmetics, and also having the pipeline of the pharmaceutical products and also vaccines and the injectable version of HA nanogel. Thank you so much for your attention, and I would like to answer the questions you may have.
Okay, thank you very much, Dr. Ami. It was wonderful and impressive presentation. And we have one question in the chat box. Maybe you can, from Fauzan. Can you see it, Professor?
Sorry, I had one question in my chat box here. Okay. anti-cancer activity of your curcumin nanogels, did you test the other parameter like apoptosis induced and oh yeah, actually now it's ongoing. We doing the cell cycle arrest is ongoing. We had a first data out last month, and now we are discussing to do the confirmation next. So please be in touch, and I should publish the set of data soon. Yeah. And also, if you did, how is it affect on curcumin? Okay, yeah. It's not very conclusive right now, but I should have everything as a manuscript within the first half of this year. Thank you.
Okay, we have another question from Professor Anayanti.
Okay. I want to ask you how about the stability of H. Okay. This HA nanogel could withstand. So now we tested on the autoclavable conditions. So it's this HA nanogel is pretty tough. It could withstand the pH range from 2 to 8, and sorry, from 2 to 10. And for the temperature, it could go up to 100 degrees Celsius. And now we tested, and our preliminary data show that it could withstand autoclaving condition. So high pressure, high temperature. Yeah, and there is no aggregation along the way because it's very special because it's nanogel. So the surface of the nanoparticle is very flexible. So it could, swell and it could shrink, and also it's very, very stable. Yeah.
Okay, Professor. Last question from me, Professor. Have it ever tested in human? The HA nanogel.
For skin application, yes.
For a skin application.
Yes, in skin.
Mm-hmm.
But
Okay.
for injectable and oral route of administration, we are testing still in animal model.
For injectable nanogel, is it any side effect like blockage the blood vessel?
No, not right now that we see from animal though, but not in
From
human.
animal.
Mm.
Okay, so it's totally safe.
Mm-hmm.
Okay, great. Thank you, Professor. Impressive presentation.
Thank you so much.
Okay, that's all the presentation from Professor Ami, and we are glad that you want to share your latest research with us. It's very, very informative and also impressive, and full of knowledge. And for a lot of students here in lectures, we are so grateful and thank you again.
No,
Thank you, Professor Ami.
Yeah, thank you for the invitation, and hopefully I see most of you in Thailand soon. And Professor, you have to be here, and also Ronnie, and also the MC is Muhammad, right? Okay, you are all welcome to Chulalongkorn University. So
hey,
thank you, Dr. Ami. Dr. Ami, thank you very much.
Thank you.
Thank you. And after this, I give it back to MC. Thank you.
Okay, thank you, Dr. Roni, for moderating this session. Also, thank you for the amazing presentation, Dr. Ami. The session was so informative and useful for the future research. All right, all participants, now we move to the next agenda, which is the presentation session by our other invited speaker, Dr. Sumaya, SSE, MSE, Apotheker. And this session will be guided by a similar person to the Vivo session. So please welcome once again, Dr. Roni Abdisaputra, SFAM, MFAM.
Okay, thank you, Mr. Fauzan. Okay, we have a invited speaker in here, Dr. Sumaya. Okay, can you please? Okay, Dr. Sumaya is an associate professor in pharmaceutics in Faculty of Pharmacy, Universitas Sumatera Utara. Extensively research in nano, and also the latest publication is hydrothermal nitrogen doped carbon dots from isolated lignin of oil palm antifruit for bacterial imaging of Staphylococcus aureus. Okay, we will wait. You have a 15 minutes, doctor, for presentation and also discussion. Thank you. Time is yours.
Thank you, Mr. Roni, for this opportunity. Good afternoon, ladies and gentlemen. Let me present our research with the title Carbon Dots from Isolated Lignin of Oil Palm Empty Fruit Bunch for Bacterial Imaging of Escherichia coli and Staphylococcus aureus. Lignin is the world's second largest complex organic material and is a low-value waste product. Lignin is produced in the pulp and paper industry at 50 to 70 million tons every year throughout the world. One rare and underutilized source comes from oil palm empty fruit bunch, which is a solid waste product produced by the palm oil factory industry. And lignin from empty fruit bunch will be used as a precursor in synthesizing of carbon dots. Carbon dots is a term for nano-sized carbon materials, and carbon dots are also known as carbon quantum dots, which have the characteristic property of fluorescence. Top-down and bottom-up methods can use to synthesize carbon dots. In the top-down synthesis method, larger carbon group structures or macromolecules are destroyed into small carbon dots physically or chemically. And the bottom-up synthesis method refers to the polymerization and carbonization process of a series of small molecules into nanoscale carbon dots through chemical reaction. The hydrothermal method is one of the bottom-up approaches. In the synthesis of carbon dots, the addition of doping or passivation agents can change the properties of the carbon dots, which can improve certain performance, affect the characteristics of the carbon dots, and affect the luminescence intensity of the carbon dots. Dopant materials that can be used include nitric acid as doping element nitrogen, sodium metabisulfite as doping element sulfur, boric acid as doping element boron, ethylene diamine as doping element nitrogen. And its application, carbon dots can be used for sensing by imaging, drug delivery, photocatalysis, and antibacterial application. The study suggests that carbon-containing biomass with nitrogen dopes can be used for bioimaging microorganisms. And in this present work, lignin isolated from oil palm empty fruit bunch was used as a precursor for synthesizing nitrogen doped C dots or NC dots with urea as the dopant agent. And the practical uses of the NC dots were then used for bioimaging applications to determine Escherichia coli and Staphylococcus aureus under radiation of 395 nanometer of UV light within optical microscope in buffer solution. The objective of this study was to find out if carbon dots formed from oil palm empty fruit bunch, lignin, can be used for imaging Escherichia coli and Staphylococcus aureus bacteria. Lignin isolation from oil palm empty fruit branch by soda pulping method. The sample was added with sodium hydroxide solution, and then were added with sulfuric acid to lower the pH. And then after we add the sulfuric acid until pH 2, it will be the carbon dots will be precipitated. And synthesis of carbon dots from oil palm empty fruit bunch lignin using the hydrothermal method. 100 milligrams of isolated lignin dispersed in aqua PI, and urea with solution was added with varying concentration of urea as dopant in aqua pro injection, and added sulfuric acid as oxidator. Then dialyzed the carbon dots in dialyzing membrane against aqua PI for 5 days to get pure carbon dots. And we concentrated the carbon dots using a rotary evaporator at temperature 75 degree, and the results of the 3 carbon dots were then characterized. Adding variations in urea concentration based on the density of urea, namely 1.32 gram per centimeter cubic, then dissolving it with 10 milliliters of aqua PI, which can be seen in table 1. Carbon dots characterization included testing for functional group analysis, the absorbance value, the luminescence intensity values, and the photoluminescence decay time of the 3 carbon dots. Escherichia coli bacterial imaging test. One colony of Escherichia coli bacteria was taken and placed in sterile nutrient-growth media and incubated at 37 degrees for 24 hours, and then centrifuged the E. coli bacteria in the column, and then we suspended the bacterial cells in phosphate buffer solution, and then mixed the E. coli bacterial suspension with the carbon dots 3 solution, and homogenize for 10 minutes and incubate for an hour. And then the mixture we took and placed on a glass object, dried and observed under a light microscope, and in a microscope with 395 nanometers UV light source. And we did the same procedure for Staphylococcus aureus bacterial imaging test. We can see the lignin isolated from oil palm empty fruit bunch was dark brown with a yield 7.20%. And the characterization of lignin with FTIR spectrophotometer, the spectra showed that the isolated compound was lignin because functional groups characterize lignin at certain wave numbers. And there were similarities in functional groups between lignin isolated from sample and commercial lignin. Synthesis results of carbon dots from sample lignin using the hydrothermal method. We can see that the characteristic of the 3 C dots in yellow solution, and at UV 365 nanometers and at UV 395 nanometers. The aqueous showed no fluorescence, and the 3 of carbon dots showed cyan fluorescence. Characterization of carbon dots with FTIR spectrophotometer. Based on the figure, all carbon dots samples had similar functional groups that indicate NC dot is successful synthesis. And the characterization of carbon dots with UV visible spectrophotometer. As shown in figure, the highest intensities of emission spectra were observed for NC.3. Characterization of carbon dots with photoluminescent spectrophotometer. The emission peaks of the 3 carbon dots showed a cyan spectrum. This is in accordance with the cyan color spectrum at a wavelength between 435 to 520 nanometers. And the morphology of carbon dots with HRTEM. Carbon dots particles were distributed and tended to have a spherical shape. And carbon dots with urea dopant had an average particle size of 7.381 nanometers, and waxy dots without urea dopant had an average particle size of 16.521 nanometers. Imaging test results for Escherichia coli and Staphylococcus aureus bacteria. A, B, C, and D were bioimaging results of daylight radiation, and E, F, G, and H are bioimaging results of 395 UV radiation. We can see from the figure, gram-negative bacteria, Escherichia coli, did not fluoresce when mixed with the carbon dot sample, which was observed under a microscope with 395 nanometers UV light source. However, gram-positive bacteria, Staphylococcus aureus, showed fluorescence by coccus. And the illustration of the fluorescence mechanism of carbon dots in Staphylococcus aureus. Gram-positive bacteria have a thick peptidoglycan layer containing lots of negatively charged teichoic acid as the anionic part, resulting in electrostatic binding of carbon dots nanoparticles or cationic molecules to the cell surface of the bacteria. And the conclusion of the study, carbon dots synthesized from empty palm fruit bunch lignin using the hydrothermal method can be used for imaging Escherichia coli and Staphylococcus aureus bacteria. And Staphylococcus aureus bacteria show cyan fluorescence, while Escherichia coli bacteria do not fluoresce. However, further study is required to determine the toxicity test of the C dots from the lignin of empty palm fruit bunch. And this our publication of this study. Thank you for your attention. I give it to moderator.
Thank you very much, Dr. Sumaiya. We have 2 questions. If you have any question, any question, can raise your hand, and you can chat in the chat box. Okay, since there is no question, so I will ask Dr. Sumaiya. Could you please explain more why the Staphylococcus aureus is cyan fluorescence, while bacteria Escherichia coli is not fluorescence?
Thank you, Mr. Roni, for your question. This slide is for gram-positive bacteria and Staphylococcus aureus. But for Escherichia coli, do not flourish because the gram-negative bacteria have more complex structures with thinner peptidoglycan layer and higher amounts of lipopolysaccharides. So it makes them impermeable to NC-dots. Yeah, and then E. coli displayed a weak electrostatic interaction. So we could not be observed with a microscope with UV 395 nanometers. I think that's the answer.
Okay, thank you, Dr. Sumaya. Any question? We have 1 minute left. If there is no question, that's the end of the session of Dr. Sumaya. Thank you so much for amazing presentation about the carbon dots. And also last, thank you. I have done my job as a moderating in this seminar. I give it back to MC.
All right. Thank you once again, Dr. Roni, for moderating this session. Also, thank you for the amazing presentation, Dr. Sumaya. The session was so insightful and wonderful. So, all participants, our next agenda is still to learn about nanomedicine. And the session will be delivered by Dr. T. Ismaneli Hanum, SSI, MSI Apotheker. And let me to invite our moderator for this session, Mrs. Mariana, SSI, MSI Apotheker. Mrs. Mariana is a lecturer in the Faculty of Pharmacy, Universitas Sumatera Utara, as a Vice Dean 3, and Mrs. Mariani also has several publications in international index journals with a specialty on the pharmacology side. So, Mrs. Mariana, hope you are ready to guide us. And Mrs. Mariana, time is yours.
Thank you, Mr. Fauzan. Can you listen to my voice?
Yes, of course, it's very
Okay.
clear.
Thank you. Assalamualaikum warahmatullahi wabarakatuh. Dear participants, we are going to begin with invited speaker session in a moment. I'm Mariana, will be your moderator in the session. Moving on here with us, our invited speaker, Dr. T. Ismaneli Hanum, is a lecturer in Faculty of Pharmacy, Universitas Sumatera Utara. Dr. Ismaneli research is highly concentrated in the field of nanosuspension studies, reflecting a significant level of interest in this area. Dr. Ismaneli will be discussing her paper with the topic of development nanosuspension ketoprofen as an anti-inflammatory in vitro in vivo evaluation. So here with us, Dr. T. Ismaneli Hanum. Assalamualaikum, Dr. T. Ismaneli. How are you? You are still mute, Dr. Ismaneli.
Thank you, Ms. Mariana.
Okay.
I'm fine today.
Alhamdulillah. Thank you, Dr. T. Ismaneli. We start our the presentation now. The time is yours.
Thank you, moderator and committee, for giving me the opportunity to present our research. Our research in development ketoprofen nanosuspension as an anti-inflammatory in vitro in vivo evaluation. As we know, ketoprofen is a class of non-steroidal anti-inflammatory drugs to treat pain and inflammation in rheumatic disease. Ketoprofen categorized into biopharmaceutical classification system, class 2, which has low solubility in water, high membrane permeability, less absorbed in the digestive tract. Ketoprofen can be found in enteric-coated tablets on the market. It has some disadvantage. Slow onset of treatment can cause duodenal ulcers and dose dumping. And the mechanism of NSAID side effect. During the drug dissolution process, a thin layer containing in high drug level surround the drug particles, so that if the NSAID is in solid crystalline form, the large particle size in an insoluble state comes into direct contact with the gastric mucosa. So we can damage the phospholipid layer of the mucosa. This weakness is overcome by nanosuspension formulation that have small particle sizes and fine distribution. In nanosuspension, solid drug particles are dispersed in a liquid phase. They are stabilized by surfactants and polymers, creating a submicron colloidal dispersion with a size of less than 1 micrometer. Advantages of nanosuspension increase drug absorption and bioavailability, dosage reduction, and side effects. The objective of this research compares the ketoprofen nanosuspensions with ketoprofen suspension with regard of dissolution rate, bioavailability, anti-inflammatory effect, and local irritation effect on the stomach. These are the methods: experimental research, preparation of ketoprofen, characterization, organoleptic stability, morphology observation of ketoprofen by time method, and particle crystal state observation, and then determination of ketoprofen level in the preparation. Here we can see preparation of ketoprofen nanosuspension by using evaporative precipitation method. Ketoprofen dissolved in ethanol as organic phase, and then twin 80s as surfactant in distilled water, and then add polymer polyphenol pyrrolidone, and then add 20 milliliter distilled water as liquid phase, and then slowly edit the organic phase into the aqueous phase, and then stir using a hot plate stirrer at 50 temperature at 1500 RPM for 1 hour, and then put into a vial. Next, this is the result of the observation: physical stability during 3 months of storage. This is a variation of concentration of PVP. As we can see, after 3 months of storage, data reveal that F3 and F4 remain stable, and while F2 and F5 precipitated or sedimentation. And then if you can see the suspension, it looks cloudy, like milky white. And then we can go to measure the particle size or dispersity index and zeta potential at the beginning and after 3 months of storage. The higher the PVP concentration, make the larger the particle size. and the polydispersity index less than 0.5 are stable, indicating sufficient nanosize distribution. When values greater than 0.5 indicate very broad distribution. And but the zeta potential of plus minus 20 millivolt can be sufficient to prevent drug particles from aggregation and precipitation. And then here we can see a dissolution test of nanosuspension and suspension ketoprofen. First, ketoprofen has low solubility in the solution medium pH, pH acidic medium. And ketoprofen suspension has a larger particle size than 3 nanosuspension formulas. Reduction in drug particle size leads to increased surface area and consequently increased contact of nanoparticles with dissolution medium, so can increase dissolution rate. As we can see, 3, the higher dissolution rate. And morphology observation of ketoprofen nanosuspension. As we can see, nanosuspension particles were spherical and amorphous with a vertical size of 100 nanometers. With PV polymer and twin actinic as surfactant as stabilizer, cause no crystal growth. And the measure of crystal state observation of nanosuspension particles. Ketoprofen nanosuspension, there is a decrease in crystal structure. We can see ketoprofen in pure form and in nanosuspension. There is decrease in crystal structure due to endothermic melt, peak shifting from 90 to 103. The presence of broad peak indicates, broad peak indicates the change of ketoprofen into an amorphous form. And then the bioavailability test, ketoprofen nanosuspension and suspension, we do in animal rabbits with crossover design method. And then testing anti-inflammatory activity with the Paul edema method, calculating the value percent inflammation. And then study of the irritating effect of the preparation on the rat stoma. Measure the macroscopic observation of the stoma. Bioavailability testing of ketoprofen nanosuspension and suspension. We do male rabbits and drug level in serum were measured using HPLC. The result at half hours, it is clear that the plasma level of ketoprofen nanosuspensions are higher than suspension. The highest drug concentration was achieved by the nanosuspension preparation at 2 hours. And then the smaller particle size of nanosuspension than suspension increased drug solubility and drug absorption in the gastrointestinal tract, so increased drug level in blood plasma. Here's the data correlation of in vitro and in vivo tests. There is a correlation between in vitro and in vivo tests. The coefficient correlation is near to 1. And then anti-inflammatory activity tests, we do 28 reps. So we do whole edema method with the instrument of plethysmometer. And then use carrageenan to induce inflammation. The test substance is given orally according to the group in each rat, allowed to stand for 60 minutes, and then injected carrageenan, allowed to stand for 60 minutes, and then insert the rat's left paw into the transducer on the digital plethysmometer until the liquid rises, and then record the number on the plethysmometer, and measurement were taken every 60 minutes for 6 hours. Here's the data, anti-inflammatory activity testing. The negative control, highest percent inflammation value, while nanosuspension 300 dosage, lowest percent inflammation value. And percent inhibition of inflammation, nanosuspension 300 milligrams, inhibition inflammation more higher compared to suspension. Inhibition of inflammation until 360 minutes. And then we can see the area under curve result percent inflammation and inhibition of inflammation. And the conclusion of this method, with increasing doses in both suspension and nanosuspensions, the anti-inflammatory effect increases. The nanosuspension preparation provided a higher anti-inflammatory effect compared to the ketoprofen suspension. The smaller the particle size and consequently higher dissolution rate contributed to the greater effect on anti-inflammatory activity. And then the data of correlation between plasma level of ketoprofen nanosuspension and anti-inflammatory effect, there is a correlation near to 1 coefficient correlation. And the method of irritation test, we did 40 ml rates divided into 8 groups. The test preparation were given to rats after 6 hours, sacrificed by dislocating the neck of the rat. Gastric organs were dissected, and then the stomach were isolated and opened by surgery on the major curvature for further cleaning with a solution of 0.01 normal sodium chloride solution, and then observe erosion of the mucosal layer. The result of irritation study, number of ulcer of nanosuspension 100 milligram was significantly different from 100 milligram suspension. In terms of ulcer index, only the nanosuspension group and the suspension with a dose of 200 milligram were significantly different. Nanosuspension that dissolves evenly distributed in stomach with small particle size and doesn't agglomerate can reduce irritation in the stomach of rat compared to ketoprofen suspension. Here's the microscopic observation of rat stomach. As we can see, the nanosuspension, the higher the dosage, the minimal of irritation. The conclusion of this study, ketoprofen nanosuspension with PVT and twin AT as a stabilizer has a higher dissolution rate, bioavailability value, and anti-inflammatory effect, and lower irritating effect than ketoprofen suspension. Thank you for your attention. I will give it back to the moderator.
Thank you, Dr. Ismaneli, for sharing your research. It was so insightful. Next, we open the Q&A session, question and answer session. Dear participants, if you have any questions, please raise hand, unmute yourself, or write the question in the chat box. Okay. While waiting for the question, maybe I have a question for Dr. Ismanili. I want to know what are critical things or critical points in preparing nanosuspension, especially with ketoprofen.
Yeah, thank you for the question, Ms. Marianne. Preparing nanosuspension, especially with a drug like ketoprofen, involves several critical points to ensure the stability, uniformity, and effectiveness of the formulation. There's some critical consideration in preparing nanosuspension with a particle size reduction. So many technique, one high pressure homogenization, medium milling, or sonication. In this research, we were conduct with evaporative precipitation method. But the weakness of the evaporation precipitation method is the using the need for suitable solvent. The choice of solvent is critical in the evaporative method. Some solvent may not be suitable for certain drugs. Or may pose challenges in terms of toxicity or safety or environmental concerns. But if we use method of homogenization, high pressure homogenization, the challenges because the instrument is so expensive, yeah, so expensive. But the high pressure homogenization not need soften, so more natural, yeah. So maybe this is my answer, Miss Marian.
Okay, so if we want to make the nano suspension to another drugs, so the method can be different with the ketoprofen, or solvent must be can be different with the ketoprofen, right?
Maybe you can repeat the question.
Okay.
Yeah.
Okay. I mean, so if you want to make the nanosuspension for another drugs instead of ketoprofen, so the solvent is different and the method can be different also, or we can use the same method to make a nanosuspension ketoprofen and we apply to another drugs, or we have to choose different method.
Yeah, we can choose a different method, or we can use the evaporative precipitation method. But in especially to drugs for nanosuspension, usually we usually of drugs with poor soluble, yeah. So to make soluble the drug is have to use solvent, or we can use the high pressure homogenization, like I said. Yeah. Maybe we can so many research, yeah, about the nanosuspension with other drugs. Yeah, maybe I think this is my answer.
Okay, thank you very much, Dr. Ismanili Hanum. Once again, I would like to say thank you for your amazing presentation and useful knowledge you just shared with us. Dear participants, the presentation is really interesting. The study compares the dissolution rate, bioavailability, anti-inflammatory effect, and local irritation of ketoprofen nanosuspension and ketoprofen suspension. And the nanosuspension prepared with the thin HE and PVP, a stabilizer, and ethanol as a ketoprofen solvent showed clear appearance, smallest particle size, spherical particle surface morphology, amorphous crystalline state, and stability for 3 months at room temperature. The nanosuspensions also had a strong in vitro, in vivo correlation with plasma drug levels and inflammation inhibition. The ketoprofen nanosuspension provide higher anti-inflammatory effects and lower gastric irritation. Thank you, Dr. Isma Nelly, and dear participant, I give back to MC Mr. Fauzan. Thank you.
Thank you very much, Mrs. Mariana, for bringing the session. Also, thank you for the amazing presentation, Dr. Ismaneli. The session was so wonderful and insightful for future research. Okay, all participants, I'm sure you are still enthusiastic at this time. So let me to inform you that our next speaker is Professor Kanjiro Miyata, PhD, and the session will be guided by Professor Dr. Hakim Bangun Apoteker. He is a professor of pharmaceutical technology in the Faculty of Pharmacy, Universitas Sumatera Utara. And Professor Hakim Bangun also has several publications in internationally indexed journals with a specialty on the pharmaceutical technology side. He has so much experience and skill in pharmaceutical development, particularly in nanomedicine. So all participants, please welcome Professor Dr. Hakim Bangun Apoteker. Professor Hakim, time is yours.
Thank you, Mr. Fauzan. Please welcome Professor Miyata. Our keynote speaker now, Professor Kanjiro Miyata, PhD. He is a professor in Tokyo University, Department of Material Engineering. Dr. Miyata graduated from Tokyo University, bachelor, master, and PhD also from Tokyo University. After graduation, he worked as academic staff in Tokyo University, and his position now is a professor. He writes many papers, and his index or high index now about 56, and he received many, how to say, he is also a member of various society, and he is also an associate editor of journal Control Release. Professor Miyata received many awards as a scientist from many society. So please welcome Professor Miyata.
Yeah, thank you for your kind introduction. And yeah, I'm very happy to present the research here. And can I share my slide?
Not yet.
Oh, yeah, okay, sorry. Okay.
Yeah.
Okay, anyway, thank you. Thank you for inviting me to this exciting meeting regarding the nanomedicine. Thank you.
Still not seen yours.
Right?
Not yet.
Oh, yeah, okay, sorry. Okay.
Yeah.
Okay, anyway, thank you. Thank you for inviting me. Sorry, can you hear me?
Not yet. Oh, okay. That's okay. All right. Konnichiwa, Professor Kiwa.
Yeah.
Okay.
Yeah, nice Japanese. Yeah. Okay, should I start my
Yeah, okay. Yeah.
Can you see my slide?
Yeah, that's right. Yeah.
Okay, so let's start my presentation. Okay, anyway, thank you for inviting me. And today I will talk about our polymeric nanomedicines for nucleic acid delivery. But, sorry. Okay, first of all, let me introduce our laboratory. My laboratory is in University of Tokyo, and our laboratory is especially focused on the polymeric nanomedicine. So we have developed a variety of polymeric materials for mainly drug delivery. And importantly, my supervisor, Professor Kazunori Kataoka, he was a previous PI professor in this laboratory, and he is a pioneering researcher for polymeric nanomedicines, especially developing polymeric micelles. Okay, of course, polymeric micelles are a kind of just a polymer nanoparticle, but he has started the application of polymeric micelles for drug delivery for nanomedicine. And in Japan, this year, he was selected kind of Nobel Prize candidate, but unfortunately, he didn't get it. But I hope in the near future, he'll get it. And now our laboratory has 18 people, and approximately 30% of the students are from foreign countries. And also, if you are interested in our laboratory, please contact with me or Aurea from Indonesia. She's working in our laboratory as a postdoc. Okay. My first question or my first issue is why nanomedicine? I guess you all know what is nanomedicine and also the significance of nanomedicine. But actually, I'm so sure about your research background. So I put this slide, the initial stage of my presentation. So my answer is nanomedicine can enhance the drug efficacy and or reduce the adverse side effects by controlling the drug distribution in your body. So this is a very simple example. This is a cisplatin, is very conventional anti-cancer drug with a low, small molecular weight. So if we now, so you know, this cisplatin can be available for human cancer patients. This is very conventional anti-cancer drug. But if we inject this small drug into our body, the resulting biodistribution shown here. Of course, this is not from a human cancer patient, but from a mouse cancer model. But similar biodistribution is observed for the human cancer patient. Importantly, almost all cisplatin was accumulated in the kidney. This means cisplatin treatment resulting in the kidney dysfunction as a kind of adverse side effect. And the other important point is very low tumor accumulation. Just less than 1% of injected drugs are accumulated in the tumor tissue. Okay, so you know, conventional cancer chemotherapy resulted in the very small anti-cancer effect with strong adverse side effects. Okay, so this is the reason for the strong adverse side effect of conventional anti-cancer drug. So the initial purpose of cancer nanomedicine is to control the biodistribution of anti-cancer drugs, especially by decreasing the kidney accumulation and also increasing the drug accumulation in the tumor tissue. Okay, the purpose is very simple. We just increase the tumor accumulation and decrease the drug accumulation in the healthy tissues. But of course, it's not easy. So most important design criteria of cancer nanomedicine is the targetability. Okay, so how can we target special organs, tissues, especially cancer tissues? Okay, conventional methods are there are two conventional methods. One is passive or size-dependent cancer targeting. The other one is active or ligand-mediated targeting. Okay, so today I will talk about both strategies tested in our laboratory. But first topic is the passive or size-dependent cancer targeting. Okay, the underlying mechanism of passive tumor targeting is based on the blood circulation system. As you can see, bloodstream controls the transport of drugs injected into our body. So, drugs distribute into organ and tissues through the pores of blood capillaries. So, the pore size is very critical for the drug distribution in your body. And for this purpose, we need to know the pore structure in our organs and tissues. So the pore size varied according to the tissue. For example, in the case of muscle, skin, lungs, the pore size are very small, approximately less than 10 nanometer. Okay, and in case of small intestine and kidney, these tissue blood capillary have a kind of fenestra for enhanced molecule penetration. The other interesting tissues are liver and spleen, which has a discontinuous endothelium. This means liver, spleen can accept much larger nanoparticle drug formulation, presumed due to the much larger pore size of approximately 100 to 1,000 nanometer. Okay, this is the summary of the correlation of size and tissue pore size. So as you can see, small muscle, skin, and lung vasculature has a smaller pore. So small nanoparticle, small molecular anti-cancer drug can penetrate these blood capillary vasculature. So small molecule cannot be, doesn't have any targetability for cancer treatment. But if we increase the size of nanomedicine, so larger nanoparticle cannot penetrate the small pore of small intestine and kidney and muscle, blah, blah, blah. So we can control the biodistribution of drugs by changing the nanoparticle size. And another important point is the pore of tumor vasculature. Okay, it considers as around 100 nanometer. So if we prepare nanoparticles with the size of 10 nanometer, they can penetrate the tumor vasculature for selective tumor targeting without distribution into the healthy tissue with a much smaller blood vasculature pores. Okay, and I show kind of movie. Sorry, this guys includes Japanese, but yeah. So small anti-cancer drug can penetrate healthy tissue with the small size, but the larger micellar nanoparticle cannot penetrate the healthy tissue vasculature. In contrast, in the tumor tissue, the blood capillary pores are much larger. So this type of larger micellar nanoparticle can penetrate selectively into the tumor tissue. So this is a kind of site-dependent tumor targeting mechanism. Sorry, I have the message. Sorry. Okay, and this is a experimental example of the site-dependent tumor targeting. This is published by Professor Maruyama in Tokyo University. I like this paper because the result is very interesting and very informative. So in this paper, he tested 3 types of nanomedicines. The one is pegylated liposome with the size of 400 nanometer. And the second one is 100 nanometer pegylated liposomes. And the other one is 100 nanometer liposomes without PEG, okay, surface coating or surface bioengineering. And he compares the different biodistribution using these 3 types of nanomedicines. And from the standpoint of blood circulation property, 100 nanometer pegylated liposome, this one, shows the highest blood circulation property. And this one, 100 nanometer non-pegylated liposome, shows the lowest blood circulation property. And the final destination of this nanoparticle was mainly in the liver, okay, as you can see. And in case of this larger nanoparticle with PG, accumulated preferably in the spleen, okay? And this small one selectively accumulated in the tumor tissue. So by changing the surface modification and the size of nanomedicine, we can control the final destination or target tissue in cancer, treatment. So if our target is liver, we had better use this formulation. And if our target is spleen, we had better use this formulation. And if our target is cancer tissue, this 100 nanometer pegylated liposome should be best for treatment or cancer accumulation. So based on this finding, the actually pegylated liposomes, so-called doxil formulation, is currently approved for treatment of Kaposi sarcoma and recurrent ovarian cancer. But this anti-cancer nanomedicine is not so, how can I say, not so general for cancer treatment. So I need to mention about the current problem or issue in cancer nanomedicine. Okay, one is cancer heterogeneity, and the other one is the tumor tissue impermeability. Okay, this is the example of the fibrotic pancreatic cancer model in mice. And this is a pancreatic cancer tissue immunostaining figure. So this T means cancer cell nests. So you can see many cancer cell nests, but this cancer cell nest is, how can I say, surrounding with a red and green color. That means blood vessel and stromal tissue, fibrotic stromal tissue. Okay, so these cancer cell nests are surrounded or sequestered by fibrotic stromal tissue. So if we inject pegylated liposome with the size of 100 nanometer, they apparently accumulated in this region. This means pegylated liposomes are mainly accumulated in the stromal tissue, not cancer cell nest. Okay, so this means the fibrotic stromal tissue may act as a physical barrier for pegylated nanomedicine or drug delivery. Okay, this is a kind of illustration. So 100 nanometer size nanoparticle, like a pegylated liposome, can stably circulate in the bloodstream, and the so-called enhanced permeability and retention effect, EPR effect. And through EPR effect, this nanoparticle can penetrate or extravasate tumor blood capillary. But unfortunately, there is kind of stromal tissue before reaching the cancer cell nest. So this nanoparticle seems to be entrapped by stromal tissue. As a result, they cannot reach the cancer cell. So almost no accumulation in pancreatic cancer cell. So this is a kind of current problem of cancer nanomedicine. So we need to overcome this type of biological barriers. So I will talk about our research approach. And as I mentioned, we have developed a variety of block copolymers or functional block copolymers to make it's okay to encapsulate various drugs, especially nucleic acid drug. And we can prepare a variety of nanoparticulate formulations by changing the block polymer structures. So we can prepare small, very small nanoparticle and polymeric micelles and polymeric vesicles. And this is a kind of animation for nanoparticle formation. And also, I'm sorry, this slide includes Japanese. This means a hydrophilic segment and a hydrophobic segment. So if we covalently conjugate hydrophobic anti-cancer drug into the side chain of hydrophobic segment in aqueous solution, this polymeric material can self-assembly to make a polymeric micelle to decrease the hydrophobic region from the aqueous solution. And this is polymeric micelle. Composed of a quasar structure. Core is including anti-cancer drug, and the outer hydrophilic collagen glycol shell called nanoparticle core for better biocompatibility or longer blood circulation property. And the other point of my talk is nucleic acid delivery. So recently mRNA vaccine is developed by Pfizer, BioNTech, and Moderna. So recently, mRNA delivery is especially highlighted as the next generation of pharmaceuticals. And our laboratory highlighting mRNA delivery, gene delivery, and also small nucleic acid, siRNA, mRNA, antisense oligonucleotide delivery. And importantly, these nucleic acid drugs are very powerful as a kind of to treat various intractable diseases. However, as you may know, the problem of nucleic acid drugs are poor bioavailability, or they cannot directly penetrate the cytoplasmic membrane, and they need delivery formulation. mRNA vaccine is utilizing lipid nanoparticles for efficient intracellular delivery. So now we are developing nucleic acid delivery formulation by using polymeric materials or polymeric nanomedicines. Okay, so I introduced our current approach. So as I mentioned, pancreatic cancer has a kind of physical barrier for 100 nanometer nanoparticle tumor accumulation. So our approach or idea is very simple. We just decrease the size of nanomedicine much lower than 100 nanometer. So downsizing is our strategy. Okay, this is a example or a paper published from our group. So actually, we have developed this ultra small oligonucleotide loaded carrier for better tumor tissue penetrability. Okay, so lipid nanoparticle has a size of 100 nanometer, and our previous polymeric micelle has approximately 30 nanometer, and small drug or naked oligonucleotide has a size of 5 nanometer. So of course, most smallest formulation is a naked drug, but unfortunately, our body has a kind of threshold for renal clearance at approximately 10 nanometer. So the drug with the size of less than 10 nanometer is readily excreted from our body due to the kidney filtration. So we need to prepare a nanomedicine with a size of more than 10 nanometer. So we focused on the approximately 15 nanometer for high tumor penetrability with reduced renal clearance. Okay. So this is a kind of phase diagram of polymeric nanoparticle formulation. So this means we can control the nanoparticle formation by changing the polymer structure or polymer molecular weight. Okay, so we are using a kind of block copolymer protein glycol under drug loading segment. So this is block copolymer, and the important parameter is the molecular weight of protein glycol. MWPG. So if we apply MWPG, molecular weight PG is 2000, we can prepare vesicular hollow nanoparticles. And if we apply much longer polyethylene glycol, we can prepare a smaller micellar nanoparticle with a size of approximately 40 nanometer. Okay. And importantly, the nanoparticle formulation is affected by the preparation concentration. So we can clearly observe the nanoparticle formation at a higher polymer concentration. But if we decrease the preparation concentration, the nanoparticle is dissociated due to the equilibrium shift to the free polymer or free structure. So if we focus on these lower concentration range, we clearly observe the smaller, we can prepare a much smaller nanomedicine, okay, due to the dissociation of large nanoparticles. And also, you can see PG molecular weight or length is very critical. So if we increase the molecular weight of PG, or if we apply longer PG, we can prepare much smaller nanoparticle due to the strong steric hindrance of flexible polyethylene glycol chains. Okay, so actually we prepared siRNA-loaded nanomedicine by using this block copolymer. So finally, we this also includes Japanese, and but importantly, if we precisely control the charge number of polymer, we can precisely control the association number between polymer and oligonucleotide drugs, siRNA. So if we utilize this Y-shaped polymer materials, we can selectively prepare this small nanoparticles comprising 2 polymers and 1 oligonucleotide. Okay, this is an illustration. So actually, we prepare this Y-shaped polymers, and this includes 2 very long polyethylene glycol chains and cationic polylysine chains. Okay, and siRNA or antisense oligonucleotides have negative charges derived from the phosphate groups. So this polylysine cationic chains can electrostatically interact with the negatively charged nucleic acid to form a polyion complex, PIC, or nanomedicine. Okay, so siRNA has 40 negative charges. So if we apply polylysine with 20 positive charges, it can neutralize the siRNA negative charges by 2-to-1 complex formulation. Okay, 2 polymer can charge neutralize siRNA negative charges, and also in case ASO antisense oligonucleotide with a negative charge of 20, in this case, 1 polymer can selectively bind to antisense oligonucleotide to form a 1-to-1 unit polyion complex, UPIC. And actually, we observe the size of nanomedicines by fluorescence correlation spectroscopy, and in both cases, siRNA and antisense oligonucleotides, we successfully prepared small nanomedicines with a size of approximately 16 or 18 nanometers. And also, we clearly observe the siRNA loading number or antisense oligonucleotide loading number is approximately just 1. This means just 1 oligonucleotide drugs are selectively incorporated into 1 nanomedicine formulation. So next step is to check the blood circulation property. Recently, we directly observe the blood circulation nanomedicines by using a confocal laser scanning microscopy. So this is mice, and this cover glass, and we can observe the blood circulation of nanomedicines by focusing of these large blood vessels. So this is a real-time movie. Okay, the left one from naked siRNA, the right one from our small nanocarriers. So oligonucleotide is fluorescently labeled, so we can see the red color of circulating siRNA. So In case of naked sRNA, we couldn't see any red fluorescence signal. This means naked sRNA is readily eliminated from the blood circulation. In contrast, we can clearly observe the red fluorescence from the bloodstream by using a small nanocarrier. This means a small nanocarrier can stably circulating in the bloodstream by delivering oligonucleotide sRNA. This is a quantified result, and the x-axis means the time after intravenous IV injection. And in case of sRNA, naked sRNA, the fluorescence signal is readily reduced with time because of the renal excretion. In case of our small nanocarrier, it shows a much higher blood circulation property as observed in the real-time movie. And importantly, this is a commercially available transfection reagent, imlifeptamine. In this case, we clearly observe the similar blood circulation profile to the naked sRNA. But in this case, imlifeptamine should not be eliminated from the kidney filtration. So we are thinking this nanoparticle can selectively accumulate in the liver without eliminating from the kidney. So the blood circulation profile are very similar between naked sRNA and the imlifeptamine, but the final destination or biodistribution should be quite different. But anyway, our nanoparticle formulation shows a much better blood circulation property. And yeah, this is kind of a mechanistic study to understand our small nanocarrier blood circulation properties. So this NP ratio means kind of the molar ratio between polymer positive charges and oligonucleotide negative charges. So the increase, the NP ratio means we added much more polymer materials compared with oligonucleotides. So this NP 10 means small nanocarriers co-circulating with much amount of free polymers. Okay, so importantly, the blood circulation property is significantly affected by the adding polymer amount. Okay, so if we increase the added polymer, the resulting blood circulation property is dramatically enhanced. So now thinking these free polymers are very important for good or high blood circulation property of our small nanocarriers. So, and also we tested different oligonucleotide structure. So this is a non-modified, non-chemically modified sRNA. This means this is kind of a natural oligonucleotide structures. And but recently, many pharmaceutical companies have developed kind of chemically modified oligonucleotide for showing the much better enzymatic tolerability. And yeah, we clearly observe the difference between non-modified sRNA and highly modified sRNA. So in case of non-modified sRNA, we need much more polymeric materials or higher NP ratio. But if we apply highly chemically modified or stable sRNA, we don't need much amount of polymeric materials, probably due to the higher tolerability to enzymatic degradation of sRNA. So anyway, now we are thinking our small nanocarrier is kind of equilibrium or dynamic formulation shown in this movie. So in case of a conventional lipid nanoparticle or liposome, they are very stable, almost no change in the composition. But our small nanocarrier is a very, very dynamic formulation. So polymer is always exchanging in the bloodstream shown in this movie. And we directly observe or demonstrate this type of dynamic formulation or dynamic docking in our body based on the fluorescence resonance energy transfer technology. So here we applied fluorescence, donor fluorescence molecule into the polymeric materials. And yeah, also we conjugated acceptor fluorescence molecules to the sRNA. So if these materials are docking in our body, it shows a fluorescence energy transfer or FRET signal. Okay, but if we apply additional inhibitor polymer without any fluorescence molecules, it should, if this inhibitor polymer can combine with siRNA or acceptor oligonucleotide, the FRET signal should be decreased or relaxed. So based on the FRET signal, this signal, we can directly or indirectly observe the interaction between polymer and oligonucleotide in the bloodstream. Okay, this is a movie. So first, the FRET signal is very low because just donor polymer is circulating in the blood. But 5 after minutes, we injected acceptor siRNA, and we clearly observe the change in the color. This means this injected siRNA is directly caught by donor polymers. And 10 minutes later, we injected inhibitor polymer, and we also clearly observe the change in the color at around 10 minutes. Yeah, color is changed. This means inhibitor polymer is also interacting with the siRNA in the bloodstream. So this is kind of evidence to demonstrate the direct interaction between polymers and siRNA in our blood, in the bloodstream. So this is blood vessel. Okay, and we confirmed a very good blood circulation property of our small nanocarrier. Then we confirmed the tumor targetability. This is more important for cancer nanomedicine. And this is the result. So here we applied a GFP, green fluorescence protein expressing pancreatic cancer cells. So we can see the cancer cell based on the green fluorescence color. Okay. And here we labeled siRNA with Cy5 fluorescence molecules. So we can see the drug distribution from the red color. So the next figure from immunopectamine, a commercially available transfection reagent, and the right one is from our small nanocarriers. And you can see we clearly observe the co-localization of red and green fluorescence signal. This means our small nanocarrier can penetrate or can reach the pancreatic cancer itself. In contrast, we couldn't see any red fluorescence signal in the immunopectamine treated samples. This means larger immunopectamine lipid nanocarrier cannot accumulate in the pancreatic cancer tissue. Okay, this is a more precise analysis of tumor tissue penetrability of our small nanocarriers. So this, the center one is from our small nanocarriers. So we, quantitatively analyze the fluorescence signal from here to here, along with the arrow. Okay, and we observe the this 0 micrometer means a kind of blood vessel, and this direction, the means more depth tumor tissue penetration. So a small nanocarrier can penetrate in the tumor tissue at around 80 or 90 micrometer because of the small size. In contrast, immunopectamine didn't show any fluorescence signal from the blood vessel to the deep tumor tissues. So we clearly observe the much better tumor penetrability of our small nanocarriers. And this is more, how can I say, mass biodistribution in the subcutaneous pancreatic cancer model. So we compare the naked siRNA oligonucleotide and the small nanocarriers, and we clearly observe the much higher accumulation of our small nanocarriers in the pancreatic cancer compared with naked siRNA. And importantly, if we observe the other healthy tissues, almost no significant difference between naked siRNA and the small nanocarriers. This means our small nanocarrier has a very, very good tumor tissue targetability without without specifically accumulating in the healthy tissue. Okay, this is an anti-cancer effect. So we deliver PLK1 siRNA by using a small nanocarriers, and we observe the gene silencing or gene knockdown by PCR for PLK1, and this is Western blotting result. And we observe the gene silencing effect by using our small nanocarriers, and maybe significant anti-tumor activity. So this is a result from a pancreatic tumor model. And we also applied our small nanocarriers for orthotopic brain cancer model. So here, in case of naked ASO, it can distribute whole mouse body, but 6 hours after injection, almost all antisense oligonucleotides eliminated from the mouse body due to the kidney filtration. In contrast, a small nanocarrier efficiently accumulated in the blood, orthotopic brain tumor model here. And it can keep the accumulation even after 6 hours after intravenous administration. And also we clearly observe the very strong gene silencing efficiency by delivering a TUG1, TAG1 taurine upregulated gene 1 antisense oligonucleotide. And we also observe the strong anti-tumor effect by delivering this special antisense oligonucleotide. So we clearly observe the much smaller or shrinking tumor tissue by delivering this antisense oligonucleotide. And also, if we deliver a control ASO, it's kind of non-therapeutic antisense oligonucleotide control. We didn't see any therapeutic effect. And now our small nanocarriers under clinical trial in human cancer patients for siRNA delivery. So now our formulation is under phase 1 clinical trials. And antisense oligonucleotide delivery to the brain tumor tissue. It's now we are preparing the phase 1 clinical trial. Maybe this clinical trial will start in the March or April this year. So so far I introduced our site-dependent passive tumor targeting strategy, but another important delivery technology is active or ligand-mediated targeting. Okay, and why we need active targeting? So we demonstrated ultra small nanocarrier can penetrate the pancreatic cancer and brain cancer tissue with small blood vessel pores. So if there are pores in the blood vessel like this, it's okay. It can be overcome by downsizing nanomedicines, but unfortunately, in some tissues, if there are no such pores, for example, non-cancerous brain tissue, so our downsizing strategy cannot be used for targeting, for example, blood brain tissue. Okay, so we need to think additional delivery strategies. Of course, that is active tumor, active ligand mediated targeting. Okay, so this is a illustration of blood vessel. We have a blood-brain barrier. So this blood-brain barrier, BBB, can disturb the nanomedicine penetration into the brain tissue. This is good because if this type of nanomedicine is readily accumulated in the brain, it may be dangerous because it may induce kind of adverse side effect. So blood-brain barrier is very important for keeping our healthy body. But of course, if we have a kind of brain tissue disease, that problem, we need to overcome the blood-brain barrier, BBB, to get a better therapeutic effect to treat brain disease. So the second topic is how to overcome the blood-brain barrier. So, this is a blood brain endothelial endothelium, and this is bloodstream, and this is brain parenchyma. So we need to consider how to overcome the blood-brain barrier or this blood vessel or endothelial cells. So, but we need our brain need much nutrition. So if we focus on the nutrition, this endothelial cell automatically encapsulate or transfer such nutrition molecules. So, glucose is needed for, how can I say, work needed to keep our brain function. So we focused on the glucose molecule to deliver oligonucleotide to the brain. So actually, we conjugated glucose molecules on the nanoparticle surface by delivering a drug to the brain or to overcome the blood-brain barrier. So in this case, our brain-blood capillary recognize this nanoparticle as a kind of nutrition. Okay, the reason why we focus on the glucose molecules because our brain needs much more glucose molecules. So cerebral vascular endothelial cells has a much amount of glucose transporter glutamate transporter protein. So this is an expression level of glutamate in the brain capillary. And importantly, glucose molecules can be selectively recognized by glutamate structure by delivering nanomedicine to the brain. So, and another important mechanism is glycemic control. So generally, our blood capillary, how can I say, glutamate is distributed in whole blood capillary cells like this. But if we decrease the blood glucose level or under the hypoglycemia condition, so to get more glucose molecule from the bloodstream, glutamate molecule aligned on the surface of these endothelial cells. Then if we increase the blood glucose level, they don't need to overexpressed on the surface. So they are going back to the inside of capillary. So if we synchronize this glutamate recycling system with our glucose conjugated nanoparticles, the glucose conjugated nanoparticles can be transported from the bloodstream to the parenchyma because of the glutamate recycling mobility. So that is kind of glycemic control trigger transcytosis. Okay, and this is a nanoparticle formation result. We clearly observe the 30 nanometer nanoparticle formation by dynamic scattering, and very uniform spherical structures by on the TM imaging. And this is the actual result of brain accumulation. Compared with the non-glucose conjugated nanoparticle, we clearly observe the enhanced accumulation of glucose conjugated nanoparticle in the brain. Importantly, in the accumulation in the other tissue, lung, liver, kidney, spleen, they are not changed by conjugating glucose molecules. So glucose conjugated nanoparticle can be selectively accumulated in the brain tissue without increasing the accumulation in the other tissues. So we clearly observe the brain selectivity or targetability of glucose conjugated nanoparticles. Then we observe the glucose conjugated nanoparticles are rarely distributed from the blood stream to the brain tissue by using intravitreal confocal microscopy. So nanoparticle is labeled with green color, and you can see the green fluorescence outside the blood vessel with time. So now we are thinking this green fluorescence should be due to the BBB penetration of our glucose conjugated nanoparticles. And then we delivered oligonucleotide using glucose conjugated nanoparticles. So actually we, again, we prepare nanoparticles using antisense oligonucleotide by engineering this block copolymers, glucose molecule, polyethylene glycol, and polylysine or cationic chains to form a nanoparticle with glucose molecules. Okay, this is actual result. And here we checked the impact of the number of glucose conjugated on the micellar surface. So this means 0 means no glucose conjugation on nanoparticle, and this means 24 glucose conjugated on one nanoparticle surface. So from this result, we observed the highest brain accumulation of 52 glucose conjugated nanoparticles. Okay, so from this result, the glucose number is very critical for BBB penetration or brain accumulation of glucose conjugated nanoparticles. And interestingly, if we further increase the conjugated glucose number, the brain accumulation efficiency was dramatically reduced. Okay, actually, I'm not sure about this reason, but now we are thinking strong glucose binding to the brain blood vessel affect the negative impact on BBB penetration. For example, if our nanoparticle strongly bound to the blood vessel surface, it cannot be endocytosed, or it cannot enter the cell, or if even though nanoparticle can enter the cells, it cannot be released from the cell. So we are thinking these nanoparticles with much more glucose molecules should be accumulated in the brain capillary. Okay, so in that case, it cannot penetrate the blood-brain barrier to move to the brain parenchyma. And importantly, we observe the gene silencing effect by delivering antisense oligonucleotide. So this result is very consistent with the accumulation tendency. So we observe the highest gene knockdown efficiency by using 52 glucose conjugated nanoparticles. Okay, so the third topic is mRNA delivery. So recently mRNA delivery is quite highlighted as a next generation of nanomedicine. And recently lipid nanoparticles are very powerful for mRNA delivery. But we need to emphasize polymeric nanoparticle is also useful for mRNA delivery. But this is very preliminary result. We don't know what kind of polymeric structure is really useful for mRNA delivery. So our first study is kind of screening to pick up the better delivery chemical structures. Okay, so the important chemical reaction is shown here. So this is a backbone structure or precursor. And if we add amine molecules, we can readily introduce various structure into the side chain of polyaspartic acid. Okay, so by using this amino lysis reaction, we have prepared a variety of polymeric materials to optimize the chemical structure for mRNA delivery. And the important structure is this. This is a diethylene triamine DT, which work as a cationic unit to form a nanoparticle with RNA. So this unit can be used for mRNA binding due to the electrostatic interactions. And also this cationic unit can be useful for endosomal escape after endocytosis into the cells. But yeah, this is okay. But still we need a kind of additional chemical structures to stabilize mRNA loaded particles. Okay, you know, mRNA is quite fragile molecules. I introduced antisense oligonucleotide and sRNA, but they are how can I say, they are relatively stable compared with mRNA. So they don't need a special stabilizing approach. But yeah, mRNA is quite fragile, so we need special approach to stabilize nanoparticle or to protect mRNA from the enzymatic degradation. So, but yeah, we don't know which structure is very useful for mRNA delivery. So we tested various hydrophobic structures to form a stable mRNA-loaded nanomedicine or nanoparticles. Okay, the lower one has a higher hydrophobicity. So we try to optimize the hydrophobicity of polymeric materials for better mRNA delivery. Okay, this is a nanoparticle result by getting the DES measurement and the ELS, electrophoretic mobility, electrophoretic light scattering, ELS. And yeah, now we are focusing on NP5. And yeah, this is size, PDI, and the potential. And yeah, as you can see, we tested various polymer structure, but the resulting nanoparticle physicochemical properties are very similar. So almost 100 nanometer, and the PDI is around 0.2, and the positive zeta potential of around 20 millivolt or yeah, 10 or 20 millivolt. But the transfection result, in vitro transfection result or mRNA delivery efficiency was quite different based on the polymeric structure. So for example, this is P8. This one shows the highest mRNA delivery, cyclohexylated group, but this is PHE. PHE is this one, okay? So this and this, of course, they are quite different from the standpoint of organic chemistry, but yeah, from the standpoint of hydrophobicity, they are kind of similar, but the result is quite different, okay? CHE, this one showed much 1, 2, 3, 4, 5 orders of magnitude higher mRNA delivery efficiency was obtained from this structure, but not this one, okay? So this result clearly showed a chemical structure of polypeptide or polymeric materials quite critical for mRNA delivery efficiency. But of course, yeah, we want to know what is the critical parameter. And of course, hydrophobicity is very important. So we plotted this mRNA delivery efficiency against the hydrophobicity of polypeptide materials. And we clearly observe the kind of threshold in the hydrophobicity. As you can see, if we apply polypeptide with a higher hydrophobicity at around minus 2.4, resulting nanoparticles show the very good mRNA delivery efficiency. But if we apply less hydrophobic polypeptide materials, we didn't see any good mRNA delivery efficiency. So the hydrophobicity of polypeptide materials is quite important for effective mRNA transfection. And the reason why such a difference in mRNA delivery efficiency was further examined in cellular uptake experiment as consistent with this result. And the lower hydrophobic material nanoparticle didn't show good cell uptake efficiency, probably due to the unstable nanoparticle formation. So hydrophobicity is very important for making a stable nanoparticles, which can introduce mRNA effectively into the cells. And yeah, this is a kind of best one. In the first screening, we further applied for genome editing experiment. So actually, we deliver single guide RNA and SP-Cas9 mRNA for genome editing, and yeah, we used the transgenic AI9 mouse. If we use this transgenic mice, we can clearly observe the genome editing efficiency by evaluating the fluorescence protein expression. Okay, so if we successfully edit genome, we clearly observe the tomato fluorescence protein by reducing or cutting the stop sequence in the genome. And actually, we after intracranial administration of our nanoparticles, we clearly observe the significant gene editing effect around the ependymal layer around the surrounding ventricle. Okay, but of course, we need to further development for better penetrability of brain tissue. But anyway, this our nanoparticle shows a kind of very good diffusion in the ventricle, this region, to introduce genome editing in the kind of outside or distant site from the injection point. So if when we injected our nanoparticle here, we clearly observe the genome editing effect around here. But unfortunately, we didn't see any genome editing effect inside the brain. So the currently the tissue penetration of our nanoparticle is a kind of a problem, because in the first topic, we developed a very small nanoparticle with a size of approximately 10 nanometer. It's okay for better penetration, but now in this case, our nanoparticle has a size of 100 nanometer. In this case, size is a little bit large for better penetration. So we need to further develop a much smaller nanoparticle for mRNA delivery. But the problem is mRNA has originally has a larger size compared with the oligonucleotides. So now I don't have enough good idea to deliver mRNA into the deep brain tissue. It's a kind of future study. But anyway, we further develop a variety of polymeric materials for systemic administration. In the first screening, we clearly observed this CHE structure is the best one. So we slightly change the chemical structure. For example, this is C2, carbon 2, so carbon 1, carbon 3, carbon 4. So it's kind of fine-tuning of polymeric materials for better mRNA delivery. And yeah, this is nanoparticle physical-chemical properties. Yeah, they have a 100 nanometer size, and PDI is less than 2.0, and the positive Z potential of around 20 to 30 millivolt. So very similar, but even in the case of different side chain structures. But yeah, the result is also different. This is a whole, how can I say, mRNA expression in whole mouse body after intravenous IV injection. Okay, and interestingly, CHE polymer, which was selected from the first screening, this shows the highest mRNA expression even in the second screening. So we couldn't find a better one in the second screening, but yeah, we confirmed this CHE structure is quite useful for systemic mRNA delivery and also intracranial administration. But anyway, the main target is lung. Okay, you can see. So our nanoparticle shows a very high mRNA expression in the lung after intravenous administration, which mRNA expression profile is totally different from that by lipid nanoparticle lipohectamine sample. So in this case, lipohectamine shows a very high expression level in the spleen. Okay, but our nanoparticle didn't show high mRNA expression in the spleen. So actually, now I'm not sure about the reason, but mRNA expression profiles are quite different between lipid nanoparticles and polymeric nanoparticles. And this is in vitro and in vivo correlation in the lung expression. Okay, blue ones from in vitro cell culture experiment, and magenta from in vivo lung expression. So, and these expression levels are plotted against hydrophobicity parameter log D. And you can see, in case of in vitro transfection experiment, we don't need to care about the higher hydrophobicity. Even in the highest hydrophobic polymer, it can work well for in vitro mRNA delivery. But if we decrease, but in case of in vivo mRNA delivery, higher hydrophobicity resulting in the much lower expression level. So we need to carefully design a polymeric material for in vivo experiment. So we need more, this means higher hydrophobicity didn't work well for in vivo mRNA delivery. So now we found this range of hydrophobicity was very good for in vitro and in vivo mRNA delivery. Okay, this is a selected one, a best one from the first and second screening. And finally, we tested our polypeptide materials for kind of vaccination study, similar to COVID-19 vaccine, but this is a kind of model antigen of albumin mRNA were delivered by our CAG polymer with CPG oligo is kind of adjuvant for enhancing the immunogenic effect of OVA mRNA or OVA protein. And this is the immunogenic result. We observed the IgG, immunoglobulin G, G1, G2A expression level after delivering mRNA with or without CPG oligonucleotide. And we clearly observed the hydrophobicity is very important. CH 70. Okay, this is a very high substitution degree of CH molecules, cyclohexylated group. This showed a very good IgG expression or antibody inducing effect as a kind of vaccine. And unfortunately, lower substituted CH polypeptide didn't show any IgG production. So now we are thinking hydrophobicity of polypeptide materials are very critical for mRNA delivery and also for mRNA vaccination. But this is a very preliminary study. So we need to further confirm the impact of hydrophobicity on the mRNA vaccination. Anyway, now we are trying to transfer our polypeptide material technology to the society or to the company. So for mRNA delivery, we are collaborating with Blazon Therapeutics company, and also for protein delivery, we collaborating with a gene edit company established in the UC Berkeley laboratory. Okay, this is a summary of my talk. Today, we talked about 3 types of delivery formulation as a next generation of oligonucleotide acid delivery nanomedicines. First topic is a unit PIC, which has a very small size of approximately 10 or 20 nanometer for a better systemic targeting on pancreatic and brain tumors. And the second topic is a glucose conjugated micelle, which can penetrate the blood-brain barrier to deliver oligonucleotides to the brain. And the final topic is mRNA delivery by fine-tuning of polypeptide hydrophobicity to get a better mRNA transfection efficiency in vitro and in vivo. And again, these small nanoformulations are currently under clinical trials for breast cancer, sRNA therapy, phase 1, and antisense oligonucleotide to the brain, preclinical. And maybe next month, the phase 1 clinical trial will start. And this is final slide. So I really appreciate these collaborators and student and postdoc and staff in our laboratory. Thank you for your attention. That's all.
for attention. That's all.
Sorry. Thank you, Professor Kanjurumiaka. Your research, I think, is very important for cancer treatment. Now let's discuss. Yeah, I invite question from the audience, please.
Yeah, any question, any basic and simple questions are welcome.
Oh, yeah, okay, yeah. This is the question from Muhammad Faujan Lubis. Thank you very much for your nice explanation, Professor. Actually, I focus on testing plant activity against pancreatic cancer through in vitro test. After I saw your presentation, you should use pancreatic cancer in animal model, and it's so interesting for me. Could you give me a suggestion or something important that I need to do to reduce pancreatic cancer in animal model? This become a serious issue for me to create the models. Is the question?
Yeah, it's very good question because yeah, so you know there are many nanomedicine formulations, but most of cancer targeted nanomedicine clinical trials are failed, unfortunately. The reason why the model problem. So in many cases, we didn't use a good tumor model. So we clearly observe the very good anti-tumor activity in the mouse model. But even in that case, we didn't get a very good result in human cancer patients. That means we need to carefully choose animal models by mimicking actual human cancer patients. So now, yeah, important criteria is kind of fibrotic tissue. So today I used BXPC3 pancreatic cancer cell, BXPC3. It can make a kind of fibrotic pancreatic cancer model. So such a fibrotic stromal tissue is very important to mimic the actual human cancer patient. But of course, it's even in that case, it's kind of a model. So our initial paper has been published by Nature Communications, but the reviewer asked us to check more fibrotic pancreatic cancer. So you know there are more fibrotic pancreatic cancers. So now we are thinking we had better use more fibrotic pancreatic cancer model. So in my presentation, actually, honestly speaking, the mouse model is not the best one. But yeah, we don't have enough time to additional experiment. So we protect. Actually, we mentioned it's kind of future study to satisfy the paper reviewer. But yeah, we know we had better use more fibrotic pancreatic cancer. So now our collaborators or my previous my supervisor, Professor Kataoka, his research, Nanomedicine Research Center is using more fibrotic pancreatic cancer. Sorry, I don't remember the actual cancer cell line name, but yeah, we had better use such a cancer cell line to make a more fibrotic pancreatic cancer model.
Okay, thank you. I have a question. For example, if you want to measure the encapsulation concentration for nucleic acid, how do you measure the concentration, the nucleic acid? This is first question. And second question, since the cancer is, the cancer cell is, the surface is negative charge. And if you make a particle, we should always modify to positive particle, so the nanoparticle can easily enter to the cancer cell.
Sorry, you mean, first question, you asked a concentration of nanomedicine.
No, I mean, if you want to measure the encapsulation concentration in the nanoparticle, so how do you measure the nucleic acid concentration, or for example, RNA concentration?
Generally, we use a kind of fluorescence technique. So we can determine the number of fluorescence molecules in the kind of defined area. So if, for example, if fluorescence molecules, two fluorescence molecules are co-localized in the measurement system, that means encapsulation numbers are 2. But if we clearly observe the three fluorescence molecules from the 1 nanoparticles, the association concentration should be 3. So we, if we focus on the fluorescence intensity from 1 nanoparticles, we can estimate the loading number of drug. But unfortunately, if we, the problem is the fluorescence labeling may affect the nanomedicine form complexation. In that case, it's quite difficult to determine the drug concentration. So we need fluorescence labeling to determine the drug-loaded concentration.
Yes.
And sorry, what is the
second question? Since the surface of cancer cell is it? I mean the charge is negative, right? Negative.
Mm-hmm.
So if we want to make a nanoparticle, should we change to the positive particle using another?
It depends on the polymeric materials.
Yeah,
For example,
yeah.
In the last part, I introduced mRNA delivery nanoparticles. In that case, we didn't use polyethylene glycol chain. So the formed nanoparticle has a significant positive charges at around 20 or 30 millivolts positive charges. But in case of, for example, small nanoparticles, it can be comprising very long polyethylene glycol chains. So, of course, sRNA has negative charges and the polymer has a positive charges. So initially, they can make a complex due to the electrostatic interactions, but it has a kind of very long polyethylene glycol chains. So detecting surface potential or Z potential is around neutral. Is okay?
Yes. So I mean the surface of cancer cell, is it always negative?
Yeah, probably, yeah.
Probably, I see.
Yeah. So So in case of cationic materials, it can directly interact with the negatively charged cytoplasmic membrane. So we can get a very good transfection efficiency in case of in vitro experiment. But in case of in vivo, so such a positive nanoparticle can be bound to the negatively charged blood proteins to make a kind of protein corona. In that case, the surface positive charges can be canceled with the negatively charged protein in blood. So it can not directly associated with the negatively charged cancer cell. So that's maybe problem. So in that case, we need a kind of ligand strategy, as I mentioned, for BBB targeting. So of course, today our target is brain tissue or blood-brain barrier. So we utilize glucose molecules as a targeting ligand. But if our target is cancer cell, we can employ cancer targeting ligand for example, transferrin proteins or cyclic RGD peptide or any other antibody molecules to specifically target cancer cell.
Any question from the audience? One question more? Okay, maybe no more question. So thank you very much, Professor Kanjiro Miyata. Very nice presentation.
Thank you.
I would like to say hello to Aulia. She is now here.
Yes, Professor Hakim.
Aulia.
Yes, long time no see. Yes, long time no see.
Sorry, huh? Okay.
Can you hear me?
So, Dr. Aulia or Aulia Tan, she was my student during undergraduate research in USU in this university. So
She's
luck.
good.
Yeah.
She's working well in our laboratory, and now she greatly contribute to our research as a kind of course of bioconjugated paper. I'm very happy.
Yes. Okay. Thank you very much.
Thank you. You're welcome.
Thank you. Yeah.
Right. Thank you very much, Professor Hakim, for bringing the session. Also, thank you for the amazing presentation from Professor Kanjiro. The session was so informative and insightful for future research. All right, for participants, now our last but not least, the session with our invited speaker, Dr. Alexandra Rossi Paul, and this session will be guided by the similar person to the previous session. He is Professor Dr. Hakim Bangun Apoteker. So, all participants, once again, please welcome Professor Dr. Hakim Bangun Apoteker. Professor, time is yours.
Hello, Dr. Alexandra.
Hello, good afternoon.
Good afternoon. How are you?
I'm good, thank you. How are you?
Yeah, yes. Okay. The last session is will be presented by Dr. Alessandra Rossi Paul from School of Cancer and Pharmaceutical Sciences, Institute of Pharmaceutical Sciences, King's College London, UK. Dr. Alexandra got PhD from University of Kent, and she has many experience. Now she is a postdoctoral fellow in Nanomedicine, King's College London. Yeah, so she has many experience. Yeah, so maybe now let's begin. Dr. Alexandra, time is yours now. Yeah.
Thank you. I'm just going to share my screen. Hope everyone can hear me okay. Hopefully the internet holds up for us. That's not letting me. Okay, yeah, thank you so much for that introduction, and thank you so much for inviting me to present my work here today. So yeah, I'm Alexandra, I'm from the UK, and I'm currently a researcher at King's College London. And my presentation is titled Nucleic Acids from Drug Discovery to Drug Delivery for Cancer Research. So just a little bit about my background. Obviously I had an introduction just now, but I will tell you again. So I undertook my education at the University of Kent, which is an hour outside of London in a city called Canterbury. So I did my undergraduate degree in chemistry, followed by a PhD in chemistry, focusing on drug discovery methods. I was supervised by Dr. Christopher Serple and Professor Michelle Garrett, and I collaborated with Century Therapeutics, who are a small biotech company. So I will present some of my PhD work today, and then I will also present some of my postdoctoral work also. So then I started a postdoc in 2022 under the supervision of Professor Khulud Al-Jamal. I then was awarded the Mapplethorpe Research and Teaching Fellowship in October 22, which I'm currently still holding a post at, where I'm carrying on the same research, and I'm also teaching on the pharmacy undergraduate degree, and I supervise master's students on the pharmaceutical analysis program here. So I also present some of my work from this as well. So yeah, like I said, I'm going to present two different projects today. One is titled The Selection of Optimized Ligands by Fluorescent Activated Bead Sorting. This is my PhD work, and I'm going to go through every single step of this method that we created during my PhD. And this is the drug discovery portion of my talk. And then I will move on to a new aptamer delivery system transporting nucleic acids to T cells for glioblastoma immunotherapy. And this is the drug delivery portion of my talk. And this is my current work that is ongoing. And so it's not complete, but you'll see some of the beginning stuff. So selection of optimized ligands by fluorescent activated bead sorting is quite a long title. So we've shortened this to SOLFAB. This is the acronym that we used for this project. And we're using this method to select aptamers against the epidermal growth factor receptor. So firstly, just in case you don't know what aptamers are, they are single-stranded oligonucleotides. They're normally DNA or RNA, and they fold into complex 3D shapes. They can bind very strongly with their target with high affinity and precision. And because of this, they can be harnessed to be therapeutic agents or drug delivery vehicles or biosensors for diagnostics. So in this project, this is the parent aptamer that I used here. It's called Minio7. It is an RNA aptamer, and it originates from the University of Texas from the group Dr. Andrew Ellington's research group, and it was selected against the epidermal growth factor in 2010. And it has quite a high binding affinity for that protein. So the current aptamers that are on the market, so we have this aptamer here, which was approved for a therapeutic use in 2004. It is a treatment for neovascular age-related molecular degradation, and it's currently marketed by Pfizer and ITEC, and it targets VEGF, specifically this protein. And yeah, this is the current aptamer that is on the market. However, the global aptamer market size at the moment is $242 million in 2022, but it's expected to grow to over 500 million by 2027. And so this is quite an exciting area to be in at the moment, as people are putting a lot of money into aptamers as they have a lot of potential therapeutically. So generally, aptamers are selected against their targets by using this method called SELEX, which is systematic evolution of ligands by exponential enrichment. And so this, you have an aptamer library pool, you then have your target. So this could either be a protein or a cell. You then bind your whole aptamer library to this. You then remove the ones that are unbound. You then carry forward the ones that did bind. You will then use PCR to amplify these aptamers, and then you would carry on and you would do this cycle over and over again until you have a very top selection of aptamers that bind, and you would decrease the amount of target each time so that only the top aptamers are binding. You probably would also add in some negative selections as well, just to make sure that the aptamers are binding specifically to your target and not to everything that it finds. So this is the generic method that everyone uses to select aptamers. However, I'm going to show you today a new method which does not use PCR, and I'll show you that in a second. So just in case you don't know what the epidermal growth factor receptor is, it's a transmembrane protein, and it's a member of the EGF family for extracellular protein ligands. EGFR signaling is triggered by ligand-induced receptor dimerization, causing tyrosine phosphorylation. And because of this, it transmits signals through the membrane into the cell, and this causes the growth, migration, division and, of course, the survival of cancer cells, which we do not want. So EGFR has two conformations, this closed conformation and this open conformation, and it constantly flicks in between both of them all the time. However, sometimes it gets stuck in this open conformation. And when it is stuck in this open conformation, it is then constantly taking in more and more ligands. And when it's constantly taking in more and more ligands, it means it's constantly transmitting those signals into the cell, which means the growth of the cells is happening a lot, which is how we then get this cancerous and tumor growth from this. So EGFR plays a key role in tumor growth and development, like I just said, and the overexpression of EGFR is associated with many different types of cancers, but the main ones being breast, brain, and lung. EGFR is one of the most altered genes in cancer, and because of this reason, it's really, really important that we have EGFR targeted therapies. And you can see here from this pathway, it's really, really linked to so many other really key cancer mutated proteins inside the cell. So it's very important thing to target. So now I'm going to explain to you about this new method that we designed. So the step one of Solfabs is to find the best solid support to synthesize the aptamer library onto. Things that need to be thought about are the size, the rigidity, the chemical binding capacity, and also is it able to run through our flow cytometer or the fax. So originally I started looking at these glass beads, as a lot of people use glass beads to synthesize chemically onto, and they were 3 micrometers in diameter. However, they were not homogeneous in size. They were ranging from 1 to 30 micrometers. They were very fragile. When I looked at them on the SEM, a lot of them were broken. They were strange shapes. Not all of them were circular. So I decided this was not a good move to go down. So the next thing I then decided to look at are these Tentagel polystyrene beads. When they are dry, they're 10 micrometers in diameter, and when you put them in solution, they swell up to about 12, 13 micrometers, as you can see by this here. So it's still not completely homogeneous, but it's much better than the glass beads and a much smaller range of change in the size here. You can then see them here in this figure B under the SEM, and again you can see they look really similar and they're all circular, and that's exactly what we wanted. I then bound some rhodamine to the surface of them to check if they can be bound to, and this is them under the fluorescent microscope, and you can see every single bead has had the fluorescent tag bound to it. So this proves that they have a good chemical binding capacity. I then bound different amounts of rhodamine to the surface, as you can see by these different amounts here. I then run them through our fluorescent activated cell sorter. First of all, they run through fine, which means they're not blocking it, which is one of the things we need to make sure happened. We also can see them. They're very obvious and they're very fluorescent. And we can also see the distinguish between the different amounts of fluorescence I added to each one. So you can see that the 100% down to 0%, there's definitely a distinction between each amount. So this is good, and this shows these Tentagel beads that we've decided to use fit the categories of the solid support that we want. So the next step is to synthesize the modified monomers. So when modifying nucleotides, they can be modified in several different places. So you have this R1 position here, which is a modifying of the backbone of your RNA or DNA. Quite a few people do this to make them more stable, especially if they're going in vivo. And it's a good thing to do. Another thing you can modify is the sugar modification, this R2 position here. Again, a lot of people do this, especially if it's RNA, as the OH in this position in normal RNA is very easily degraded, and so it's very often modified in this location. For the mini 07 aptamer that I'm using, it has a fluorine at this position, and it's exactly for that reason, to stop it from degrading. But in this project, I personally am modifying the R3 position, this base modification, and people modify this to normally make the binding with their target stronger or weaker, or to change the shape of how the aptamer folds on itself. Also, you can add different groups that maybe will introduce different types of bonding, more hydrogen bonding, more halogen bonding, etc. So at the bottom here, you can see the 4 different modifications that we chose to use in this R3 position. So we have a phenol, and we chose this one because it's quite a large group, and so it causes a lot of bulkiness and potential steric hindrance, which will change how the aptamer is going to fold on itself if you introduce this larger molecule in this position. And the next one is a vinyl. Again, it's slightly larger than a hydrogen, so potentially it will also cause some steric hindrance. There's also extra hydrogens here now, so this could also increase some hydrogen bonding potentially between different nucleotides in the aptamer when folding. We then have added an iodo modification. You see quite a lot in drug molecules. They're using a lot of halogens, especially fluorine or iodine, such as 5,4-uracil. So we thought it would just be interesting to see this halogen in this position, and also this can create some halogen bonding as well. And then, of course, we have our classic, just non-modified hydrogen version as well to compare with all of these. So here is the general reaction scheme. I'm really sorry if you're not a chemist. I will go through this quickly. So we start off here. I add some protecting groups onto this. Then I add our iodine into this position here. If I'm then doing the iodo modification only, I will then skip straight to here and carry on. However, if I am then doing either the phenol or the vinyl modification, I will then add another step where I either do a Suzuki coupling to get the phenol, or I will do a City coupling to get the vinyl. After this, we remove the protecting groups I just added on. Then we add a different protecting group, which I'm going to call DMT. And in the next slide, I will explain to you why we have this protecting group here. And then the final step is to add the phosphoramidite onto the end, which is the phosphorus in the backbone. And this is how we join all of the nucleotides together. So once I've then synthesized the nucleotides that I want, I will then use them together to make the aptamer library. So I'm just going to introduce you to my oligonucleotide synthesizer here. So the first thing is we have these columns, and this is where my solid support is going to go in. So those tensigel beads that I've now decided are good, I'm going to put them into this column and put it onto the machine. And you can see it links up with these tubes where the solvent and solution is going to flow through. We have all of our reagents in here. So we'll have our nucleotides up here, and at the bottom we have our reagents such as our coupling reagent, our activation reagent, oxidation, etc. And you program the machine to put through different amounts of each one that you want in the correct order so that it couples together each nucleotide. And then I said about this DMT protecting group that we added at the end, and the reason we've done this is so that we can monitor the progression of the aptamer library synthesis. So each time it adds on a new nucleotide, it will then remove that protecting group. Once it's removed, this protecting group gives off a UV signal. So it will run through the machine, the UV will detect it. It will then give me a bar on this bar chart here. If there is no bar, there has not been a protecting group removed, which means the synthesis is not working. And so this is how we monitor the progression of the synthesis. So on the right-hand side here, you can see this is the reaction scheme of how we synthesize this aptamer library. So we add one nucleotide on top of the other using activation coupling reactions. And we would then cap off anything that didn't react so that it doesn't carry on reacting. We then oxidize this, we then remove the protecting groups, and then it basically just keeps circling all the way until the end. And then we'll do the final deprotection right at the end, and then that's the end of the synthesis. So we're making this library via the split and mix method. So the first thing I'm going to do on the tensigel beads, I'm going to add a photocleavable linker first. And the reason I'm doing this is so at the end of the synthesis, when I want to then analyze these aptamers, I can cleave them off the beads and I can use them for whatever I want. So with the split and mix method, you put the solid support into the column. It then will do each nucleotide in a row. However, when I then get to the uridines, which are the ones that I'm modifying, I then take off the column. I will split it into 4 separate columns. I will then put them on one by one, and I will run each of these through. So I'll add one into one, one into the other one, one into the other one, one into the other one. So that then each section will have had a different uridine modification added to it. I will then recombine all the beads back together, put it back on the synthesizer. It will then carry on doing its aptamer chain. The next time it gets to a uridine, the same thing will happen. I will take it out, split it into 4, add each one separately, put it back together, and then it will carry on the synthesis. So this is the only way you can get every single possible sequence possible from using these modifications in each location along the chain. There are 8 uridines in the sequence. It's a 48 ml long aptamer. So yeah, 8 different times I will split this out and do this one by one thing. So this then gives us over 65,000 different aptamers at the end of this, all the different possibilities you can possibly imagine. And every single bead will have over 1 million of these aptamers on the surface. But every single bead will have the same aptamer on its surface, and then each bead will have an individual different one. So once you've then made our aptamer library, we're then going to incubate it with EGFR. Our EGFR has a fluorescent tag on it, and this is how we're going to work out what has bound to EGFR and what has not bound to EGFR. So we've got the beads with the aptamers, and then we've got the EGFR is bound to the end. So our beads are going to run through our flow cytometer. Once it hits the laser, it will either determine whether it has EGFR because of the fluorescence, or it does not have EGFR because it's not fluorescent. The beads inside the water droplets will then be charged if they are positive, and will be pulled into the positive selection pot. The ones that are negative will then be pulled into the negative selection pot. So this is how we're going to sort out which aptamers bind to EGFR and which ones do not. So here are some of my gating strategies to show you. So this graph here is just the beads on their own, and you can see they sit quite low and in the corner as there's no fluorescence on these beads on their own. Round 1 here, this is the beads with the EGFR. And so we're hoping that, yeah, just to pull out the top one. So we don't want every single aptamer to be binding to EGFR. So we're just selecting this very top corner, the highest, the most fluorescent that we can find. These ones are then retained. These ones are kept but not used again. And then each round we get a bit more stringent with how much EGFR we're adding to each round, and we get less and less and less until we get to only 170 beads left. Once I get to this point, I'm then going to sort them. Instead of having them into 2 pots, I'm going to sort them into 96 well plates. So a single droplet, so like a single bead will go into each well, just one bead per well. So that's over 296 well plates. After we've got them into the 96 well plates, we then will cleave them from the beads. So like I said before, we have this photocleavable linker on the beads. So I will use UV to cleave this off. I will then transfer all of these aptamers from the well plates into individual tubes. So in each tube, there is one type of aptamer. There are loads of aptamers because there are multiple on the surface of the bead, but it should be one distinct type of aptamer on the surface. So the way we're analysing these to work out which aptamer is what and which modification it has in it, we are using LC-MS. So the sample will run through the column. I will then select the peak of my aptamer, and this will give me the mass of my aptamer. It will then get fragmented again, and this is how we will then get smaller fragments of the aptamer, and this is how I can then work out where the modifications are along the chain. So here's just an example of how this would look. So this is how it's going to go through the column. You're going to select this peak. It will then give us this graph like this, which is the total mass of the aptamer. This has then been fragmented again, so you're going to get this breakdown. And from this, I can work out the fragmentation modification peaks. Here is an example of one that I actually have. So it gave me a mass of 14,536. And this is the fragment pattern for this aptamer. So this method gives me a huge amount of graphs, so much data from each aptamer that we looked at. So I'm just going to show you one example, as it can be quite overwhelming. So at the top here, I have the full aptamer, this top line. Every time I've put a black line is where there is a fragment. So each black line has a fragment in it, and then underneath I've put where some of these fragments have overlapped. And we do want overlapping fragments, as this really confirms that this where the modifications are. So I analysed this data with a program called RoboOligo, which is really great to use. It looks very overwhelming and the graph looks very noisy, but it's because every single fragment is stacked on top of each other. So all of the fragments will be on the same graph. And this is why it's quite difficult to interpret. But you can see that you work it out by these different numbers. So C1, C2, C3, etc. Y1, Y2, Y3, etc. And so you can work it out by the mass of each bit, which lines up with all of these peaks. We cut them in these different places here. And this is how we can work out what the fragment masses are. So time on the mass spec, because I was doing this during COVID lockdown times, but I did manage to run some. And so I've come out with Minio7UA, Minio7UB, and Minio7UC. And you can see that some of the modifications are similar between each of them. So you can see there is a phenol modification in both of these two and also in the UB in the same place. UA and UB both have 3 phenol modifications and at least 1 iodine modification. So there's a lot of similarities between these two. And then UC has a couple of different modifications in different locations. You will also notice that there is a couple of nucleotides missing from the front of these two. So we believe this is down to a synthesizer error where potentially they got capped slightly earlier than they were supposed to. However, these have actually been selected as the top binding aptamers to EGFR. So even though there's an error in synthesis, maybe it's actually a good thing, as they are seeming to bind better to EGFR than the apparent aptamer. So after this, we then want to do validation of the affinity optimization. So I want to validate the aptamers that I found, and I also then this validates the method that we have designed. So I resynthesized the aptamers that I found, along with a couple of other ones. So I resynthesized UA. I then resynthesized UA prime, which is the full-length version of the one that I found. Like I said, it was missing a few nucleotides at the beginning. Then UB, which I found, UC, which I found, UC prime, same thing. It's the one with the full length. So the ones that got chopped, I added them on just to see if that made a difference. I then also decided to synthesize a fully modified phenol version, a fully modified vinyl version, and a fully modified iodo version. And so I modified all of the uridines with this instead of just putting it in exact places, as I wanted to see, is it the modification itself that makes it bind better, or is it the location of that modification that makes it bind better? I added a bitin onto the 5' end, and this was just to help me with my protein affinity assays, which I will show you now. So the way I did my protein affinity assays is I had a protein A plate. I then bound the EGFR to this. I did this by using an FC tag, which has very high affinity for protein A. I then added the different types of Minio7 aptamer in different concentrations to this plate. Because of the biotin, I can then bind a streptavidin phosphatase with a luminescent substrate, and this is how I can then detect the binding affinity by using this signal on our plate reader. And so from this, I made some binding affinity KD graphs, and then from here I can work out what the KD is. So I want the KD to be as low as possible, the lowest binding affinity. And we can see that the best scoring ones are actually the 3 that I found in the mass spec, which is amazing, which is exactly what we wanted. The ones that were found from the selection do have the best KDs, and the best 2 are actually UA and UC. And the worst ones is the phenol and the vinyl ones, which are the ones that I just decided to make to see, which is great that confirms that. So to confirm our experimental, we also did some computational docking studies. So we used Vienna RNA composer to generate the aptamers. We then use Avogadros to modify these. And after we've made the correct aptamer, we then used auto-dock tools to dock these onto the EGFR. So you can see the yellow is the parent, and then I've compared it against each aptamer. So the different color is the different aptamer going through. So the same thing as with the KD, you want your docking score to be as low as possible. And so from this, again, we can see that UA and UC have the best docking scores, as they are the lowest docking scores. So again, this really confirms my experimental, which is fantastic. You can see that the way that they sit is slightly different. So they're kind of pushed in a little bit further. And it's potentially to do with whether modifications are on the aptamer, that the aptamer is folding in a different way. And this is allowing it to interact a lot more with EGFR, which is exactly what we wanted. Also, the same as my experimental work is that the phenol modification, fully modified one, is the worst modification. You can see that this sits in such a different place compared to all the other ones, which have only adjusted like a tiny bit, but the phenol really did disrupt the folding of this aptamer. And so it really is important that the phenols are in the correct places and not just along the whole chain, because UA has 3 phenols. So it's to do with the location of those phenols and not just phenol itself. So yeah, the phenol one is the worst one experimentally and also docking wise as well. So our results provide proof of concept for this novel strategy for the selection of chemically optimized aptamers, offering a new method for rapidly synthesizing and screening large aptamer libraries to accelerate diagnostics and drug discovery. This method could be used for any target. You could do any chemical modification. You could have any size library. So this is a much larger project than just therapeutics for EGFR. And so I hope this part of my project has, this part of my presentation has proved to you that this method has a potential for the future in drug discovery. So we published this paper on this method in the middle of last year. So if you wanted to read more on this method, this is where you can find it. We also have 2 other papers that are about to be sent out to journals, which uses the same method, but for very different targets and for very different libraries. One of them is a peptide library where we've used the same method, the same sorting method, and we have similar really good results. We have one that is targeting RAS protein, which is a very hard protein to target and disrupt. So yeah, some exciting things coming. So hopefully this year those papers will be released. Okay, so I'm going to move to the second part of my talk now. So this will be slightly shorter. And this work is all novel and unpublished. So I would ask for no photos of the slides that I'm about to show you. That'll be great. Thank you so much. So this is, the drug delivery portion of my talk, presenting a new aptamer delivery system transporting nucleic acids to T cells for glioblastoma immunotherapy. So glioblastoma is the most aggressive and lethal brain tumor in humans, and the current survival rate is about 15 months. The current treatments are surgery, chemotherapy, and radiotherapy, but GBM is still incurable due to its resistance to standard therapies, and there is a lack of effective drug carriers able to target the tumor cells specifically and penetrate the tumor. So people have been looking at using immune checkpoint blockades. It's very commonly researched as a type of immunotherapy using monoclonal antibodies to block suppressive checkpoints or target co-stimulatory molecules to elicit an immune response. They've done this using molecules such as CD80, CD86, and OX40. This aims to increase the proliferation and activation of the cells. Using antibodies to target co-stimulatory molecules has been shown effective. However, there are limitations due to antibody off-targeting. So an alternative to this is to use RNA, such as siRNA and mRNA, and they've been used for such targets as CTLA4 and PDL1, and then mRNA has been used for things such as OX40L CD80 and CD86. ICP blockade therapy has shown effective in multiple tumors, however, has not yet been effective in glioblastoma. So nucleic acids to block or stimulate the immune checkpoints of T cells has not been very successful, which is why I'm aiming to do this with T cells. And so to overcome this, we believe that we need to design a new delivery system, as these RNA therapies need suitable carriers that will help us reach the T cells safely and in the correct destination. So there are a lot of different drug delivery systems that I'm sure loads of you know about, such as gold nanoparticles, lipid nanoparticles, micelles, magnetic nanoparticles, liposomes, and polymeric nanoparticles. But which one of these is the best to use, we don't know. At our lab in King's, we mostly focus on using lipid nanoparticles, and this is where our expertise are. So this is the project overview of what I'm trying to achieve. So I have two different drug delivery systems, this one here at the top and this one here, using aptamers as the targeting molecule to deliver mRNA. Both of these aptamers are targeting T cells in the GBM tumor microenvironment, and we're hoping that once they deliver the mRNA, this will stimulate an immune response and will kill the GBM tumor. So I have a T cell targeting aptamer, which is bound to a lipid nanoparticle carrying the mRNA, and I have a T cell aptamer directly bound to the mRNA. The mRNA that we are using is OX40, which has been shown positive effects on skin cancer, but has not yet been shown in glioblastoma. So these will either be injected intracranially into the tumor or intravenously. The aptamer will recognize the T cells and deliver more of this OX40 to these cells and hopefully deliver less to other cells. This will stop the loss of the mRNA and also some of the off-targeting effects that we see. The increasing the OX40 expression, we hope, will then make the T cells interact a lot more with the macrophages and the tumor cells, because of the OX40 ligand will be happy to interact a lot more with this. So this is what we aim to achieve. So step one in this project, I'm going to introduce you to Ala Zam, who is a lovely PhD student in our lab. She is originally from Syria, but she's now living in the UK. She did some research on immune checkpoints in the tumor microenvironment in BL6 tumors and GL261 tumors. So she's done some experiments on this, and then she's also compared this to published human databases. And from this, she's pulled out the top expressing ICPs in these tumors and across different cell populations. So from this, I've then picked 3 T cell targets that express those ICPs the most. So I spoke to you earlier about selects. So after picking my ICPs, I'm then going to do a selects with them. So I've got a 30 nucleotide randomized DNA library. I've got my aptamer targeting T cell receptors, ICP1, ICP2, and ICP3. So this will be separate rounds. I won't do them at the same time. So I will bind the whole aptamer library to one of the targets. The way I'm doing this is I have magnetic beads. I'm adding the protein to the surface of this magnetic bead. I'm then adding the whole aptamer library on top of this. I will then remove anything that is unbound, and then anything that is bound will carry forward, and I will do PCR, and then I will keep cycling around. Once I've done 8 selections of this, I will take them for next generation sequencing, and this will tell me what the sequences are. I also added in some negative selections, so I use other proteins to make sure that the aptamers were the top binding ones and they were not just binding to every protein that it found. So I did this for 3 separate ICPs. So once I got the next generation sequencing data, I then went through and checked the next generation sequencing count. The ones that have been multiplied the most are the ones I then move forward into our Vienna RNA modeling, the same as I did before. And then from that, I did docking studies similar to how I did in my PhD, using the AutoDock tools. So from the NGS count and from the docking scores, I then worked out what were the top 3 aptamers from each different ICP, and these ones went forward experimentally. So after this, I did some binding affinity assays. So with these aptamers, I ran them at different concentrations against different cell lines. I did the ICP protein, I did jacket cells, which are human T cells, and I did ex vivo mouse splenocytes, which have obviously different types of immune cells, but also they have T cells in them. And I also had a negative control cell line. So and then I ran these on the flow, and this is again how I made these KD graphs here. And from here, I could work out the KD. My master's student, Alexandra, who is from the US, she did some of this for her master's work last year. She is now currently a PhD student at the University of Sheffield in the UK. So this is a result of all of my binding affinity assays. And then from this, I picked out one from each ICP, which had the best KD in my positive assays and had the worst KD in my negative assay. And so these 3 highlighted in green are the ones that I took forward in vivo now. So we wanted to do T-cell aptamer biodistribution in vivo. So I have my mice, they are injected with GL261 cells. I then monitor their tumor growth over 10 to 14 days using our in vivo imaging system, the IVIS. Once they've got to the size that I want them to be, I then inject the aptamer into these, which has a fluorescent tag on it, ATO655. From this, I can then see this on the IVIS machine, and I can also then take the tissues and process these to look at them on the flow and to see the immune cell uptake of these aptamers. So I did them at different time points. I have 10 minutes, 1 hour, and 4 hours. And you can see on the left-hand side here, this is the tumors. So we use something called D-luciferin because our tumor cells, our GL261 cells have luciferase that they interact, and this causes us to have this signal. So this is how we can check that the tumors are still growing. And then on the right-hand side, you can see one has no aptamer injected and the other one has an aptamer injected. So you can see it even through the skin that the aptamer is there inside the tumor. It looks much better when it's just the brain on its own. So at their time points, they are then cold and the organs are collected. And so here is an example of some of the brains at different time points. So again, you can see 10 minutes, 1 hour, 4 hours. I actually have more data of this. I have N of 6, and I also have it for all of the aptamers, but I'm just showing you one example here of what we're looking at. And you can see a slight decrease in the aptamer over the time. And you can also see that the aptamer is kind of stays with the tumor, which is good and which is exactly what we want. Like I said, this work is currently ongoing. So then I can highlight to you the fluorescence of the brain on its own. So you can see the control is quite low, and then you can see the fluorescence over the different time points. It's interesting to see that across all of the aptamers, we are seeing a drop at 1 hour and then an increase again at 4 hours. And we believe maybe this is something to do with ICP recycling, that it's taking the aptamer in and then it's recycling it, and then it's taking more in afterwards, which is the reason why we're seeing it again at 4 hours. Because yeah, we're seeing this with every single aptamer at this time point. So this is interesting. But again, I have more data of this and we are doing more experiments. So we'll see in the future how this goes. So once I've then looked at the whole brain, I've then taken the tumor and I have put them into different cell types. So we have immune cells, tumor cells, CD4 positive T cells, CD8 positive T cells, F480, which is our macrophages, CD11C, which is our dendritic cells. So again, you can see uptake into all of the immune cells. And again, we have this weird drop at 1 hour, which we believe is to do with the recycling of these immune checkpoints. So I'm going to move on to our drug delivery systems. So like I said, we have 2. We have this lipid nanoparticle with the aptamers on the surface encapsulating MOX40. We then have this aptamer with mRNA direct conjugation via hydrogen bonding. I'm going to introduce somebody else. This is another lovely PhD student we have in our lab. Her name is Hiba Gaffer. She is originally from Sudan. She is a lecturer there at university, and she's currently working on the formulation of this. She's nearly finished, and as soon as she's optimized it, I'm going to be taking this in vitro and in vivo. So our lipid nanoparticles have an ionized double lipid, a phospholipid, cholesterol, PEG lipids, and of course we're encapsulating this MOX40. She's currently working on different types of chemistry of how we can combine this aptamer to the surface, and she's working on different ratios just to see which is the best one, what makes it the most stable, and what makes it the best to penetrate these cells. So my other drug delivery system is the aptamer directly conjugated to the mRNA. So how I've done this is called a sticky bridge. So we have the aptamer, which has the poly T tail, and our mRNA has a poly A tail. So they're quite long. So when they interact, these two just click together via hydrogen bonding, and it should not disrupt the natural folding of the other two, as the chain is so long that it's not going to interact with them. Then from this, we just remove the excess aptamer using a Zyphodex column, and then we remove the excess mRNA using that same bead method I explained earlier, the magnetic beads with the protein on the surface is how I remove the excess mRNA. So I optimize the conjugation of this, and my new master student is taking this in vitro now. So there'll be some data from this very soon. So I wanted to do some transfection studies of the MOX40 without the aptamer. So this is the basic level of just no aptamer on the surface. So I wanted to do this on a couple of different ways. So I've done it on the Jacket human T cell line, and then I've also done it on the ex vivo mouse splenocytes, and then I've also done this in vivo. So in my in vitro data, we can see a 10% increase of this OX40 expression in both the Jacket cells and the ex vivo splenocyte T cells. This is actually quite a good result. It's very, very difficult to transfect T cells, so seeing any change at all is a good start. And we hope once aptamer is on the surface of this lipid nanoparticles, that we will then see an increase in this OX40 expression, as we will not lose lipid nanoparticles to other cells that get taken up, and hopefully they'll all target the T cells more. So this is a good start from this one. And then in vivo, so I use the same model as before, the GL261 tumor mouse model. I'm then injecting these lipid nanoparticles. We normally treat them for 24 hours. I then take both the tumor and the spleen, and I process these and then run them on the flow also. So actually we see a better result in this. You can see, especially in the CD4 T cells, an increase of 20% in the expression of OX40 and about 10% in the CD8 positive T cells. I don't see much uptake from the other cells, which is also good. However, they do take in the lipid nanoparticles, they just don't get transfected with OX40. So again, this is a great start, and we hope once aptamer is on the surface of these lipid nanoparticles that this transfection will increase even more. This is the splenocytes from the same mouse. So we can see my error bars are quite large. Obviously in vivo you have quite large error bars, but we can see about 10% increased expression in both CD4 and CD8, and this links back to my ex vivo data. So it's good that I'm seeing ex vivo and in vivo to be quite similar to each other. So in conclusion from this, obviously this is still underway. We have 8 T cell targeting aptamers in a good KD range, and also they're looking good in vivo as well. I have transfection of MOX40 in vitro, ex vivo, and in vivo, and I'm seeing an OX40 expression increase of 10 to 20% across these. So in my future work, once the formulations are ready, I will be running targeting and therapy studies in vivo tumor mouse models, and we hope that the increase of this OX40 expression will get the T cells to interact more with the macrophages and the tumor cells, and this will hopefully kill the glioblastoma. Thank you so much for listening to me, and thank you to my mentors, Chris, Michelle, and Halud. And thank you so much to my lovely research groups and to my funding bodies here. And thank you so much for inviting me to speak. Does anyone have any questions for me?
Thank you very much, Dr. Alessandra.
Thank
Very
you.
nice presentation. Now we invite questions from the audience. Please, if you have any question. Dr. Alessandra, I would like to ask you. So you use lipid nanoparticle as a carrier, and the drug is mRNA, and Aptamar is as a targeting agent, and oncoprotein AGFR as a model target or as a receptor. So as we know, the RNA is the soluble in water. So maybe is it difficult to encapsulate the RNA in the lipid nanoparticles, right?
Is it difficult to encapsulate the mRNA in the lipid nanoparticles?
Is it possible? No. So the RNA in the surface, right? Not inside the nanoparticle.
So the mRNA is inside the lipid nanoparticles, and the aptamer is on the surface.
But RNA is soluble in water, so it should be difficult to encapsulate to enter to the lipid. Is it right?
We have a encapsulation efficiency of about 95% of our mRNA. We use different types of buffers to change the pH so that they get easily encapsulated into our lipid nanoparticles.
Is it the pH? I mean if soluble in water, it should be difficult soluble in lipid, right? So theoretically, in my understanding, is difficult to encapsulate in the lipid nanoparticle.
Yeah, we have no issues with encapsulating our mRNA inside of our lipid nanoparticles. The research group has been working on this formulation for quite a few years now. There have been some papers published on it, but yeah, we get quite a good encapsulation efficiency, over 90% encapsulated. It's because of the buffers that we use, so we're not using just water on its own. So we managed to change the pH, and so it manages to stay inside. Obviously, I haven't shared the ratios with you today, as this is part of somebody else's work as well. But yeah, we have the correct formulation that this manages to work. I think I have some messages in the chat.
Okay. There a question from the chat, maybe. Let's see the chat. This is the question from Adlina Aulia to everyone. Or maybe different. The question from Juan, Adlina Aulia. How is the overall percentage yield of aptamer synthesized? Number two, did you also evaluate the target binding specificity of the aptamers? Number three, in your ongoing research, why T-cell targeting aptamer showed accumulation in brain comparable to gastric and intestine? Thank you very much.
Okay, great. Thank you for these questions. They're really good. So the overall percentage yield of the aptamer synthesis is it's not always amazing. Each time you add a new nucleotide on your yield will decrease each time. So the longer that you make it, the worse your yield can be. But we're seeing from each round, we see an over 90% percentage yield, but obviously I'm adding 48 nucleotides on. And so each time, 90% yield actually is much lower than that. But this is also why we make the library so huge. And so there'll be several amounts of each aptamer, and so even if your yield is low, we will have covered every single sequence possible still. So yeah, we put in measures to make sure that even with a not a 90% yield at the end, we still have every aptamer. How did you also evaluate the target binding specificity? How did you evaluate? So the way we worked out the binding, we did a binding affinity assay. So I bound the aptamers again with the EGFR, and you do them at different concentrations from maybe like 100 nanomolar all the way down to like 0.01 nanomolar. And then from there, you can get their KD score, and this is how you know how well the aptamer is binding to its target. And then in your ongoing results, why T cell targeting aptamers show accumulation in brain comparable with so the aptamers are showing accumulation in the brain, I think because we're doing an intracranial injection, so they're not really going anywhere else and they're kind of staying there, which is good, and that's what we want. We see a high signal in the intestines, but that's actually because there's already a high signal there. So actually compared with the control, there's not a high signal at all in the intestines. But yeah, the interesting thing that to move forward with this, we would want to do an IV injection of these as well to see where this aptamer goes in the body. This is something that's currently happening now. Thank you. I hope I answered these correctly. I hope that's what you wanted to hear. There's another message from Mohammed.
Yeah, there another question. Please show the question. Yeah, from Muhammad.
Yes, this one. Okay, particularly the docking results of your aptamer. Did you have more data about the anti-cancer activity of the aptamers? Maybe like the in vitro against several cancer cell lines. Okay, so someone else did this. Like I said, this aptamer is from the University of Texas. So you can actually look up, if you look up Minio7, you can find that they have a thesis on this, and they did some anti-cancer activity of this aptamer against, again, like you said, against different cell lines. I don't think they did in vivo, but they might have done. I think they found the anti-cancer wasn't as good as they wanted it to be, even though the binding of the aptamer itself is really, really good. And so my plan for this aptamer going forward would be actually to use it as a drug delivery system as well, like similar as the research I'm currently now doing, because it is such a fantastic binder to EGFR. It just isn't producing the same level of anti-cancer activity as what we would want it for it to be a therapeutic on its own. So, or maybe you'd want to combine it with something else. So you could do this aptamer alongside a drug maybe. And so both together would give you that anti-cancer activity that you want. Yeah, that would be what I would do next with these. Again, I hope that answers your question, how you wanted it to be answered. Thank you for that question, Mohammed. Oh, there's another one. I think it's a similar.
Yeah.
Clinical test of aptamers as anti-cancer. Yes. So this is from Leonardi. Has there been clinical tests of aptamers as anti-cancer, and are there any side effects on the use of aptamers in treatment of cancer? Yeah, it's also a really good question. So there are a lot of aptamers, not these ones, that have gone through clinical trials for anti-cancer, or that they're in trials now. So there are quite a few, not for brain cancer though, for other types of cancer. There are side effects of aptamers. So similarly to antibodies, they can have off-target effects. However, the off-target effects are much lower than antibodies, which is the reason why people are starting to favor aptamers over antibodies, because of this exact reason, of there being less targeting effects. But there are still some, and this is a problem. And this is a problem I'm worried about in my research as well. So are my aptamers going to bind to more than just the immune cells? Are they going to bind to other cells as well? And I don't really want them to do that. So this is the potential side effects of aptamers. Thank you, Leonardi, for this. Again, I hope I answered that how you wanted.
Dr. Alessandra, I would like to ask you again. Aptamer is the anti-cancer or just targeting agent?
The aptamer, for my second project, the aptamer is the targeting reagent. In the first project I showed, people had thought it was anti-cancer. But in my new method, with the lipid nanoparticle, it's my targeting agent, yeah.
So you can both as anti-cancer and also as a targeting agent.
Yeah.
Okay. Yeah. Any question? Yeah. Thank you very much, Dr. Alessandra.
Thank you.
Very nice presentation. Good luck.
Thank you very much.
Thank you very much. I would like to say hello to Steven, Mr. Steven. He is here now.
Yep. Hi, Professor.
Steven.
Can you hear me? Yep. Hi. Can you hear me?
I see. No, I cannot see. Steven, huh? But no video.
Yep.
Yeah, Steven.
I'm in the lab at the moment.
Okay. Good luck.
Thank you.
Okay. Thank you. Yeah, that's all. Thank you. So I give again to the MC. Thank you very much.
Okay.
Yeah. Okay.
Thank you very much, Prof. Hakim, for
Yeah.
moderating this session. Right. Also, thank you for the amazing presentation about the aptamers, Dr. Alexandra. The session was so wonderful and insightful for future research.
Yeah.
Okay, I think now we have arrived in the end of our agenda. So let me to invite Mr. Mariadi, SFAM MSc Apotheker, to give a closing statement. So, Mr. Mariadi, time is yours.
Okay, thank you. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. Alhamdulillah, finally, the second international seminar on nanomedicine today has completed successfully. For that, as the representative of the committee, we give great appreciation to the keynote speakers and invited speakers. Thank you for sharing your knowledge and researches. Hopefully, it will be useful for all of us, and in the future, we can have another collaboration in various activities and research. Furthermore, we would like to thank. I would like to thanks to the participants who have followed the event until it was completed this afternoon. Thanks also for all of the committee members who have worked very hard to prepare everything so that today's seminar can be carried out well. Thank you to all those who have helped and been involved in the success of this event. Ladies and gentlemen, finally, we apologize for all the shortcomings and things that are not in place. I hereby represent the Nanomedicine Center of Innovation, Universitas Sumatera Utara, Medan, Indonesia, officially closing our event this afternoon. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank
Thank you very much,
you. Thank you very much.
Mr. Mariadi, for the closing statement. All right, all participants, before we leave this Zoom, allow me to remind you that you need to fill out the post-test, so you will be qualified to get the certificate after finish and collect your post-test. The post-test link was kept in the column chat so you can access it. And for your information, the certificate will be sent to you via email. All right, time check. It's already 5 p.m. and I think it's already time for us to close this seminar. Thank you for the best lecture from all speakers and invited speakers. We hope this seminar gives us extensive knowledge about nanomedicine. See you in the next international seminar on nanomedicine. Good afternoon and Assalamualaikum warahmatullahi wabarakatuh.
Steven udah keluar ya, Steven. Udah keluar Steven. Ya. Ini ujiannya sampai lulus ini kan. Kalau nggak lulus, ulang lagi ya. Iya kan? Kalau nggak lulus, ada lagi jawaban berikutnya gitu.
Prof,